Oh.
Oh my gosh.
Oh, you know what?
Oh yeah.
Yeah, let's.
Based on your preference.
Your
preference. Nice to meet you. Yes.
And how long will you be here?
Here,
All right, here, so maybe.
Yeah.
Oh, right.
That's support.
Think about it. Yes.
So
We also have pocket prints. I think yours.
Yeah. Oh, nice. Yeah, that's right. I forgot.
Yes, those should be all on. Okay.
Donna was concerned that
Okay. Good morning, everyone. Oh, that's loud. Sorry, it's gonna take a while to adjust. Welcome to our very first Microphysiological Systems World Summit. It's really wonderful to see all of you here today and be able to finally interact with each other, meeting in person in the beautiful city of New Orleans. Are you able to hear me well also at the end? Okay. Thank you so much for joining our today's roundtable, which is entitled Developers Meet Regulators and End Users. My name is Magdalena Cassandra, and together with Annie Moison, we'll be moderating this exciting discussion. As you will realize, in just a few minutes, we have assembled a wonderful and amazing group of panelists for you today who join us in New Orleans from all over the world. They come from United States, from Europe, and from Asia, and they are representing all of the major stakeholders in the MPS field. We have today at the roundtable developers of various MPS technologies. We have end users from academia and industry, and we have some members of the MTS community who bring the regulatory perspective from various continents. Our goal today is to discuss the current progress and remaining challenges in implementation of new alternative methods to replace, reduce, and refine animal testing. But really what we want to do today is whatever your role in the MPS field is, that you will come away with something, a new idea, a fresh perspective, and new strategy that you could apply to become more successful in your role in the MPS technology. So here is a format to give you an idea of what to expect. First, we'll take just a few moments for each of the panelists to briefly introduce themselves and tell us a little bit about their unique experience with microphysiological systems. Then we'll kick off the conversation. We'll ask just a few questions that draw out some major themes and trends that we should all be thinking about. And then at the very end of our today's time, we'll open up the floor to all of you to give you an opportunity to ask the questions that you were thinking about that maybe will help you to navigate upcoming opportunities and challenges in MPS field. Sounds good? Anyone has any questions? No? Great. So let's start with the panel introductions. Maybe we're gonna start from the right side of the table, Kaoru.
Hi, everybody. I'm Kaoru Sato from the National Institute of Health Sciences. This institute is a research center of Ministry of Health, Labor and Welfare, Japan. I've been involved in the project for regulatory acceptance of MPS and also in the project to develop blood-brain barrier MPS. Originally, I am a neuroscientist and the lab head of neuropharmacology. Thank you.
Yeah, hi everyone. My name is Adrian Roth. I work for Roche Basel in Switzerland. My current role is I'm Principal Director of Personalized Healthcare Safety of Pharma Clinical Development. Relevant experience in the field is mainly going back to my role before moving into clinical development. For 15 years, I've been working for Roche's preclinical safety organization, leading a team there in charge of cell models supporting drug candidate selection and characterization. And so as part of this roles, we looked into a number of different MPS systems and refined them, applied them. And actually also for my current role in clinical development, we're very interested in exploring ways how we could support personalized healthcare approaches using MPS.
Hi, everyone. I'm Tony Bahinsky. I'm currently the Chief Technology Officer for Vivadyne, and we're developing end-to-end automated high-throughput systems for 3D tissue chips. Prior to that previous experience with MPS, I was at the Wyss Institute for about 6.5 years. Helped develop a lot of technologies there, and was part of the advanced technology team that led projects with DARPA and the FDA. Also have 25 years of experience in the pharmaceutical industry. Most recently, prior to joining Vivadyne, I was with GlaxoSmithKline, where I was the global head of safety pharmacology, and we've tried to, as Adrian, integrate some of these systems into the safety side of pharmaceutical development.
So Susie Fitzpatrick, U.S. Food and Drug Administration. So 11 years ago, I worked with DARPA and NCATS to start this program on organs on a chip, never imagining how it would blossom and all of the really wonderful science that came out of this this little program that we started and the awards we gave to several of you guys, including the Wyss and MIT. Currently, I chair the FDA agency's alternative methods work group. I also am the chief toxicologist or regulator at the U.S. Food and Drug Administration Foods Program that are also looking at the use of chips for chemicals as well as drugs. And I'm excited to be here today and honored to be on this panel with all of my co-workers, co-scientists. So thank you. And very interested in what you want to tell the FDA. So.
Good morning. My name is Jan Lichtenberg. I'm the co-founder of Inspiro in Switzerland. I'm a microfluidics engineer by training, but moved into life science many years ago. We started Inspiro in 2009 with the idea to bring scalable organ-on-chip technology and scalable organoids and spheroids to the market to help improving drug discovery and safety applications. Our passion is kind of finding that balance between biological relevance on the one hand, but also then integratability, automation, compatibility for these systems to be able to really transform the way how we work with these devices and to be able to replace a lot of the current 2D and cell suspension culture with more relevant 3D models.
Hi, Nina Hobby from Albelix. I'm co-founder and co-CEO for this company. It's Swiss-based, it's a young company. And my background is in medical physiology and lung physiology. And as a PhD student, I always worked on advanced in vitro systems. And for me, it's great now to see that organs on chip and MPS has a real field that people making impact and that we really can do something for the future altogether.
Hello, everybody. I'm Danny Levner. I'm the Chief Technology Officer and one of the co-founders at Emulate. Like many of my colleagues, my roots are also in academia back at the Wyss Institute. But for the past 7 or 8 years, we've really been focusing at Emulate on bringing this technology commercially to the world out there. So I'm happy to speak about that as part of the panel.
Hi, my name is Q Beck, CEO of The Curator. As you may notice that I'm a last-minute sub for the newly. He's supposed to sit here as a panel today, but he somehow missed his flight. I hope that he can find a way joining the rest of the meetings. Anyway, The Curator is a spun-off from the Newly's lab in South Korea, and then headquartered in San Diego, California. We mainly focus on developing high-throughput MPS platforms for novel disease models in areas of the high unmet medical needs. Before joining The Curator, I used to work at the Vertex Pharmaceutical for various drug discovery programs. Thank you.
Excellent. Thanks, panelists, for the self-introduction. So we are ready to kick off this panel discussion. We have three main questions. I will raise a question, panelists will answer, discuss, and then it's open to you, audience, at the end. First question, are we ready to replace regulatory required animal studies with MPS? And I direct this first question to Tony, Adrian, given your experience interfacing pharma and regulators. So what do you think about that? Are we ready to replace animal studies with MPS?
Thanks for handing off that really simple question. I think we're close. I think there'll be on a case-by-case basis and depending on the context of use. For the study, I think we're closer on the efficacy side, especially where there are disease models that are not human relevant. I think MPS can play a significant role there. Biologics, antibody studies where there are not relevant animal models. Again, I think MPS can replace those and actually be used in regulatory packages. On the safety side, I think we're going to need to be a little more conservative. I think there'll be ancillary data initially, and then potentially, as they show their translatability to the clinic, they may replace certain studies for certain liver toxicities. But I don't know, Adrian, what do you think?
Do you agree with that, Adrian?
Yeah, no, thank you very much. I fully agree and would like to add that. Yeah, close is very optimistic if you really want to completely get rid of animal tests, particularly for drug development. But I think there are certain areas where we will step by step replace animal tests. I would like also to mention that in the end, what counts for drug development is of course patient benefit, and if MPS can show that we're generating more safe and efficacious drugs, then they will replace current tests, if be it animal tests or other types of tests. And so for some areas, I think we see nice success cases, and for others, there's still a little bit more work ongoing. I see particular opportunities for some of the newer modalities where maybe the spectrum of side effects to be expected is already a bit more narrow than for off-target effects with small molecules. So I see opportunities there that animal tests can be already at this point replaced with MPS models.
Yeah, thank you. Daniel, you want to comment?
Look, let me challenge the question a little bit. I think for one, let me divide the world between efficacy and safety models. With efficacy in development of biologics, there is certainly a lack of animal models, and there is a great need for organ chip models or MPS models. And maybe the question there isn't even replacing, because the good models don't even exist in the animal world. But focusing on safety, I'm not sure that we should be asking ourselves, when should we replace? Because it's actually an inflammatory question for many. For many in the regulatory world, for example, you're taking away this method has been trusted for decades, and you're taking that away from them. And that's not necessary, because the more data points we have, the more information we have, the better decisions we can make. Just like we don't make decisions with a single species of animals, we look at least 2 different species of animals. So too, we can add chips to this set of information to get a more complete picture. Now, with time, as we gather more information, more drugs, more confident system, things will happen, things will adjust. We'll refine and reduce the reuse of animals. Maybe eventually we'll get rid of one of the species. Maybe eventually we'll get rid of all the species. But the question of, are we ready to replace now, honestly causes an antagonism that's not necessary. We can coexist for the time being.
Thank you,
Yes,
Daniel.
Daniel, I fully agree there, because I really think the history of animal testing is so long, it would be now not fair to say we have to replace anything in 10 years. So we really have to build the trust. And it's great that we have a big push to replace. This is really needed, but we also have to take the time and maybe not already compare. Just saying, making a lot of data, store them, evaluate them, take the AI, and then go for a next step.
Yeah, that's one of side of the coins. Suzanne, could you please provide us with regulatory perspective?
Well, I wish we were done with animal testing, but I think we're working on it. There are no requirements at FDA that you have to use an animal test, and we're certainly open looking at alternatives and talking to our stakeholders about alternatives. And in addition to being open to that and working with people like you, people with CAT to look at alternatives, we're also making sure that our regulators are up to date on seeing these in an application. We have a lot of chips from different developers in our laboratories. We're trying to learn as much as possible about these chips. There are a lot of endpoints that we're interested in that do not have good animal studies. And so I think we're looking at some of developmental, neurodevelopmental, immuno as maybe test cases, but we're working on it. And we certainly feel like you that we think MPS can give us very valuable information as we put safe and effective products on the market, which is our main goal. And hopefully we're getting there. So.
Yeah, thanks. And Jan, you wanted to comment?
So I think from my perspective, the approach angle, and this is a bit along the lines what Nina and Daniel said, is first of all, finding unmet needs in the industry that are currently not properly addressed with animal testing. So Adrian, you mentioned novel modalities by specific antibodies, recombinant proteins, and so on. I think these are areas where animal testing, I think, rather poses a problem than an advantage. And I think that's where human-based models can really do a fantastic job. Another topic is patient diversity. And again, Adrian, you're looking at precision medicine and personalized medicine. I think we really need to encompass the diversity, ethnicities, but also different disease states more into our testing paradigms, not just for efficacy, but also for safety. And again, I think this is not really possible in the animal, but this needs scalable human models. What we, I think, for quite some while will not be able to deliver a systemic response prediction, because this is where animals are just the better solution at this point, unfortunately.
Yeah, actually, I joined this meeting as a regulatory person. So actually, I generally agree MPS is very good model, but in terms of regulatory acceptance, we have to collect much in-house data before that. If we could collect much data, enough data, the pros, a huge pros for MPS is we can study about the mechanism of the underlying mechanisms, and also we can use human specimens here. So yeah, but before regulatory acceptance, we have to collect much data from the in-house data in pharmaceutical companies.
So are you receiving this data? Do you have data to analyze, to review sufficiently, or is it something you would like to see more from a regulatory perspective?
Not yet. Not yet.
Not yet, not enough.
Actually, in Japan, an MPS project has now launched. So we are now searching for what is a COU concept of use. Do a pharmaceutical company have? Yeah. So yeah, maybe you guys have many good examples for that. So please let me know.
Yeah, I mean, that's a good question to ask. What is the general feeling about, do we have now data moving, to the preclinic and being submitted to regulators, or are we stuck somewhere? So.
Yeah, so if I go first, yeah, I think it's coming. There's certainly not a lot of examples there. However, I think regarding this discussion about regulatory acceptance, I think what we should remind ourselves is that there's only relatively few really regulatory required tests that need to be done in order to file in a new drug application, and those will be done. But that doesn't mean that everything else is not important. There's a whole lot of tests and experiments are being conducted, which in the end are not part of the regulatory documentation, but they are absolutely key for us internally to take decisions on molecules to move forward or not. So I think we should not be completely obsessed with the regulatory acceptance in terms of an MPS test is only valid if it has the status of a HERG assay or something like that, because that's a matter of maybe 20 years or so of validation.
So for sake of time, we take one last comment. Q.
Yes. So in the last 10 to 20 years, I know that it sounds a bit off track, but the last 10, 20 years, there have been a very successful case of the drug discovery, the programs that made no use of the animal models for the efficacy and the potency, like the cystic fibrosis 3 drugs. And also, in this morning, they announced that some of the data for that pain program for the NAB 1.8, they didn't use the animal models for the efficacy and the potency at all. So I think with a specific understanding of the disease model, disease biology, and also the judicious selection of the target, and if we are using that some near the physiological the model, the MPS, driven, I think maybe that we do not need the animal model down the road for that.
I think hearing more about case studies, like examples from all of you. We don't have time, unfortunately, this morning, but perhaps in the coming days. But that'll be very insightful, I think, for us all. And it's time to switch question. No?
Yeah, now we're gonna focus on the challenges, and I would like Jan to take up the first question or the second question or the roundtable. How can we clear the main obstacles to wider adoption of microphysiological systems?
Mm-hmm. Thanks, because this is, of course, something that I think drives the whole developer community a lot, right? So there is a clear need for better model technology, but I would still say that the MPS systems, independent of whether it's spheroids or full-blown organ-on-chip systems, they still kind of scratch on the surface. And I think there are a couple of reasons. I don't believe that regulatory acceptance is really the key here, because the driving factors in the industry are reducing attrition costs, being more efficient in developing new drugs, maybe addressing specific therapeutic areas that are currently not covered by therapeutic paradigms. We are working heavily in NASH, for instance, and there's really no drug on the market for this. So the important obstacle that we see is the lack of confidence yet in these new technologies, not just from a topic of biological relevance, but also how does it perform in the lab? What is the cost of ownership? Can we use this broadly? And there is a plethora of solutions, from very simple automation compatible solutions to quite complex ones where you need dedicated devices. And I think we as developers, we need to be work very closely with end users to make sure that the advantages are understood and that the context of use is understood, because not every system is equally useful for any application in the drug development chain.
Thank you for that. Nina, could you speak to the challenges of your own company?
Of course, happy to do so. So I fully see also we really have to take the risk to go very early already to the end user and establish something that they really then can use also for applications. So in our company, we really have the benefit that anything that we do is mostly in the preclinical phase. This means that it's very exposed. You get a lot of feedback, what is working, what is used. And what I would hope for is also that maybe it's somewhere tracked if, for example, an end user used MPS in the application, somehow that you can make a cross during application, that it's easy then for the regulatory to see who used it, who doesn't use it, and also got the feedback, what was working and what not. And also other end users can then use the same process. Also in regarding of price, I think this is a big limitation still in MPS. as more it is used, as cheaper it will get for the developer, so we can offer it to a better price. So anything that is validated and accepted, we know we can do it next time the same. We can make better prices for the next customer or for the next end user. And the third point, which I think it's personally very important, is also to give access to students. So with big core centers, that already in the education, people know that MPS is something which they can use later than in their career.
So how are we going to support that? Do we need philanthropy?
How we support this? Yes, I would really, this is very, very visionary, I would say, maybe also a budget price-wise. But I think it would be really good when using MPS, because currently it's volunteering. People use MPS on top because they have no other option, or they think, okay, we start with the MPS on top with the animal testing, so that they can somehow reimburse part of these costs. Because this, of course, would foster that people say, no, it's not on top. I have more data for the same price. And for the education, of course, we should have facility centers.
Thank you.
Yes, I think there is no single silver bullet to overcome all the obstacles, because the different beneficiary group of the MPS is facing the different obstacles. So I think in general, the first most important thing I believe for the pharmaceutical company is to provide that the MPS very compatible with that the high throughput, the comparability, the instrument and liquid handler, and the high throughput screening system as well. And also that's addressable by the very standard instruments. I think that is one of the key things. Then there is another issue of the robustness, because that usually pharmaceutical company cannot tolerate that sort of the failure rate that is okay in the academic settings. So in short, I think it is very important to provide that high throughput compatible and also the robust enough to be widely used.
Maybe we can get the take from pharma. Do you agree?
Yeah, sure. I think the robustness is a key one there. So knowing that there's good translatability to the patient, if you can get biomarkers and endpoints that translate directly to the clinic, I think that's really important. Or you can have surrogate endpoints that's important also. But I think the ones that are really holding it back, especially from pharma, are the scalability. We're still very low throughput on a lot of these systems that are available commercially or even in the academic environment. I think you really need to scale it up and actually be able to do tons of experiments and different types of variations on the cells, the conditions to get optimized systems too. And the systems now are just too slow. Reproducibility is a big thing also. You want to make sure that somebody in the same lab that's doing the same procedure can recreate that same experiment and the same tissue type and get the same endpoint and results. Even more important, we can't even do that really reliably, let alone at another site or in another country across the ocean. So I think that's really key too. Automation can be a key to both of those things. One, to help with the scalability, and then two, with the reproduction. If you can automate a lot of the tissue culturing and production of the tissues, I think you get that, remove some of that human component, human error from the systems.
So Jan, you are on the board of Society for Lab Automation.
Yeah.
Can we hear from you? Should we be developing additional instruments or make them adaptable?
I think this has been a big topic at SLAS. How can we automate this? Because ultimately, you're totally right. For a lot of applications, this needs to be throughput compatible. Also, if you want to use the same models from, let's say, secondary screening to target validation to further investigation, if you switch systems, this always introduces certain question marks, which are not necessarily helpful in the process. So if you want to pull this through, you need solutions that are robust and scalable. And I think this is a general situation that we have in our domain. Everybody knows the hype cycle curve, right? So I think we are a bit past this in the organ on chip and MPS area. There was maybe too much promise and then a certain under delivery on the results, because ultimately a non-scalable system that creates a great result is kind of leading us a little bit into a dead end. We always have to keep the industry interest in mind to create something that works 24/7 in an automated lab environment. And I think this is what we've seen also in our industry from a developer perspective, moving more into under-promising and over-delivering than the other way around.
Do you think we can catalyze wider adoption by making those models perhaps more affordable for a wider community?
Personally, I think, and I agree with Nina, so scalability also comes, or price goes down with economies of scale if the technology permits this. And so a wider use of the technology is definitely going to help not only the supplier of a specific solution, but also other suppliers, because in this case, the flood raises, I think, all boats. And I think this creates a benefit for everybody. I think what we could use from a regulatory perspective here is help in terms of developing maybe not validation, because this is already a very high bar, but qualification requirements. How should we characterize the model in terms of its cellular composition, in terms of the mechanisms, biological mechanisms that are built into that model? What are compound sets that we can use for benchmarking? Not to single out a specific vendor, but for giving everybody a certain peace of mind that a certain technology lives up to a standard that is required. And I think that could help us a lot.
Want to comment on that, perhaps, Kaoru or Suzanne from regulators on this wider adoption? What do you think would help smaller companies perhaps also to?
I agree. I mean, I think I don't see any regulatory barriers to FDA accepting an alternative method, MPS or otherwise, if we have confidence in its context of use. And I think one thing FDA wants to do, we are looking at how you decide relevance in that, and that's something I think we have tried to do in partnership with our stakeholders, because we all have the same goals in mind. So it's not just our question, or not, it's where we all work together. We're working a lot now with the IQ Consortium, MPS, to talk about standards. We invite any people with new methods to come in and present them to the FDA through our program, like once a month. And occasionally we'll adopt one of those and bring it into our laboratory, all in an effort to really understand, like you, what we need to move these forward to get more, because we want more predictive tools.
So that brings us very well to the third question, Suzanne.
Pardon?
That brings us very well to the third question, if I may switch to the.
Okay.
No, that's not what I'm saying. Just for the audience and sake of time, I'll just like verbalize the third question, which is very relevant to what you're saying. So where do you see opportunity for collaboration to support MPS development, adoption, and acceptance? So you can continue.
Going.
Yes. No.
I see working together. When I started at FDA, we were on one side of the table and everyone else was on the other. It's not like that anymore. We all want the same thing. I think it's not just us we're not actually, I don't think the regulatory block, it's the industry being worried about putting new technologies in and what are we going to say about them? But I think that means we just need, like this meeting here that Kat has organized and that Dan is, this is where we all talk together about what we see, how do we show relevance? How do we show robustness? Give us your ideas, because we're working on a lot of things and we want to hear from those people working on it. We have chips in our lab trying to learn this too. So I think it should be considered a partnership to move these exciting methods, which we helped start. And so we are invested in at FDA, move them into good tools to put better products on the market. And ideas, we have a website, we have alternatives at fda.hhs.gov. Send in your ideas, send in anything, proposals for workshops if you think it'll advance the field. We are very open to doing this.
So Jan and then Kaoru.
Just maybe quickly, and I fully agree, like iStand, for instance, is a great initiative in that direction. And I think we have a good example that we started last year. Our interest was in reducing not replacing, but reducing animal testing in the safety space. And so we developed together with partners a set of liver microtissues from different relevant species in the preclinical space so that we can test species-specific tox effects early on. And instead of doing this just inside Insphere, we decided to team up with a lot of pharmaceutical companies like Genentech, AbbVie, Sanofi, and many more that are all chipping in, that provide compounds that had species-specific toxicity, but not in man. And these are exactly the examples that we're interested in. And so we brought in academics to help us in that project. And we also then sent our feelers out to FDA using iStan so that we can really create kind of the full package, the information that's necessary, but also get the regulatory guidance early on. And so this has been so far a great experience for us.
Kaoru, I'd like to hear you from the Japanese perspective. Huh?
Yeah, actually, I've been concerning what kind of regulation is suitable for MPS. I mean, MPS is comprised of many component technologies. I mean, the cells and device itself. So what kind of regulation is the shortest cut to acceptance for everyone, acceptance by everyone. So I mean, for example, MPS package. I mean, MPS test itself, regulation is appropriate, or device itself, or cell itself, and which is better for the shortest. I'm not sure at present, but if you have any opinions, please let me know.
And do you see a good collaboration between maybe like the developers, users, and pushing that to regulations? How is the collaborative landscape over there?
I don't know. It's I mean, I'm talking about the quality of cells and devices, and also the MPS, resulting MPS, the quality of the MPS test is enough to approve the new drug. I mean, this is my core question. I mean,
maybe we could learn from.
Okay. The device itself is very.
That's an effort. Should be a collective effort
to get the robust
quality
results. So,
okay,
maybe we could learn from the FDA of how to build the partnerships of this type. Suzanne, could you please comment how to build the partnerships of this type between all of the stakeholders to make it really work? Suzanne,
I'm not quite sure what the question is, but I have a question for the
So
panel.
how to build the frameworks of this type that connects all of the stakeholders together? You have built multiple, right, with DARPA and CATS.
By having some money, obviously. We brought all the stakeholders together, didn't we? So, I want to know what the panel thinks. Do you need animal chips to show concordance to human chips to show concordance to humans? That is a debate that we have going on, and I'm just curious about what everybody here thinks about that.
Nina and others from your side in collaboration.
I mean, is that how you're going to get them into regulatory use? I mean, I have feelings on it, but.
Yeah, I think Nina just wanted to comment.
It's okay, Adrian can start.
No fight, but ladies first, then we go to
It's a hard one.
Ladies first?
Ladies first.
Okay. No, it's an important question. I think there is a lot of pressure in this direction, and I'm actually not really a supporter in this. I really think we have a lot of gaps to solve. So the first step should be what we cannot do in animals and work on this, because then this part will be really nicely settled with MPS studies. And then later, when we have the history in MPS, that we then maybe start to compare. But currently we have so many gaps to solve, which is an animal testing really don't help us, that we focus first on that.
Adrian?
So Yeah, I do see a number of use cases where it makes a lot of sense to have also animal chips, if it's about, you know, showing or absence or relevance for human, if there has been a finding somewhere in a preclinical animal test, and so these kind of exercise. But generally, I don't think it should be the general path to validate these kinds of models. It can help to validate the approach or the technology as such, because animal data tends to be more standardized. You have a lot of data to benchmark with, but I wouldn't go that route for every type of assay and application. If there's solid, good human data around, go for that.
Jan?
Yes, I also wouldn't use or wouldn't create an animal system just for the sake of showing the relevance of a specific technology, because at the end of the day, also the cell isolation and the cell source plays such an important role. Ultimately, it's about predicting clinical outcomes, right? That's what we're all interested. But we see that animal tests can be a substantial problem for pharmaceutical developers, right? If you have a signal in a specific species, it typically sets back your program by what, half a year, right? So this can be really expensive. And so being able to assess these risks early on and maybe already start to look into mitigation strategies before you go into an animal, I think it's a perfect reduction strategy of animal testing, but at the same time, it saves everybody a lot of money and especially time to get this drug to the market. And that's kind of where our interest comes from.
Nina?
Yeah, here a little bit an add-on also, because making an animal on the MPS is a lot of work, but you still have the whole data that anyway comes from the animal testings, which has currently to be done. And there, for example, we see a lot of alignment. So you still would have the feedback that your human MPS gives the same signals than the animal MPS, but it's not needed that you establish everything in the chip.
Could the real world data and AI help? Tony, what do you think?
I think we bandy about AI as the cure-all for everything, and I'd be very cautious with that. It can help. I mean, you need to first generate high-quality data, right, to feed into the systems. You need to be able to do that at scale, because these are very data-hungry paradigms. And then, you know, hopefully at the end, if you do the right modeling, either you want to call it machine learning, neural networks, AI, you can probably develop, you know, certain fingerprints and understand you know, where you're progressing towards either an adverse effect or potential efficacy. But I'm always kind of a little cautious now. You know, it's like we're kicking the can down the road. Oh, AI will solve everything for us. And I think we're being a little naive that way.
Quick comments for the AI is, I think MPS provided a very complex data set. I think it is impossible to choose at the right parameter, you know, down the road. If you see something that for that, you know, to find that correlation between the clinical response. So I think AI definitely going to help for that to find that right parameter for that data analysis.
Good. So we need resources, collaboration, and resources should go to human-based system, not animal-based system. I think that's quite a strong message and alignment we have here. That's good news. And now we have time for Q&A.
Yeah, we can open up the floor.
Go ahead, Magda.
To all of the audience, but before we're gonna do it, we would like to take the temperature of the room and better understand who the audience is. So everyone who is now developing new human cell models, please raise your hand. Okay, great. Quite some of you. Who are the end users of the technology that is supplied by other developers?
End users, okay. That's good.
Right.
You can be both, no problem.
Yeah. Who is interested in regulatory perspective, who is on the way to actually approach the regulatory acceptance? Great, perfect.
Can I ask who is actually a regulator? They're shy. I've seen a few hands. Good. So we probably need to have more from regulatory agencies joining us in these meetings. That would be very, very helpful and interesting.
So anyone would like to kick it off with the first question. You can just walk up
Go to the mic.
here
Yeah.
in the middle.
If you can just tell your name, please, and ask your question. Thank you.
Christine Mummery from Leiden. I just wanted to follow up 2 points that were made. I think one of them was by Jan. One of them is, how would I know whether my model's any good? And one of the things you can do is perhaps see or define a set of regulatory or a set of compounds to which the model should respond. And you could base that response on knowing what happens in patients or in humans. So we've already done that for the heart. We have 12 compounds we call diagnostic for a model. That's a stem cell model of the heart, in fact. And I think Lorna Hewitt's done it for the liver. So that's probably the way we could go with some organs, at least, define a diagnostic set of tools. And the other point is a somewhat sobering thought. We've done a meta-analysis, or we're still busy with it, of the literature. And whilst for organoids, about 10% of the total number of publications are of reviews, the rest are primary papers. For organs on chip, there are fewer papers, but a third of them are reviews, which means there's still more promise than there is actual hard data. And I think there are a lot of researchers here. I think if we're going to make the field successful, we're going to have to produce a lot more primary data to see whether it's actually going anywhere. So thanks.
I fully agree with the latter point, right? So more data is definitely necessary, but I also think more data sets. So I fully agree with you. There are a couple of compound sets that have been developed by the IQ Consortium and others as well that help us in benchmarking. But I think we need to agree on let's say, a more holistic view on requirements to assess these models. For me, this would be 4 topics. One would be cellular content, right? What is in the model? If you talk about liver, is it just hepatocytes, or are we looking at Kupffer cells? What's happening to the innate and other immune system, right? How we approach that? Do we need it? Is this optional? Second point would be readouts that are available for it. Third point would be what kind of biological mechanisms are visible in that system, right? How can you elucidate that there is a certain response to a specific challenge, and that is not necessarily a drug, it could also be something else. And then the last point, this is exactly what you say, what is a useful compound set and what is the proper benchmark against it? And that compound set should reflect different modalities of compounds, not just small molecules. And it should reflect the different mechanisms of toxicity, for instance, in the liver safety model. And I think if we could agree on that from an industry perspective, end users, developers, and ideally also regulators, I think it would really help us to clear up the situation.
If I could follow up on that, let me connect a few of the questions that were asked here about barriers, about collaborations, about the compound sense for qualification and so on. This community has been working hard for the past 10 years, even more, to produce tons and tons of data, lots and lots of papers. We've mapped out how many papers are in the industry, and the numbers are staggering. And in terms of promise of the technology, we're there. I think everybody believes that there's a lot of promise here in terms of showing, use cases and examples and case studies. We're there. There's a lot in terms of science. But what we don't have yet is any clear sign of adoption. And I claim, my claim is that as soon as we have some very real adoption, we'll open the floodgates to everything that's happening here, right? We're still stuck in the science stage, and we need to get into the adoption stage. Now, I think that the qualification, those compound sets and so on, is one of the main elements that we need in order to drive that adoption, because we need to get into the shoes of the end users of the technology and convince them that the technology does not just what we think is interesting enough for a first author paper, but what they actually need to use on a day in, a day out basis, including the cost, including the throughput, including the reproducibility and repeatability, but most importantly, that we actually solve the problem that they're trying to solve. And that brings me to that collaboration question, which is we need to get together around this, because up until now, I think we've all been trying to show the breadth and the depth of technology in terms of science. If we can drive this adoption, it will really shift this industry.
We have a question here from James Hickman.
So, how you doing all? Probably know most of you up there. But Jay Hickman from Hesperos. I wanted to talk about the regulatory aspects because most people don't understand this. There's a really big difference between safety and efficacy. Okay, and the bar for safety is going to be much, much higher than it is for efficacy. We've already we supplied the efficacy data for a drug with Sanofi for an IND that has already enabled authorization of a clinical trial that's been ongoing since April of last year. So that's already been done. What was for efficacy? Efficacy, the bar is lower, okay? Safety, the bar is much higher because the FDA does not want people dying, okay? And this is, and Susie, that's the main difference here is between safety and the efficacy. I mean, Jonathan Goldschmidt, who used to be head of rare diseases at FDA, got up at a meeting we had 4 years ago and said, look, as you already mentioned, Vertex, okay, that was the seminal moment at the FDA in terms of Vertex had gotten a clinical trial approved for 12 indications for cystic fibrosis, 12 mutations. Then they got 24 mutations, then approved with only in vitro data. He said, we've already established the precedent, okay? And so we've been following that at this point, and like I said, we've already got authorization of a clinical trial, okay? And I'll show the data in my talk, which is right after this, okay? But that's the big difference. It's safety versus efficacy, and too many times people mix them up together, whereas getting things for regulatory approval through with the efficacy is much lower bar than it is going to be for the safety.
Maybe
there's a comment on, well, comment, and then I go question to Jason.
Our panelists comment.
So, I think it's a great point, Jim, and the publication that you guys have on this, I think is also leading the way. We've contributed data for IND filings and for FDA responses as well. I think the problem is not so much the openness of the FDA to look at this data, right? And I think Susie says very often we are very open to this and can become a little bit angry when people put this in question, right? I think the FDA is open to it. I locate the issue more on the side of the pharmaceutical developers. So Jim is working with some that are very open to bringing this in. Not everybody is open to that. And there are some concerns of adding a new dimension to the data set. And that's something that we need to alleviate.
Yeah, hi. Yeah, Jason Nickett, I lead the CIVM group at GSK. I thought just to add to James's comment about past chair of the IQ MPS affiliate. So we've usually broken it up from a context of use into safety, ADME, and then pharmacology. So leaving efficacy out of it because of pharmacology, where we'll see it as what's the dose prediction. So there's another realm of breaking it up even further. So that's probably one area to think about when from a regulatory point of view. I think the other thing is from adoption point of view, from your comment, Daniel, is trying to demystify what MPS organoids are when bringing them into pharma. So it's working with the bench scientists to help them figure out how to use these systems in the day-to-day use of it in the lab, and also how to establish it with already established protocols. So that's something also tissue chip developers have to think about. So I'd be interested what's your thoughts on how you're going about doing that?
Daniel?
Well, let me start with the efficacy question. We totally agree that the thought process around efficacy and safety are different. And one of the you're right. There's a dichotomy, something that we need to wrap our head around, right? In efficacy, there's a lot less legacy. There are no good models for many models, especially when you're developing biologics. But on the other hand, efficacy models tend to be somewhat bespoke because every company, every group is working on different indications, and there's a chicken and the egg problem, which is you don't start a drug program around an indication you don't have a model for, right? So it really requires very intimate collaboration to get that company to start developing something around your bespoke model. Safety and ADME are much easier because everybody's doing the same questions, which is why we find it a lot easier to figure out what products are necessary when we think about it in terms of products and applications on that space. So this is a question I actually challenge this whole group. How do we get into efficacy where the problem is great, the the low hanging fruit is there, but there is the chicken and the egg problem. Perhaps it's about fostering relationships with those people that are in the right place, right at decision points where drug development programs are started, to challenge them to start adopting these models early.
We have the time for a very last question.
Yes, hi. Frank Borelli from the Humane Society. From a toxicological point of view, I've heard some of the concepts mentioned in passing, but not necessarily discussed in depth. Concepts such as validation, relevance, harmonization. In the past, we know that in order to gain regulatory acknowledgement, some of these programs will have to be coordinated, especially validation programs. I understand that the field is still in a developmental stage. However, I don't think it's too early to start thinking about harmonization and validation programs, which will require a concerted effort from many stakeholders. I'm wondering what your comments are from the panel. Thank you.
Anyone would like to take up this question? Harmonization, Tony?
And maybe Susie can elaborate on this afterwards, but when you submit something to the FDA, I mean, so there's several processes. One is the qualification process, right? So you have a standardized drug development tool, and that's one way, and that's the much longer road. But if you are a sponsor and you're bringing a program in, if you have good internal data for that validation of whatever that assay is for the endpoint and predictivity, whether it be efficacy or safety, the FDA will consider that and utilize that in the IND package. Right? It doesn't need to be a qualified drug development tool. And I think that's where we get confused here. We spend a lot of time and across the industry, I see all these consortia where, we're developing, and we still haven't made a lot of headway, because we haven't come to a consensus over the last 5 years on developing these paradigms and the parameters that we need for qualification. You don't really need it. I mean, you can actually go to the FDA if you've generated that internal data to validate your own assay and bring it forth. So,
But it's not public.
Right. Well, that's right.
It's not public. It belongs to the company, and if they decide to share it, it's their decision. But yes, you can bring anything as part of your internal discussions to the FDA. But the data belongs to the company. It doesn't belong
So
to
on
us.
that, I think it's a wrap up, but definitely we should take the time while we are all here to continue on sharing transparency, lessons learned. So how do we move the field together? I think that we can do better, definitely to accelerate how we move the field. So I hope that this discussion planted ideas, give you some insights and motivation to continue working on this amazing field of MPS. Magda?
I'd like to thank to all of the panelists for all,
Thank
all
you
of
to the
your
panelists.
insights, and to all of you in the audience for listening to us today.
So enjoy the rest of the meeting, of the symposium. Thank you.
So
Hello? I think we figured it out. We'd like to invite all of the speakers for this session to please join us on stage. We'll be getting started momentarily.
Hi, hi, nice to meet you. Do you want to sit right? I think you guys should sit over there. Yes. Yeah.
Medicinal chemistry, and my most recent graduate student, Anish Mahadia, who's leading the work that I'll finish up with. So first off, before we talk about MPS and ochratoxin, I really want to talk about chronic kidney disease, which is a global burden of disease. It's estimated 850 million people in the world suffer from some form of kidney disease. Risk factors for kidney disease include things like obesity, hypertension, type 2 diabetes, all of which are things which are increasing in frequency. To compare and contrast CKD and CKD-U, so chronic kidney disease has a number of risk factors like I just mentioned. What distinguishes CKD-U from CKD, which is chronic kidney disease of unknown etiology, are things like living in developing tropical regions, exposure to high heat and humidity, agricultural environment, and probably the most important thing is onset in young ages, people that are 25 to 50 that do not have some of those other risk factors such as type 2 diabetes or hypertension. In addition to, in terms of how the disease presents itself, it presents with a lack of proteinuria and really appears to be affecting primarily the proximal tubules, where in chronic kidney disease, you're looking more at a glomerular disease from things like hypertension. So in terms of the global distribution of chronic kidney disease of unknown etiology, again, you see it primarily in developing parts of the world, such as Sri Lanka, India, West Africa. In addition, you may have heard of a thing called Mesoamerican nephropathy, which is another type of CKD-U, which is you've got these hotspots of chronic kidney disease of unknown etiology. Again, these populations that don't have those classic risk factors for CKD. So what is ochratoxin A? So OTA, it's produced by various different molds, such as aspergillus and penicillins. So it grows on food products that are stored under hot and humid conditions. It is nephrotoxic and it is hepatotoxic. It's classified by IARC as a class 2B carcinogen. Structurally, it's a phenylalanine with this isocumarine moiety. It's got a high log P. It's extensively bound to albumin, so very small free fraction. It's got a very long half-life in the body. There was a single PK study done in Germany with one volunteer that was given a tritiated dose of OTA, and the half-life was over a month long. Its major route of secretion is renal secretion, but there's actually very little renally secreted. And the reason being is OTA is a substrate for various different transporters that are involved in uptake back into the kidney proximal tubule, such as the organic anion transporters. In addition, because at high log P, it's also going to be passively reabsorbed throughout the nephron. And it's estimated from rat studies, the concentration of OTA in the kidney can be 20 times that what you see circulating. So in terms of OTA food contamination, so it is ubiquitously found in food products. You can see wide ranges of OTA concentrations up to this 20,000 microgram per kilogram of what you see here in this pumpkin seeds imported from China. It's interesting to note that only Europe and Canada have restrictions of OTA concentrations in food products, which are somewhere between 5 to 10 microgram per gram, okay? So in terms of human exposure to OTA, estimated concentrations of serum concentrations worldwide is about 0.7 microgram per liter. But you can see serum concentrations as high as about 2.3 or 2.7 microgram per liter, which is reported in a Tunisian end-stage renal disease patient. To date, OTA has only been associated with endemic nephropathies, not been directly associated. And the mechanism of OTA-induced nephrotoxicity remains undetermined. And this comes a lot in part from the various different epidemiological studies that are listed in this table here, looking at OTA concentrations in either people with CKD or normal healthy volunteers. And you can see in some cases in the healthy volunteers, the concentrations are lower, but in other cases, they can be significantly higher. And so there's going to be a lot of different factors going on, not only exposure, but probably genetics and other risk factors. So the important thing to know about CKDU is it a multifactorial risk type of disease? Again, exposure to hot, humid conditions, various different environmental contaminants, genetics, and also these farm workers take a lot of high doses of NSAIDs, which are also known to be nephrotoxic. Let's just point this out. Oh, back. So the biotransformation of OTA, this is probably very hard for you to read. Long story short, the biotransformation, oh good, I'm gonna be able to see my screen down here. The biotransformation of OTA is very complicated. And so what you see here in the center of this is the parent compound OTA, but it's really thought that the major metabolites of OTA have no toxicities. But having said that, there are some quinone intermediates which have not been identified in biological samples, but have been known to be generated in vitro. And these can redox cycle and form DNA and protein addicts. And so while most researchers think that the parent is the primary route of toxicity, it can't be excluded that metabolites also account for toxicity, especially in terms of oxidative stress and DNA addicts, because again, these quinones are very highly reactive intermediates. So I don't need to go into the details of the MPS system that we use in our lab here at the University of Washington. It's based on the Nordis microphysiological system. So I think they actually have a booth here at the MPS World Summit. So you can go to them and talk about the details of this system. But we've used this for a variety of different things. First, with NCAT's funding to develop a kidney on a chip to look at physiological function transport. And in my lab, we've pushed that for looking at toxicity testing. But again, we've also looked at various different functions, including in extreme environments like the International Space Station. So our working hypothesis is that OTQ, this a quinone of OTA, which is produced by bioactivation of OTA, contributes to the nephrotoxicity. So this calls into the question the role of glutathione as transferase or GSTs in potentially mitigating OTA nephrotoxicity. Okay. So the way we actually approached this was showing here in this scheme here, you're going to have, again, this is orally route of exposure from contaminated foodstuffs, uptake in the liver, first pass bioactivation by the various different cytochrome P450s. Then you can potentially get circulating OTQ or the quinone, hydroquinone, and they can be detoxified in the liver by GSTs, which can then actually be cleaved again in the kidney. Or you can even have direct bioactivation within the kidney itself by the resident P450s. And so the question we're asking is there a role for phase 2 metabolism in detoxification of OTA and the reactive intermediates? And so the approach we took here was to look at simply looking at chemical inhibitors as a way of understanding this in our MPS system. So in the case of the kidney, what we took about was we synthesized a pan-GST inhibitor called NBD-hex. And what NBD-hex does, depending on the GST isoform, has picomolar levels of IC50s. And so the idea was if we put in NBD-hex into a kidney MPS, inhibited the GSTs, that should increase the amount of DNA addicts, oxidative stress, and enhanced nephrotoxicity. Conversely, if you put in ABT, 1-amino benzotriazole, which is a pan P450 inhibitor, if that P450 activation is necessary to get that reactive intermediate, that ABT should mitigate the nephrotoxicity induced by OTA. And so this is just a simple study designed looking at dosing MPS systems with various concentrations of OTA, ABT, and NBD hex. And in this case, it's simple readouts looking at effluent biomarkers as well as a live dead imaging. So in this stain here, this is, we've got four different stains here. We got bright field where you can see the kidney tubule. You got a live dead stain, which is a calcium AM for live, thetium bromide for dead, and total number of nuclei gets stained by DAPI. And what you can see in the presence of 10 micromolar OTA, which is a high concentration, but granted that level of that 2.7 microgram per mil in that one Tunisian patient is about 6 micromolar. So it's not out of the realm. But what you see here with this ABT and OTA, you don't really see any effects on viability. This is in contrast to that same concentration of OTA with NBD hex. It may be hard to see in the phase contrast image the constituents of the tubule, but what you can see with the live and the nuclei is that really there's only little spots of color, because what happens in a microfluidic flow system, as the cells die, they slough off and are gone. And this is quantified here in this slide here. And so we can see in first with nothing exposed, we normalize that to 100% viability. In the third column, looking at just treatment with OTA and ABT, you do see some protection going from 100% viability to about 70% viability. In addition, you see about 40% viability with OTA alone. But most importantly, in terms of the hypothesis of GSTs being important for detoxification, is that in the presence of 3 micromolar NBD hex with 10 micromolar OTA, literally there is 0% viability. So showing that knocking out that pathway is going to be very deleterious for protection. In addition, you don't really see much compromise in viability with either NBD hex alone or ABT alone. So when it comes to these MPS platforms, I always talk about these are not high content types of platforms, or these are not high throughput types of platforms. These are high content type of platforms. So one of our big readouts for high content is RNA-seq, so bulk RNA-seq on the chips. And so we looked at 2 different donors for different time points. So we looked at 3 days and 7 days, and we did increasing concentrations of OTA. And again, with these MPSs, we're seeding them with primary renal epithelial cells that we source from surgical waste. And so here's just a dose response looking at, on the left, it's going to be 3 days of exposure with an IC50 about 1.2. And with increasing exposure out to 7 days, that IC50 drops down even more significantly. And again, you can see the live dead stain with increasing concentrations from top to bottom, from 0 to 100 micromolar. You get very high toxicity at that 100 micromolar. And so here's just some volcano plots of the differentially expressed genes that we saw in the system, looking at either 72 hours on the left or 7 days on the right. And depending on 1 micromolar versus 10 micromolar, we saw anywhere from 2,000 to 1,000 differentially expressed genes. So to try to understand what's going on with all that huge number of data points, what we do is we do some pathway analysis using Advitus software. And of course, a number of different pathways come up, some which may not make sense whatsoever, like Parkinson's disease or things like that. But one of the things that did come up that was one of the top hits was glutathione metabolism. And so if you look at these different plots here, you can see, for example, a number of different GSTs are significantly downregulated in response to OTA exposure. Most importantly, one of the ones that's downregulated is GST-P1 or GST-Pi1. The reason why that's important is because that is the most abundant P450 in our primary renal epithelial cells, as opposed to like GST-alphas, which are very abundant in the liver. It's also important to point that out because GST-Pi1 is polymorphic. There's a common polymorphism in GST-P1, which reduces the activity by GST-P1 by 50% or even higher. And other pathways we looked at, we saw a number of different differentially regulated genes that were part of the NRF2 regulated cytoprotective genes. So we saw a number of these different genes being downregulated in response to OTA exposure. And so our proposed mechanism of OTA nephrotoxicity was, then we're looking at here, P450 bioactivation, whether that be in the liver or actually directly in the kidney, OTA to this hydroquinone, which then is very reactive, but GSTs can protect against that. And again, in response to OTA bioactivation exposure, you see downregulation of NRF2. And so NRF2 is a transcription factor which normally stays in the cytosol and bound with protein called KEAP1, but under oxidative stress conditions, KEAP1 gets oxidized and dissociates, and NERV2 goes into the nucleus and regulates a number of different genes, including things like GSTs. So in summary for this part of the work, this NBD hex's pan-GST inhibitor drastically enhances OTA-induced nephrotoxicity, while that pan-P450 inhibitor ABT had minimal or even rather protective effects against OTA toxicity. And again, under the conditions tested, neither one of these pan inhibitors had minimal effects. And again, this pathway analysis suggested that downregulation of NERV2 regulated genes by OTA treatment may help explain in part toxicity associated with OTA. So this led us to our next hypothesis, which is NERV2 activation will attenuate OTA-induced nephrotoxicity. And while MPS systems are our bread and butter, sometimes you don't need to use an MPS. One of the things we were talking about in the NCAT session days, use whatever is fit for purpose. So we thought, well, for this first screening process, let's just go ahead and use our same cell source, the primary renal epithelial cells. But in this case, let's go ahead and screen them in 2D first to see what's going on. And what we did is we used two different NERV2 activators to see if they could attenuate OTA-induced NERV2 inhibition. One is sulforaphane, which is a naturally occurring product found in very high concentrations in broccoli sprouts, for example, which is a NERV2 activator, versus TBHQ, or tert-butyl hydroquinone, which is a very potent NERV2 activator. But they both have the same mechanism where they promote the release of NERV2 from KEAP1. And so for this case, we looked at, set concentration of OTA, set concentrations of sulforaphane and TBHQ plus or minus OTA, 72 hours of exposure in 2D culture, and then did RNA-seq. So what did we see? So first off, again, what we see in response to OTA exposure, for example, glutathione reductase or GST-P1 are both significantly downregulated. We also see, for example, that a P1 is significantly upregulated by TBHQ. That's known to occur. But surprising, what we found is that in the presence of OTA, that these NERV2 activators, TBHQ or sulforaphane, that activity was completely mitigated by the presence of OTA. In addition, what's been shown when we look at these pathway analysis, OTA exposure induces a hypoxic response in our proximal tubular epithelial cells, or PTECs. And so if you look here in the bottom, in A, you're looking at HIF1 alpha. In response to OTA exposure, you see significant induction at the mRNA level of HIF1 alpha. Also ARNT, which is aryl hydrocarbon receptor nuclear translocator, or also called HIF1B, which partners with HIF1 alpha. That is also significantly upregulated. In addition, we saw, I had this one here, this protein or enzyme here, BNIP3L was actually it's a receptor on the mitochondria, which is important for one of our hypotheses for OTA-induced nephrotoxicity. So this is one of the things when we do our tissue culture, standard tissue culture experiments, we're doing them wrong. Our standard culture conditions are 20% oxygen tension, yet naturally, depending on the tissue, there's going to be much lower concentrations. So for example, here in the kidney is physiologically hypoxic. Renal cortex is about 30 millimeters of mercury. The renal medulla is one of the most hypoxic parts of our body with only about 3 to 10 millimeters of mercury. And so what we did is we cultured our cells under physiologically hypoxic conditions, in other words, normal conditions. Again, looking at this 2D culture and comparing the exposure of OTA plus or minus the NERV2 activators, sulforaphane or TBHQ. And here's something interesting to look at here. So under 20% oxygen conditions, our standard conditions in the lab, you can see some translocation of HIF1 alpha into the nucleus as shown in green in the center panel with 10 micromolar OTA. Don't see much for ARNT. In contrast, when you look at 10% OTA plus 5%, so hypoxic conditions, you see a significant translocation of HIF1 alpha to the nucleus as well as ARNT. But in addition, what we also see, if you look at this under phase contrast, when you look at, for example, in the DAPI stain, the nuke blue, you see regions where there's no cells because it's actually quite toxic. So in conclusions, what we want to do is, we're defining the mechanism behind OTA nephrotoxicity because it's important for understanding the multifactorial causes of CKD. It looks like our work indicates that nephrotoxicity may be caused by reactive oxygen species, OTHQ and OTQ. Current work indicates that NERV2 activation will not attenuate OTA toxicity. And finally, the message is, culturing your cells under low oxygen environment, in other words, these hypoxic conditions, plus OTA is going to be a more physiologically relevant toxicity testing. How are we doing on time? Okay, important thing here is acknowledgements. So first off, I'd like to acknowledge Jonathan Himmelfarb, who's the director of the recent director of Kidney Research Institute. He's a world-renowned nephrologist. He's been PI in multiple of the grants involved in this. Again, I'd like to acknowledge the folks involved in doing the kidney MPS. Again, that's Moki Imoka, Jade Yang, and Nish Mahadio. Also at the bottom there, Kathy Young, who's got a talk later this week on using our MPS system for PBK modeling. The inhibitor studies were done by my colleagues in medicinal chemistry, Alan Reddy, and the bioinformatics was done by Teo Bamler, Lu Wang, and James McDonald, which are part of our NIHS EDGE core. And so again, acknowledging funding, NIHS, CASIS, and CATS.
Sorry, so the panel is open for questions, please.
I just have a quick question, Victor Hernandez from Lone Bio. In your model, when you were assessing the effect of the drug and with the live data assay, I saw a lot of the cells were round and you consider that as a still live cells. If you change your analysis, would you have increased toxicity? If you only consider the ones that are physiologically elongated and the right shape?
Right, I mean, so if I understand your question correctly, you know, what's going on with the cells that are still alive? Yeah, I mean, so one of the other things we look at, I mean, I just showed you the live dead stain, but the other thing, the effluent that comes out of that chip, because it's a single pass, just like it is in your kidney. We also look at a variety of different biomarkers, urinary biomarkers that are associated with injury. And so we can measure those in the chips as well. And so if we look at early time points, we're not really getting acute toxicity by shedding of the cells. You can see release of those biomarkers, whether they be things like kidney injury molecule 1 or KIM1, or even urinary biomarkers, microRNAs we can see as well. So.
Any other question? With that, Professor Kidd, I have a question myself. So you mentioned about applying shear stress about 1 din per centimeter square. To my knowledge, the proximal tubule, the apical side is exposed to 0.1 to 1 din per centimeter square. Why did you use 1? And then you're studying probably OAT1 and OAT3, which are basolateral transborders. So how the shear stress affects the basolateral area? That's my question.
Great question. So the 1 dyne per square centimeter is what I've seen is the average, depending on where you're in the proximal tubule. Yeah, and we're actually, I'd say we've got the S3 segment in that it's not convoluted. But I've seen numbers anywhere from 0.1 to up to 10 dynes per square centimeter, depending on blood pressure. So those are all variables. And so we picked, you know, 1 because we're in that segmental. And in terms of the question about the basolateral transporters. So great question. I mean, the way that we did these experiments, basolateral exposure is not the route of exposure, it was luminal. But there is a fair bit of OTA that winds up in urine and is actually filtered as well, especially when you have kidney damage. But we've also, we've done a number of studies using a vascularized system where it's very important to look at organic anion transport there as well. And we did some earlier studies using that system to look at, can we get transport of OTA from the basolateral side into the effluent. And that was a really easy experiment to do because OTA turns out to be very fluorescent. And so we just measure the fluorescence. So, but in terms of the, you know, the basolateral transporters are absolutely critical in terms of understanding drug disposition. And so, for example, my colleague Kathy Young is going to be talking about the opioid PBK modeling we did. That was, of course, all done using a vascularized system. And the organic anion transporters are absolutely critical for that disposition.
Thank you very much.
Okay, now we'll move on to our next speaker, transitioning a bit now to the intestines. And we'll be hearing from Dr. Abhinav Bhusham at the Illinois Institute of Technology. He'll be telling us about a standalone microphysiological system with precise oxygen control for intestinal microbial interaction. The floor is yours.
Hello. Okay, thank you. I want to thank the organizers for inviting me for this talk. And what I'll present to you is, first of all, let me introduce the people who did the work. And most of this work was done by my graduate student or ex-graduate student, Cheng Yao. So I'll be presenting to you work which is, some of it has been published, but a lot of it is unpublished. So happy to receive your feedback. So the story starts where we look at gut bacteria, also called the microbiome. And just like AI, gut bacteria has been linked to pretty much everything that goes on with our bodies. So gut bacteria is linked to homeostasis in the body. It's linked to good health, linked to bad health, and specifically is really tied to several diseases, especially of the intestine, such as inflammatory bowel disorder, colon cancer, as well as diseases you will not think about originating in the intestine, such as diabetes and obesity, basically because of the impact of the gut bacteria on nutrient regulation. So we wanted to study the impact of bacteria on host health and some specific functions, which I'll go into a little bit later. And one of the things about the intestinal environment is that it is complex. Actually, all organs are complex. It just depends on what you're studying. So in this case, the complexity can come from the number of or the different types of cells that are present in the intestine, which part of the intestine you're looking at. So in our case, we're looking at the colon, and we're really interested in the heterogeneity that comes from the oxygen. And so in the section of the colon, if you look at the cross section of the colon, going from the epithelial side to the lumen side, there's intestinal cells, and then there's a thick mucus layer that is secreted by the intestinal cells, and then in the mucosal layer, there's the gut bacteria or the microbiome. So our interest in looking at how the oxygen is partitioned within this mucus layer. And typically it goes from, just like this was presented earlier in the kidney, the colonic environment itself is low oxygenated. But if you consider, for this, just as the purpose of this talk, we just call that normoxic. We just call that low oxygen environment normoxic. It goes from normoxic to hypoxic pretty fast in that mucosal layer. So most of the bacteria in this environment are going to be obligate anaerobes. So we wanted to capture this through our platform. So what we have developed is a very standard two-layer microfluidic device, an organ on chip. I say typical because most of these devices you will see are two layers. In the middle, the layer, they're separated by a membrane, a porous membrane, which is typically made out of polymer, such as PDMS or polyester. And then so in our case, we started off with a membrane that was made out of polyester. And then the intestinal cells or the colon cells are cultured on the top of the membrane. And after a few days, when the cells get mature, we introduce the bacteria. So one difference here is that we found out early on that the polymer membrane actually prevented remodeling of the colon cells. And so we completely replaced the membrane with a membrane that was freestanding, basically has no support anymore. The membrane is made out of type 1 collagen. We found that once we have the membrane out of type 1 collagen, it really allowed the cells to grow in three-dimensional architecture. That was one thing I wanted to share. The other thing I wanted to share with you is the oxygen. So the oxygen here in the system, we carried out a oxygen flux modeling, basically looking at pointer. So I'm looking at this figure. So basically, we're looking at the oxygen. Sorry, I'm on your left-hand side. So we're looking at the oxygen balance in the chip, right? So just think about the sources of oxygen. The source of oxygen is the environment, the oxygen coming from the media, and the sinks for oxygen or the usage of oxygen is the intestinal cell. So I want to create a hypoxic environment for the bacteria, an oxic environment. So what we did is we blocked the oxygen from the environment using a device. And then we modeled the flow such that the oxygen consumption by the epithelial cells is going to completely balance the oxygen provided by the media. By balancing this, we were able to, first of all, measure the oxygen through the oxygen sensors here. We can measure the oxygen actually coming out of the media. which is 0. And we can also, the good part is that we can, with this engineered model, is that we can tighten the flow rate. We can tighten the amount of oxygen that is in the media by tightening the flow rate. And we can tell specifically when or for which bacteria the amount of oxygen we need to provide. So if you look at the pictures here, we show that if the cells are, if you don't provide enough oxygen to the cells, as you would think, the cells will be hypoxic. If you provide enough oxygen to the epithelial cells, the cells will be normoxic. Basically, they don't have the, they don't create these hypoxic adducts. When the cells are happy, they generate really nice looking tight junctions, like the ZO1 protein here, which is absent again if the cells are not happy. So the experimental model itself gives us a really nice way to engineer the oxygen environment within the device. So the one thing here is that we do all of this without using an anaerobic chamber. Our devices can actually be outside the chamber because all the requirement for the oxygen in the media are provided only through perfusion. So it provides us a really good way of doing these experiments. So another validation we carried out was by just growing only bacteria. We have two types of bacteria. One is facultative anaerobe, that's a lower one. And it's E. coli Nissel 1917 strain, which has been GFP, encoded with the GFP protein. And then the other bacteria is the Bifidobacterium adelocentis, which is an obligate anaerobe. So we can look at the obligate anaerobe first. And so we try different conditions where the device is sitting in a standard incubator, the device sitting in an anaerobic chamber, a device with an oxygen scavenger. And basically, it turns out that this is the plot that you're really looking at. Our device works really well when you're sitting in an environment outside the incubator, because we completely block the environmental oxygen from entering the device, and the bacteria, in this case, the obligate anaerobe, can grow really well. So like this one, if you provide oxygen to it, it doesn't grow. And we see the same thing when we have the facultative anaerobe, E. coli Nissel, which basically grows, the growth rate is very similar depending upon whichever device you use, because this bacteria can grow regardless of the oxygen levels. So one thing is that when the cells, the intestinal cells, when they are really hypoxic, they don't produce, they don't provide good function. So you have to provide them the physiological amount of oxygen. One way to tell it is by looking at the amount of mucin proteins. So here we're looking at the expression of mucin 2, but we also have the immunofluorescent images for the stain for the protein. You see when the oxygen levels are appropriate, the cells produce a lot of mucin proteins. And also another modulator here is the bacteria. If depending upon the bacterial strain, we found that when the bacteria are cultured in the correct environment, they upregulate the mucin proteins in these intestinal cells. And our interest was in looking at the transporters, the absorptive function of the intestine. So we started looking at several transporters. These are associated with a lot of drugs. So we look at drug transporters, MRP2, BCRP, OATP, we have looked at many other transporters. And basically across the board, we find that a lot of the transporters are significantly upregulated just by the bacteria. We don't see any, we haven't done we haven't looked at the genes, the bacterial genes, is to see why the bacterial transporters would be upregulated. Why the transporters in the intestinal cells would be upregulated. So we don't see any pattern as to which bacteria would upregulate which transporters, but the finding itself is very interesting, because if we go up also looking at more mechanism in terms of transcription factors, we find that the bacteria also upregulate the transcription factors in these colon cells. And so what we ended up with is a possible mechanism. I know the slide, the font is very small, But basically what we're showing is that so the bacteria never come in contact with the colon cells, right? So the communication between the bacteria and the intestinal colon cells is taking place just by paracrine signaling. So what we hypothesize is that the bacteria is secreting the metabolites, and if that which happens only in the correct oxygen environment. So like adolescentis, it's the environment is hypoxic or it's anaerobic. Then the bacteria will release the metabolites, which will upregulate or downregulate depending on the transporter, and then pass the membrane, and then upregulate or downregulate or in this case, mostly upregulate the transcription factors. So we created a very simple PK model, just since these transporters are drug transporters, by looking at the change in absorption of an example molecule, just by looking at the pharmacokinetic profile and then pharmacokinetic profile if the absorption is actually modified. And we see, like 15% change in the PK profile of the drug. And so we feel that there's a strong, like if you look at other reports in which one example that I have is for levodopa, which is really impacted by the bacteria that have the tyrosine decarboxylase gene. And basically it will reduce these bacteria which have TDC will lower the amount of available levodopa in the body. And there's other examples for digoxin, similar example of digoxin, which basically these bacteria will directly affect the availability of a drug molecule. What we are showing is that there's a second level possible where the bacteria may not affect the drug itself, but the bacteria may affect the ability of the intestine to absorb the drug. In this case, I'm showing you it basically makes it more absorptive, but I'm sure there are cases where it could make it less absorptive. So we feel this is in our minds, it is a good, this could have a big impact. So in summary, I only wanted to share with you this story because we feel that we have a really nice engineered system in which we can really modulate the oxygen levels in a very simple way. You don't need any oxygen scavengers. We don't need incubators. You can you can create this environment in your microfluidic device really easily and create this partitioned oxygen environment, really tailor the oxygen consumption rate. And then also it takes a step towards finding mechanism and specifically for us to look at the absorption of molecules. And one of the key things here was eliminating the polymer membrane, which we all use. We love to use it. It actually is very easy, much simpler to use the polymer membrane than to use the membrane made out of ECM. But the ECM membrane does provide an opportunity for the cells to remodel, especially in the context of having co-culture with the bacteria. So with that, I'm going to end. This work was mainly funded through the ARDF and a current UO1 grant. This is a more recent picture without masks in Chinatown with a much smaller group. But I want to acknowledge the students who worked on it, mainly the PhD student Cheng Yao and also undergraduate students Tao Dang. And then my lab is always looking for graduate student postdoctoral scholars. If they're interested, please email me and I'm happy to take questions.
Thank you, Abhinav, for that nice presentation. Can everybody hear me okay? Okay. Maybe I'll get us started with a question. So I was wondering, where did you source your intestinal cells and the bacteria? And then kind of a follow-up on that, I've been thinking about how this model would be great for intestinal disease models like inflammatory bowel disease. I'm just curious if you've thought about that.
Yeah, great questions, thank you. Yep, so for this study, most of the for this study in particular, the cells were all the KICO2 cell line. I didn't present data on, we have data on animal mouse organoids and also a little bit on human organoids. We don't have, I didn't present data, we don't have enough data on that. So We're not shy of using Keiko2 cells. That's really the only standard cell line that's available, and that's really steady. The bacterial strains, E. coli 1917 was a gift from BSMG, and then the other bacteria purchased from ATCC, Bifido. And then your other question about the modeling intestinal disease is really relevant, just because the bacteria cause loss of permeability in the colon. One of the more common studies is on inflammatory bowel disease. My group does not look at IBD. In particular, we have been looking at colon cancer, and we've been looking at how engineered bacteria will colonize the colon cancer. So we have moved in a different direction. Yeah, thanks. Do you have a transporter question for me? Oh, sorry.
So sorry, have you been able to grow really super strict microbes like fecal bacterium Proxenitzii? And this gets into, it gets difficult to use Keiko2s with them because they make butyrate and the Keikos love glucose and that whole thing. But the FP is so involved in so many of the IB, really, really implicated. So, and they're like, you say the word oxygen in the room and they're like dead. So can you talk a little bit about how low you can go? And F. Proxenitzii in particular would be a very good test case.
There's a lot of echo, I'm realizing now.
Yeah.
But if I understand
I'll
you.
stand back here. Now can you hear fecal bacterium Proxenitzii?
Yes.
As an example, because it's one of the major butyrate producers. And, this gets into how you might not be able to do it with Keiko2s because, but FP in particular is super unbelievably oxygen sensitive. And that's sort of a real test case if you can do the IBD type stuff.
Yeah, thank you for the question. Yeah, I think it goes to say like our sensitivity for modulating the oxygen is, I would say it's not that great in terms of just being able to measure the total amount of oxygen, just because we rely on a fluorescence-based oxygen sensor. So we've been relying on actual growth curves of the bacteria within the system.
Yeah, we do the same thing.
Yeah.
And we can do all of them, C. diff, all the ones that are supposed to be strict anaerobes. Well, you can kind of get them to grow, but it's really F. Proxenitzii is sort of the canary in the coal mine, if you will. So I would encourage you to try FP because that one's the most super oxygen sensitive.
Okay. We'll thank you for
Except
the
it does make butyrate, so you might not be able to do it with Keiko2.
Okay, thanks.
Oh, should I use a microphone or not? Yeah, okay. So Robert Strimship, University from Latvia. I was thinking, so you mentioned that you are making your chips from PDMS, but then you're saying that you're using anaerobic culture media. I was thinking, so could you expand on how do you think about gas conditions in the surrounding area of the chip, and then sort of take it from there?
Yeah, great question. You're talking about the permeability of PDMS, I think that's what you're referring to. And then that is precisely the reason why we stick with PDMS, because we mathematically model the permeability and we take into account the permeability of the PDMS to environmental oxygen. And so we basically created a layer of PDMS which is thick enough that prevents the oxygen from permeating from the atmosphere to the chip. So like you can block it, right? You can basically, this is just transport of oxygen. So if you take into account the permeability, then you can basically model it. I can share with you all the details. It's already this published. Yeah. Thanks.
Hi. Can you tell us more a little bit about the manufacturing process for the ECM freestanding membrane, and how did you manage in particular to have it not sagging and not collapsing under its own weight?
Could you repeat the second part?
I mean, how did you manage to have it not sagging or like collapsing under its own weight?
Okay, so I think if I understand the question, it's about how do we create the collagen membrane? Yeah, and then how do we integrate it with the device, right? So basically we use a bovine type 1 collagen. And we polymerize it we basically create suspension, spin coat it, peel it off, and then we cut it to shape and we bond it electrostatically to the PDMS device. The way we do it is just like we do it for the polymer membranes. We have very little sag in the membrane when we bond the membrane. in the PD, and then and then we usually use oxygen plasma to bond the top layer of the device. I don't know if that answers the question. I can chat with you afterwards. I know these guys are getting anxious. I think I'm running over time. Okay, thank you.
Thank you very much. Okay, I'm happy to introduce our third speaker, Dr. Thomas Kostrozewski from CN Bio, United Kingdom, who's going to give a talk on characterizing the reproducibility of a liver microphysiological system for assaying drug toxicity, metabolism, and accumulation, joined to study by an MPS developer and a regulator. Dr. Thomas, please.
Thanks, guys. Good afternoon, everybody, and thanks for the organizers for the opportunity for us to present some of this work, which, as has been said, is a collaborative work between ourselves at CN Bio and the group of applied regulatory science at the FDA. So I think everyone knows that as a doing preclinical biology now, there's a really wide range of tools available, and most prominently, the rise of MPS has become a really key part of this toolbox to complement a wide range of other tools that are available, and picking the right tool out of this toolbox is key if you're ultimately going to get the translation that you need to predict what's going to happen in a human. And so how do we model the human liver? Well, the human liver is a key metabolic organ. It's the site of most common forms of drug toxicity. It's the key site where drugs are metabolized. And there are obviously a bunch of ways to model a human liver. Well, you can go and get some mice or some dogs. This is often used for modeling liver disease. And for many, many years, we've cultured primary human hepatocytes in plastic dishes. And typically, primary hepatocytes don't like that. And their functionality disappears after just a couple of days, trying to culture them on 2D plastic, even on collagen. So people have now been growing these in 3D and spheroids, sometimes as co-cultures, and this improves their functionality and is very, very nice. But can we go one step further? And this is where MPS organs on chips really comes in. And another novel technology that's also recently been developed is the use of precision cut liver slices. So how are you going to make a really good MPS to model the human liver? Well, the industry IQ MPS consortium a couple of years ago did a really great job of articulating this for the community. And I'm sure a number of you are aware, a group of lab on a chip papers was published describing some of the key criteria for what a liver MPS should be able to do. And so these include the production of the key biomarkers, the expression of key enzymes and transporters, and the right signals that a liver MPS would produce in response to damage. And so this is a real framework now for developers to work off to describe how we should build a successful liver MPS system. And there's been a whole bunch of liver MPS systems been built over the years. And probably another we're going to get described at this conference. And one of the key challenges still remains, as was talked about in some of the previous conversations, was, well, how do we drive the adoption of these technologies? How do we make them being used every day? And one of the key activities is to engage with the regulators to make and to do cross-lab into an intra-lab comparisons to show that these are reproducible, robust tools that can be used every day to ask key questions and to do this in a context-dependent manner. So let me show you more about our liver MPS that we have at CN Bio. So this was originally developed in Linda Griffith's lab at MIT a number of years ago, and we've taken this on and developed it further. So we have quite a unique MPS system. It's an open well-based system, looks very much like a cell culture plate. It has 12 individual wells. Each one of these wells is individually perfused, and the media recirculates through a three-dimensional scaffold that's held within the fluid path. And what we do is we seed single cells into this scaffold. These are typically cryopreserved primary human hepatocytes. They go in, and over a couple of days, these single cells form three-dimensional microtissues within the scaffold. And actually what we have is about 300 of these microtissues arrayed across a scaffold. So we basically recapitulate sort of mini columns of to mimic the liver microarchitecture. And so what these kind of look like, and these can be hepatocytes, or I'll show you in a minute how we can incorporate other cell types of the liver within these three-dimensional perfused microtissues. And that perfusion of media continually through the scaffold, through these microtissues, is really key to give the cells their functionality and maintain their functionality which they lose in typical 2D culture. So we get these beautiful tissues. They produce key biomarkers like albumin. They have bicanaliculi. They have tight junctions, and all the fluidics is controlled by our control system that you see there on the screen. And so a typical experiment with this setup normally takes a couple of days to seed those cells in, and we then have a really pretty long. Oh wait a minute, why do I keep going forward? We then have a pretty long window to run experiments in. So this allows us a lot of opportunity to do a wide variety of different studies, either in the tox or efficacy space, to really evaluate how liver biology is changing following an insult. And this model typically uses, I say, primary human hepatocytes, but it's not limited to that. We use the system with stem cell derived materials or even clinical trial samples now to recapitulate their function. And we can do a wide variety of endpoints, as I'm going to show you, to assess liver functionality. So is this model actually functional, and does it look like a real human liver in some way? Well, the answer is yes, it does. So if we take the bulk RNA-seq data from these chips, and we can do this longitudinally over time, and we ask like an online portal what it thinks it's looking at, it's probably pretty difficult for you to see slightly, but it thinks it's looking at bulk liver tissue come straight from a patient. It thinks it's primary hepatocytes, and all the key markers and key genes, the CYP enzymes, et cetera, are all still expressed, albumin being one of the most abundantly expressed genes. We express the key biomarkers of the liver, so those same biomarkers that were described in our IQ MPS paper, we produce in the system. We produce them to exactly the levels that were described in that paper, and were pretty much what your human liver would produce in any one of you guys. And importantly, we have metabolic activity. So from a drug metabolism perspective, we've got to express all the CYP enzymes that are present, and we can do this across multiple donors of cells, pools of donors, etc. And as I alluded to earlier, these microtissues primarily need to be formed of hepatocytes, but they can incorporate a whole range of other cell types too. So we can typically incorporate Kupffer cells, and I'll come back to this later. To have an immune component, we could add stellate cells, and we can see all the markers of those being expressed if we need to. We could also even add peripheral immune cells into these chips and allow those immune cells to perfuse around if we want to. So we've taken this liver MPS and put it in a whole bunch of different applications over the years. We have a wide variety of publications on this, ranging from work on DMPK and ADME, which I'll touch on in a minute. We've done a lot of work at disease modeling. I've got a lot of expertise in building models of human liver diseases, particularly of fatty liver disease, NAFLD, NASH, and of viral hepatitis. And we've used the system extensively for looking at drug-induced liver injury, which is still a really big key issue and a clear context of use where liver MPS can add a lot of value. So let me tell you a little bit more about the collaborative study that we did with the group at CEDAR in the FDA. So this came off the back of a DARPA-funded program we did along with Linda Griffith's lab, and we got interacting with the FDA, and they were very keen to see the outputs of that DARPA-funded program and the technologies that have been developed, and could they really be evaluated in much greater levels of detail and shown that they are reproducible and ready to produce IND-like package data. So what we did was we got the system set up in the labs at the FDA, and we have the system obviously set up in our own labs, and we could then cross-validate between those two labs. So first of all, we started pretty simply. Well, could they form the same consistent liver microtissues? And the answer is, well, yes, we could. See the standard sort of images there. Could these cells are also metabolically active? So this is the CYP activity. And these microtissues, we specifically decided to have an immune component, so they all contain primary human Kupffer cells. And so when we add an immune challenge, an LPS challenge into these microtissues, that metabolic activity goes down, just like it does in the human liver. And very importantly, I know these systems could reproducibly produce these microtissues. This is a really key attribute that the FDA are interested in. And if these technologies are going to be widely developed, any end user really wants a reproducible, consistent system that they can use over and over again, and again with multiple different sources of primary cells from different donors. And we assess the reproducibility. We assess the reproducibility of the system across different cell sources. What we then did was then take the same cells, the same combination of hepatocytes and primary Kupffer cells, and put them into the liver MPS, and at the same time put them into spheroidal culture and 2D matrigel or sandwich culture. and asked, well, are the same cells like functional in all of these systems? And can they maintain their function? And what we could clearly see was that in the liver MPS, the same cells were much more metabolically active. They produced much greater levels of albumin, and this was maintained over a long period of time. The standard spheroidal and 2D sandwich cultures, I know at the beginning, for the first few days of the culture were okay, and they were doing as you would expect. But over, 3, 4, 5 days, that functionality and that metabolism really dropped away. And you do see a slight decrease. Oh, I keep pressing the wrong button. You do see a slight decrease in the metabolism over time, but this is still way above what could be achieved in any of the other systems. And exactly the same was seen for albumin. So we really see that the liver MPS is creating the right microenvironment to maintain the phenotype and function of these cells. So then we really wanted to understand, well, can this be used for key context of use that the pharma and drug development industry cares about? And so we chose 2 different areas to look at. The first one being DMPK and pharmacology, and the second one being safety, pharmacology, and toxicity. So to assess DMPK, could the system be used to assess the clearance of human drugs? We looked at the pro-molecule diclofenac, which has been well used and well studied for this type of application. And what we could see if we dosed diclofenac into the system, without any cells, it was very stable in the system, it didn't go anywhere, but as soon as the system contained those liver microtissues, it was rapidly turned over and cleared away by the liver microtissues. And this is exactly what you'd expect when this drug is dosed in vivo. Now, another small but very, very important piece of data that was generated was to show that the system has really low, almost minimal non-specific binding to these small molecules. This is a real chronic issue that's plagued the MPS industry for a while. The use of soft polymers are well known to absorb small molecules in particular, and also can absorb a whole range of different proteins too. So we did a nice study, part of this, we took a whole bunch of range of pro compounds, we put them in the system, we showed that they didn't bind and don't get absorbed. So this enables these DMPK studies to become much more straightforward. The other key thing that the DMPK study wants to be able to do is to see the production of human-specific metabolites and understand the breakdown products of any particular compound. So here we use the example of triglyceridone. We could put this into the system, incubate it with the liver microtissues, and we could pick out the primary and secondary metabolites for triglyceridone being produced. And this is key because you've got human-specific enzymes here, so you'll get the human-specific readouts that are coming out of this system. So we could demonstrate that the system was very applicable for DMPK type studies. Well, could it be used for toxicological assays and understanding how could you predict the toxicological response of a compound in the system in the same way that you do or you more standardly do in 2D or spheroidal cultures? And again, the answer was yes. So we used a range of pro compounds. They were all, again, run in parallel through the 3 different models, the liver MPS, the spheroid, and the sandwich culture. And we could look at a wide variety of endpoints of biomarkers. We could see decrease in CYP activity and albumin, dose-responsive effects. We could see the production of and release of LDH. And all the different systems were able to produce IC50 curves, which is a key fundamental of any liver toxicological assay. But we wanted to go a little bit deeper than that, and could we show a real elegance and a real depth to what the liver MPS could do? And so we used the example of trovafloxacin and its twin molecule levofloxacin. And trovafloxacin was removed from the market for causing idiosyncratic DILI. In the presence of inflammation, it was found that trovafloxacin, the antibiotic, would cause real severe liver damage in some patients. And so we asked, well, could we recapitulate this in the system? And to walk you through this data, the answer was yes, we could. So we dosed both levofloxacin and trovafloxacin in the presence and absence of an inflammatory stimulus, so here LPS. And we see that the trovafloxacin causes significant DILI, so we see the release of LDH and the reduction of metabolic activity of these cells. And this was driven and enhanced in the presence of the lipopolysaccharides. So in the presence of the inflamed, damaged liver, we get much more susceptibility to DILI, and this is exactly what is seen clinically. And importantly, to really add to the reproducibility piece, this study was done with multiple lots of cells, which is key, and it was also done at multiple sites. So we could do exactly the same study, the same system, and really demonstrate the kind of reproducibility and robustness that you could get this data wherever you are in the world and in whatever lab. So in summary, hopefully I've shown you this afternoon that we've built a liver MPS system that is really robust and ready for use for a wide variety of different uses. The liver microtissues that we form are really, really highly functional and can be used to assess a whole variety of different clinically relevant biomarkers. The microtissues can be very consistently formed from different sources of cells to give you really high content data. It was just mentioned by one of the previous speakers. These systems are really high content. These not really HTS screens. They need to complement and work alongside HTS screens to get the right set of preclinical data that you want to ultimately be successful. So thanks for listening. We have more information about this MPS system across the week here at the MPS World Congress. So we have a number of posters and we have a booth. You can come and see the system and play with it yourself. So thanks for listening, and I'll take any questions.
I'm Donna Mender from NCTR FDA. So I have a question. At the beginning, you mentioned you can use primary hepatocytes, cell lines, iPSC. Have you done that kind of comparison where you looked at metabolic activity over time and other aspects to see which incoming cell type is most important?
Sorry, I think I only caught half that.
Okay,
was your question, have we done HEPs, IPS, and cell lines in parallel?
And looked at metabolic activity over time and the other analysis you did with primary hepatocytes. I'm just wondering how the other cell types acted.
Yeah, so the system is totally compatible with all of those other cell types. We see enhanced functionality of those other cell types in the MPS compared to static 2D cultures. Those cell types don't have the same metabolic capacity, particularly of the primary hepatocytes. They just simply don't have the horsepower. IPS particularly, I think, are the future to some extent, and I say that with a sort of wince, because of their unlimited source, their kind of ability to genetically control them. I think we really want to be able to build all sorts of MPS using IPS-derived cells, but their functionality is still not the same level as primary human cells. So the focus for building MPS at this point has got to be on those primary cells. And whilst we work with the IPS community closely, because IPS cells or IPS-derived cells in these type of systems do function much better, but they don't suddenly become fully mature just because you put it in MPS.
Okay, thank you.
Any questions? Well, thank you for your talk. I have a small question, if I may. So how did you quantify reproducibility in your MPS?
That's a great question. We really just looked at the ability for the microtissues to form across multiple wells, multiple plates, as I say, across multiple sites and with multiple cell sources. We didn't do a deep statistical analysis to determine how specific numbers that we should look at. But when we looked at across a range of plates, range of site range owners, we see greater than 90% of all the microtissues are the same. And I think that, but what's important, that is done with cells that we know will work in these three-dimensional tissues. There's lots of primary cells out there that won't form good quality three-dimensional tissues in any MPS or even any spheroids. So I think I should qualify that these cells were pre-qualified to do that. And once you do it, you then get that high level of reproducibility. Thanks.
Great, thank you so much. I'd like to invite our last speaker up to the podium, Dr. Carly Strelas from the University of Southern California. She's going to tell us about peristalsis-like deformations, increased tumor cell intravasation through GABAergic signaling, and colorectal cancer on a chip model. The floor is yours, Carly.
Thank you so much for letting me speak here today. I'm going to be talking a little bit more mechanistic. I'm a biologist by training, not an engineer. So it's going to be a lot of signaling and cell biology. So I hope this is a great example of how we can use these chips to uncover novel biology. The green button? Oh, thank you. Colon cancer is a complex adaptive system. We know a little bit about how the primary tumor evolves, how the tumor cells invade into the bloodstream, and how they set up the distant metastatic site, but there's so much that we don't understand. And so we're hoping to really uncover what we're missing during the metastatic cascade, which is a really great example of how I hope we can use these organ on chips, particularly because of the organ-like tissue architecture and function, especially when you consider all of the different cell types present within the tumor microenvironment, all of the physical forces that are very important in cancer development, and the microfluidic component as well as we start to think about this metastatic cascade. I've been working mostly with the Amulet organ on chips, which comprised of two channels, microfluidic channels separated by a porous membrane. In our top epithelial channel, we can layer on different types of cells. Specifically here today, I'm talking about a normal epithelium, and then we can layer on these colon cancer tumor cells that will form these spheres in the top channel. Or we can actually use patient-derived colon cancer tumor organoids I'll be talking a little bit about today, although our model is, someone else will be describing our model at a different time. In our bottom channel, we're using human umbilical vein endothelial cells. And then in this top channel, we've worked with a few different colon cancer tumor cell lines, whether the HT29s, the more aggressive HCT116s, and again, like I said, patient-derived colon cancer organoids. I've described this tissue-tissue interface. I'm going to talk through a few different assays that we're using to interrogate this colon cancer on a chip, with the whole goal of modulating the tumor microenvironment, whether it be different cells, whether it be the physical forces like I'm talking about today, and then using these different assays to understand how the tumor is progressing. I'm first going to talk about an imaging-based tumor cell invasion assay, and then I'm going to go into some of the effluent analysis, whether it be cell-based effluent analysis or a metabolic approach. We've developed an imaging-based invasion assay. You can see here, we're looking at a worm's eye view of one particular region of the chip. We can see our green tumor cells in the top channel and a tube of the red endothelial cells in the bottom channel. And you can see over time from day 0 to day 6, my green colon cancer tumor cells are actually invading into the bottom membrane where they're adhering to the endothelial cells and proliferating. And we can actually quantitate that over time, with my more invasive HCT116 cells invading more at the end of the experiment on day 6 than my less invasive and less aggressive HT29 cells, replicating the biology that we see from which these tumor cells were derived. In addition, that's measuring the tumor cells that have invaded into the bottom channel and adhered, but tumor cells do also slough off and can be found in circulation. And because this is a microfluidic device, we can collect the effluent, and then we can actually capture these tumor cells that have invaded into the bottom channel and end up in the endothelial effluent. They do remain viable. We can quantitate them, so we can see that we get more of these circulating tumor cells in my HCT116 chips, which are my more aggressive colon cancer tumor cells, and less circulating tumor cells in my HT29 chips. In addition, we can perform some qPCRs and look at gene expressions. In this example here, we're seeing that Vimentin is increased, E-cadherin is decreased in these circulating tumor cells, suggesting that they are undergoing some epithelial to mesenchymal transition. And finally, my last assay that I'm going to be describing before we get into the cool biology is that this is a microfluidic device. We can collect the effluent and perform various analyses on the effluent. The one that we're talking about today is a mass spec-based metabolomics. So we can look at the differentially expressed metabolites. We can do over time. We can do between different perturbations, different versions of the colon cancer chips. And so when we look at the differential metabolites from the start of my experiment to the end of my experiment, we can see that there's a lot of differentially expressed metabolites present in my colon cancer chips as we compare to the non-diseased epithelial chip. When we put these through the IPA analysis, the ingenuity pathway analysis, we see a lot of TCA cycle and amino acid metabolism changes in my colon cancer chip over time. And in addition, when we map that back to colon cancer with using metabolic markers of colon cancer that we see in human patients, we can see that my 2 colon cancer chips actually map to the colon cancer disease state very significantly, with again, my more aggressive HCT116 chips mapping more significantly to the colon cancer disease state. Now then I'm using those 3 assays to then interrogate some biology here. And one of the projects that I've been working on during my graduate studies and as well as a postdoc is trying to understand how these physical forces that we see in the body that surround the tumor cell, how does that influence the colon cancer progression? There's a lot of talk about fluid flow and shear stress, especially within these organ-on-chip conferences and in the world of organ-on-chips. But we also have to think about the mechanical forces in every single organ. And that's not often very difficult to recapitulate. It's also very difficult to study. Today, I'm going to be talking about peristalsis, how food is moving through the intestine. And we can do that. We can model that with this particular organ-on-chip system, where there's a vacuum channel where vacuum is applied and released throughout the course of the experiment. And you can see here, this is a video from Emulet, actually showing the cells on the chip constraining and retracting. One of the first things that we did was we looked at the invasion, how these tumor cells are invading over time in the course of my experiment. And so I'm looking at the invasion fold change here as we compare to stretch conditions and not stretch conditions. And those last two bars are my colon cancer chips. And you can see that there's a 6-fold increase in invasion when the peristalsis motions are applied to these HTT116 chips. In the HT29s, we see about a 2.5-fold change. And then when we go to the patient-derived organoids, we see about a 2-fold increase in invasion as well. So this is not just a cell line phenotype. We are working to expand the number of patient-derived organoids that we're working with. But overall big picture, if we have these mechanical deformations, these tumor cells are increasing their invasive capabilities. We looked a little bit at some of the barrier functionalities of these chips as well. We don't see any large scale changes in tight junction formation, which is measured here by ZO1 staining between stretched and not stretched chips. And in addition, we don't see any huge deformations of the barrier functionality of these chips as well in the stretched and not stretched conditions based off of a permeability assay. We have more work to do to really characterize this, but we're not seeing huge issues in how these cells are growing in response to these mechanical deformations. And in addition, one of the cool things about the peristalsis abilities with this particular chip is that you can apply the stretching and remove it over the course of the experiment. And so we set up different perturbations that start and stop at various times of the experiment. So we could have stretching applied for 2 days and then remove this peristaltic motion, and we can have the reverse. And we can actually look at the course of the invasion and how these tumor cells are responding. And I want you to look at 2 different colors here, the blue and the green. And you can see that in my green chips, we didn't have stretching at the beginning of the experiment, so we don't really see much invasion. And then on day 2, when we start this stretching, we can see the invasion really increases over the rest of the experiment. And the vice versa with this blue line here, when we do have stretching in the beginning of the experiment, we turn it off and the invasion really levels off, suggesting that this really is a plastic phenotype and they're really responding very quickly to this perturbation. In order to understand better what was going on, we went back to our mass spec-based metabolomics to understand on a bigger scale and try to focus more on what we should be looking at with this particular phenotype. And when we did some differential metabolites of stretched versus not stretched effluent, cancer came up as a very significant pathway, which is good. But we also saw neurological disease pop up, which was pretty interesting to us and made us start to research and think about the gut-brain axis and all the crosstalk that happens with the neurotransmitters from the gut all the way up to the brain and vice versa. We performed some aspect-based metabolomics with a neurotransmitter-specific metabolite library and saw quite a few of the neurotransmitters that significantly changed between stretched and not stretched chips. GABA was very significant here with a highly increased fold change in stretched and not stretched conditions. So we decided to focus a lot of the work on GABA. And so what we're looking at here is we're looking at GABA metabolic intensities from our mass spec-based metabolomics. I'm sorry, the scale is cut off on that middle graph, but we're looking at the fold change in the top channel effluent of my chips. And you can see that in my cancerous chips, we're seeing, again, a very big fold change in the amount of GABA present coming off of these chips. So with my HTT116 chips, with my HT29 chips, as well as with my patient-derived organoids, OOOUS, we're seeing about 5-fold increase in GABA in the effluent. And in addition, we can actually introduce some C13 labeled GABA into the top channel, and then we can collect the cells off of that top channel and perform mass-spec-based metabolomics to see how much of this C13 labeled GABA is actually in the cells. And we can see that in my stretched cells, in purple, we see quite a bit more of this heavy labeled GABA, suggesting that these cells are not only secreting more, but they're also taking up more as well. To try to focus more of this mechanistic a little bit more, we performed some studies adding different GABA receptors, agonists or antagonists into the top channel of the chip. This is a complicated graph, but basically I'm looking at GABA receptor A and GABA receptor B antagonists or agonists. And so if we look in the blue section of the graph, you can see I have my stretched invasion happening. So we have lots of invasion, tumor cells are invading into the bottom channel. And then when we block that with a GABA receptor A antagonist, we can see that the invasion drops significantly, which we do not see that with a GABA receptor B inhibitor, suggesting that this is a GABA receptor A mediated response. And in addition, we can also perform some immunofluorescence on the invaded cells. So the tumor cells that have invaded into the bottom channel, they've adhered, and then we can see that they stain positive for GABA receptor A and they stain negative for GABA receptor B, although my endothelial cells stain very nicely for GABA receptor B in the image on the right. So where we are with this mechanism right now is that we have this peristalsis-like motion happening. The tumor cells are responding to that mechanical force. They're secreting GABA, but they're also taking GABA receptor in, and we believe that they're metabolizing it, although we have much work to do there to understand that. And there's also some binding to GABA receptor A and some downstream signaling that, again, we need to better understand as well. But altogether, these cells are becoming more invasive. But quickly and very preliminarily, I want to ask the question about what does this ultimately mean for metastasis? Great, these cells are becoming more invasive. What does that actually mean for the secondary, more deadly tumor? And when we were doing some RNA-seq on stretched and not stretched circulating tumor cells, so remember the cells that we can collect and remain viable from the effluent, we noticed that there were quite a few different pathways that were differentially changed in the peristaltic-like conditions. One of them was these GABAergic synaptic transmission pathways, which is great compared to the other data that I've shown here. But we also saw a lot of cell adhesion and cell-to-cell interactions, integrin binding pathways as well. And so we wondered a little bit about if this mechanical motion was actually helping these cells adhere in the liver secondary metastatic tumor setting. What we can do here is we can start to think about the metastatic process and linking. Unfortunately, we're linking by hand right now these colon cancer chips with a healthy liver chip, because most of our colon cancer tumors actually metastasize to the liver. And so what we can do is we can take the effluent, we can take some of these circulating tumor cells from my colon cancer chip, and then we can flow it into the endothelial channel of a healthy liver chip and see if these cells will actually adhere to the endothelial channel and then extravasate and colonize the hepatocyte layer. And what we can see is that after a lot of optimizing, these tumor cells will actually extravasate and colonize the hepatocyte layer. And then, of course, I asked the question, what happens when you have this mechanical pressure in the colon tumor setting? How does that apply in the liver setting? And so what we did is I took some circulating tumor cells from my stretched colon cancer chips, load them into the liver chip, and then monitored how they actually adhered to the endothelial layer. And we can see that the stretched cells adhere more than not stretched cells on that very first day after introducing them into the liver chip. In addition, we can start to think about metastatic events by seeing by the end of my experiment how many tumor colonies are in the top channel, the hepatocyte layer of my liver chip. And we can see that in the when the cells have been stretched before, they flow into the liver chip, 3 out of 4 times there's a metastatic event, whereas when the cells are not stretched, only 1 out of 4. And that means 3 out of 4 chips actually had a metastatic event. And so to conclude, this colon cancer on a chip is really a great tunable model to study the role of the tumor microenvironment in cancer progression. And the tumor microenvironment that I'm talking about today is these peristaltic-like motions in which the tumor cells secrete and take up more GABA, and then they increase their invasive potential. And the future work that we're looking at here is really, hopefully all patients have peristalsis. And so what does that actually mean in terms of how treating these patients? And so we're starting to think about using these patient-derived organoids from different patients across the different colorectal cancer stages, across different mutational statuses as well, and trying to understand if all of those organoids respond the same way to these mechanical motions. And then as well, of course, we're also interested in how all of the other stromal cells, immune cells, fibroblasts as well, how do they respond in a GABAergic environment? This is a really highly collaborative effort. Everyone at the Ellison Institute has been very helpful, as well as a lot of labs at USC. We worked closely with Emulate over the years, and they've been very helpful as well. And we are funded by NCI Tissue Engineering R01. Thank you very much for your attention.
Thank you, Carly. We have time for one question. Here we go.
Hi, it's Jason from MIT. So about the stretching experiments. So I know your research actually focus on the cancer the cancer part. But and can you also comment on the stretch effects on the vessel side, blood vessel side? Because I think those endothelial cells probably will also respond to the stretch, and maybe they secrete something to enhance the invasion. Thanks.
Are you talking about the endothelial cells?
Yes.
Yes, yeah, great question. And we just got back some single cell RNA-seq about that. And there's a lot of endothelial crosstalk that we're really interested in looking at. It's too preliminary right now. We're still going through all the data, but we think that the endothelial cells are really important here. And so we're hopefully following up on that. Thank you.
Thank you again, Carly, and thank you everyone for your attention.
Thank you very much.
Yeah. Okay, so we don't see the time here.
You do actually.
It's there.
Yeah.
Sorry.
Emily, how you doing?
So the criteria. I'm sorry.
Right, yeah, so why do you guys feel free? Oh, it's not a bit overkill, but hey, you know. Yeah, that's cool. Hi, how you doing? Great to meet you.
Were you here?
Yeah, of course. Yeah. Yes, hi. Just
Hello, testing. There we go. Lovely. All right. Thank you, everyone. Good afternoon. Welcome to the session on lung MPS. We've got a great lineup of speakers this afternoon. So starting off, we have Arno Gutlev from the Luxembourg Institute of Science and Technology. Arno, please start.
Moyen, this room is almost the size of my country. It's impressive. Yeah, for those that don't know, Luxembourg is a small country, 600,000 people between Belgium, Germany, and France. And we do research. I will only show a bit of research from my group. The rest of the lecture is more an overview on the landscape of models that we do have for the pulmonary system. I will show you really a lot of slides from different companies. And because of this, I choose today to have a conflict of interest statement expressing explicitly that I do not have any interest in any of these companies. But you may think so. What I show is not an endorsement for any specific technology, for any specific company. And I also want to say that I have got from some of the companies, so to say, official slides, and some slides were simply taken from their websites. You will also see that some of the companies I mentioned are here exhibitors, and I think even 2 companies are giving talks in this session. So how far has the community come when it comes to pulmonary system? What you see on this slide is a representation of an alveolar barrier. You see endothelial cells below with some dendritic cells in the blood. You see macrophages on top of the alveolar cells. It's a nice graphical representation, but how are we really scientifically? When it comes to the lung, there's actually an interesting documentation from the JRC. The ECFAM has done an extensive literature research on cell culture models representing the respiratory tract. And what they found, and this is really amazing, is 284 different models. Now, this sounds like a lot, but if you use a different cell line in a slightly different setting, it counted at 2. If you start to combine 2 cells in a different setting, it also counted as a new model. So the number of actual models is partly slower. And most of these models have been used in an academic setting, and very often in do it once, publish once, and never do it again setting. Well, I will try to show you things that are really at the market or very close to the market and can be used by industry. There's another kind of disclaimer or baseline that I want to set here. We often do cell culture in a classical way submerged. And I really want to make here the statement, when it comes to the lung, we have to work at the air-liquid interface. It's not possible if you think about particles to work submerged, because you know protein corona, etc. You test something which is not human relevant. And if you already have to work for nanoparticles at the air-liquid interface, why not work for chemicals too? Because this allows you to better transfer and compare data between particles and chemicals. It's a long list. The only reason I want to show is that air-liquid interface is not something exotic. Most of them are from the last years. Some of them are already 20 years old. And they have been used for airway cells, for alveolar cells. You can use them in 3D culture settings. There are human primary models commercially available. And even you can have air-liquid interface, and I will show you some examples, in the form of organ on a chip. So there is no reason to work with lung in a submerged setting. Before I go to all these commercial things, I will show a few slides from the work that we do in Luxembourg. On the right side, you see a schematic representation of how an alveolar epithelial area looks like. You have epithelial cells type 1, type 2. You have capillary cells, and of course, you will find some immune cells. And that's what you try to do. We established transwell insert models, which you see on the left side. Again, in a transvaal insert with a membrane of different pore size, depending on what you want to do. Endothelial cells, epithelial cells, and immune cells. And this allows you, and this is indicated with this orange dot, to study, for example, particles in a natural way without any interference with serum components. When it comes to these cell models, and especially when you, for example, switch for the first time from submerged culture to air liquid interface, you have to do some characterization steps. One of them, you want to be sure that your alveolar cells produce the surfactant, that your bronchial cells produce mucus. There is a very easy test. If you culture them and they would not express surfactant, the cells would anyhow die. But I also want to show that the cells really produce surfactants. And what you see here is a simple test. And on the upper part, you see alveolar cells A549. You use a specific phthalate solution with a blue dye. And if it's a small spot, you know you have surfactant expressed at the surface, not intracellular, but really at the surface. And as a comparison, you see the endothelial cells that we use in our model. You see a big spot showing that, of course, there is no surfactant produced in an endothelial cell line. Just one easy step to show that your model is properly working. We have macrophages in our system, and I want to show you also that the macrophages do what they're supposed to do, namely to take up, to phagocytose particles. In this case, we used 50 nanometer large silica nanoparticles. They were rhodamine labeled. We sprayed them for 30 minutes. Cells were incubated, the models for 48 hours. And then we stained the cells for for confocal microscopy. What you see is in blue the nuclei, in red the cells, and in green antibody against CD54, a marker for mature macrophages. And you see several of these macrophages. In the inserts BC, in the middle of the picture, you see that these macrophages, the green marked cells, have white spots in the cytoplasma. And these white spots are accumulations of the silica particles that we have sprayed 2 days before. So the macrophages have exactly done what they were supposed to do. They've taken up the silica nanoparticles, and you also see no white material outside of the macrophages. So they have really cleaned the surface of this transvaal insert. Now we have a complex model, and I want to show you also kind of the beauty of complexity and what you can do with a complex model. And in this case, we sprayed diesel exhaust particles, diesel on the cell culture, on the epithelial side, like you would inhale. And what I will show you as a result is effects that we have seen on the endothelial side. So whatever you see is due to either material coming from the diesel particles that have migrated through the pores into the endothelial cells, or second messengers, chemokines that have come from the epithelial side that induce something on the endothelial side. The material was sprayed using exposure equipment from a German company, Vitrocell, which is also exhibiting here. And that's what we saw. So we sprayed our cell culture models at what we think were realistic concentrations. And the data I show you again are from the endothelial side. So I do not talk now about the epithelial side, let's say the direct exposure. Everything I show you is indirect exposure. And if you see in the red box, there were no statistically significant differences in cell viability between the exposures and between the air-liquid interface. So the culture is fully viable. That's nice to see. Then you should never do that. I show you so many digits in the next box, and you don't have to read them because there were no differences in any of the genes measured on the endothelial side. So that's easy. Forget about the numbers. No effects due to the exposure. And the next I don't show, but I zoom immediately in because that was the last thing. And here it becomes really interesting. 4 hours after we had exposed the tetraculture to diesel exhaust particles, we see something very interesting. And this is the lower left quadrant, the green picture that is of interest now. That's immune staining against NRF2, which is a cytoplasmatic receptor that circulates between the cytoplasm and the nucleus. And upon exposure to aliphatic chemicals like benzpyrene, which is present in diesel, it binds to the DNA. And what you see is on the left, there's a little bit of green in the nucleus because it's, as I said, a shuttling receptor, but on the right side, after exposure, the green dye accumulates in the nucleus. That's an indirect effect, a very early effect, and this will turn on genes later on, and as a result of indirect exposure. Now coming to complexity, I have 2 more slides. Why is complexity so interesting? We have 4 cell types in our model. Is this really necessary? What is the added value? We did this with an industrial partner, Philip Morris International in Switzerland. And there was no exposure at all, what I show you now. This is just epithelial cells alone. And then we looked into the active gene networks. Then you add endothelial cells, and again, you look into the active gene networks in the 2 sides of the transwell. You add the immune cells, and again, you look into the active gene networks. What do we see? First, on the left side in blue, you see the results from a 3D model, and in violet, pink, you see the results of a 2D, and the first observation is that the 3D model has higher bars than the 2D model. Now, the y-axis of these 2 figures is the similarity between an in vitro model in respect to active gene networks and the human non-exposed in vivo situation. This is compared to patient material from surgical operations. So you see that the 3D is very similar to the human in vivo situation, and within the red circle you see also something interesting. That's the tree culture, meaning epithelial cells, endothelial cells, and macrophages, or in addition the mast cells. And what you see is that the addition of the mast cells does not add anything relevant anymore, because you are on a kind of asymptotic curve. You are at around 80% similarity to the human in vivo situation. So that's why we kicked out the mast cells from our system, because it has no added value. You only have consumable costs, time costs, etc. So now I come to the second part kind of my presentation. Some of the commercial cell systems, simply to show what is available for the community. One company, Swiss French, Epithelix, they have primary human material, and they can represent the nose, the trachea, the bronchi. You get them in different well sizes. And maybe I go direct to the next one. If you know human bronchial histology, that looks very much like a human bronchial tissue would look like. You even see the cilia. The cilia are also beating in the system, and the beat rate can even be used as a readout. What is also nice is you have certain diseases available, meaning material coming from diseased patients, such as asthma, COPD, cystic fibrosis, etc. Second company, US based for Europe in Slovakia. They have similar models. They also have an alveolar model, and they have also tissues for organs which are relevant for the lung but are not lung. If you think about inhalation, of course, having a model also for the eye, the cornea is of interest because you will inhale what you also get in your eye. So you can have a quite complex and complete exposure model. One model, which is very endpoint specific that I want to show you, this is now when it comes to respiratory sensitization. Swedish company SensaGen, they have the GARD Air. It's a model based on one cell line and an AI readout of a set of genes, which allows to differentiate between chemical sensitizers and chemical irritants. Another company, they are also here in Screenex, and they are specialized in producing certain cell types. Why is this of interest? I showed you data and cells from A549, and A549 are often criticized of having not fully functional tight junctions. This is a cell line that has fully functional tight junctions. The cells develop, or the models develop, a very high transepithelial electrical resistance, which the A549 don't do. So dependent on what you want to study, this may be a very important feature that you want to have. Another aspect that you can have, Immuno1, it's a British company. They provide immortalized alveolar cell line in combination with immortalized alveolar macrophage cell like cells. These models are available both at the air-liquid interface, but also liquid-liquid, meaning submerged, and in a 24-well format. They are now since 2 years commercially available. A bit more recent, Japanese company, Hailang, they have developed for the first time, to my knowledge, IPS-based model that is also available and may have promising features as it is different from the cell line and tumor cell line based models that I've showed so far. Some of these models are also available as a lung on a chip. That's one of them, Emulate. They are also here. You can culture them in a similar system that we have seen at the end of the liver talk, but you can also culture in this system lung cells. You can have stretch as an additional factor. Another model from Mimetas that's quite complex. You see, it's almost like a multi-well organ on a chip system. You can load it with primary human cells, dependent what you want, alveolar, endothelial, and you can also add stretch again. That's lung on a chip model. The company is also here. You can combine it with inhales incubation, and then next speaker will show you about this. What is missing is this, the pressure. Company is here. It's a pressurized system. It's a kind of 96 well plate format, and you can add pressure and the system is breathing. With this, I come to my last slide. It's very important that you, if you develop something new, that you properly characterize your system. Think about complexity, how complex you want to be. If you go for the lung, you should work at the air-liquid interface. Keep in mind dosimetry. The smaller your models start to become, can be very difficult. And all models have advantages and disadvantages. So it's your research question that will drive the choice. For those that have still time, in 3 weeks, there's a very interesting conference in Europe, in Nice, Live 2020, where the lung model environment will be collected. And with this, I finish. Some more Luxembourgish words. Merci vielmals. And I'm happy to answer questions.
Lovely. Thank you very much, Arne. That was a really great introduction to the lung MPS field and lung in vitro field in general. We've got time for one very quick question, but then we're going to have to move on. All right, lovely. Thank you very much, Arne. Cheers.
Hi. So our next speaker is going to be Casper Rengili, and he's from Philip Morris International, USA.
Thank you very much. It's great to be here. I will show you our development of the human aerosol test platform, Humimic Inhales, that we do together with tissues for the evaluation of respiratory toxicity and systemic effects of inhaled aerosols. So I will talk about the challenges that we face with aerosols, how we want to model them in vitro, and show you the approach we use for that, and then the combination with the tissue system. Aerosol is a suspension of liquids and or solid particles in a carrier gas, and they can range in various particle sizes from around 1 nanometer to 1 millimeter. The properties might be altered by order of magnitudes depending on different conditions like temperature, time, distribution. And you can see here some examples from welding fumes that are very small, almost gas-like, up to pollens, for example, or mineral dust. And these particles have different shapes and structures. There are liquid spheres up to rigid pointy needles, like sprays or liquid particles. Then you have things like clouds that are around solid nuclei, or even complex stuff like pollen that are just solid particles, or even needles or fibers. And if you take the anatomical complexity into consideration, it's not just these particles enter from the top and go to the bottom of your lungs. There are a lot of things influencing how these particles end up where they end up. That's dependent on flow speed, on surface of resistance. And you can see here these blue and red bars showing sort of the distribution of this, these properties within the lungs. And this results then in particle fractions that are supplied in blue and green that are actually deposited. And you see that the deeper you go down, the less big size particles you have that are absorbed or entered. So it really depends on where you get your aerosol. It's not straight line from top to bottom, you just get less there. It really depends, it's like a filtering system, and depends a lot on the anatomy. So if there's really no easy strategy to work with aerosols in vitro, because you cannot just store the aerosol and then put it on your cell cultures. They need to be generated or usually freshly as part of the experiment, and you have to bring them to the cell culture. So you have to be very careful because the aerosols change rapidly, and they usually, if you do it not correctly, they leave you with a biological response that's of limited relevance, and you end up with an assumption and not really what happens in the lung. There are various in vitro aerosol exposure systems. You saw a few before, injection systems, but there is also continuous flow system where you just deposit the aerosol on your cell culture. But there are limitations because this is the target doses are ambiguous depending on what cell culture you use. The parameters cannot be modified independently, and the in vitro dose is not a straight line from in vivo. So there are a lot of things that are unclear and not really easy to just translate into an in vitro system. So our approach is the inhaled system. I can also give you more details a bit later if you come to me in person. And essentially it's a system that drives the objective to simulate aerosol generation and uptake like in humans, that have simulate physiological volumes of the human head respiratory tract, that simulates the physiological effect of velocities, flow directions and flow patterns, and the physiological environment in the human respiratory tract, so that you have in vivo surface properties and surface volume ratios that simulate the human airway tract. And essentially that's what we generated. It's the independent holistic air liquid exposure system called inhalers. So it's an apparatus that's able to breathe surrounding air and operating, for example, medical inhalers. And it's a real life aerosol generation and sampling without the need of accessory equipment, and functionally simulating the complex human respiratory tract. I have a short movie. Can you quickly start that in the back? I think you have to press on it. Which describes a bit what we want to do or what the concept is.
The human respiratory tract is of stunning complexity. And airborne materials entering it show dynamic patterns of deposition on the airway walls. Wouldn't it be great if we could simulate this outside the human body? For instance, when developing inhalable drugs, we could perform preclinical trials in vitro and study drug efficacy and delivery simultaneously. Amazing cell culture models and multi-organ systems for this purpose are available, but we cannot make full use of their potential, simply because achieving realistic dosing is so difficult. At Philip Morris International R&D, we have developed a new generation in vitro aerosol exposure system, the Inhales, to overcome this limitation. The system is inspired by human respiratory physiology and anatomy and consists of a mouth module, an airway module, and a lung module. Cell cultures can be placed and exposed to test aerosols in each module. But how does it work? Let's, for instance, consider the use of a medical inhaler. The inhaler is mounted at the aerosol inlet of the inhales and actuated. The aerosol is pulled through the system by the action of the lung module and deposits on the cell cultures. Because the inhales closely simulates the conditions within the respiratory tract, the aerosol deposition is well aligned with the deposition in the human airways, and so are the biological responses to the exposure. This is the next generation of in vitro aerosol exposure systems, developed by PMI.
Okay, I think you get how it works. Oh, can you go? I can myself go back one. Still, okay. So the first version was really a prototype, essentially in a lab with, you can see here, a lung module, airway models, or models built as a proof of concept. We had tracheal module, a bronchial module, and base plate for the lung module. And this was very soon developed into a version 2.0. You see it on the card here. It has the exact same physiological parameters as a human, so that the volumes are the same. And you can see the different modules. And for example, underneath here, you can see, it doesn't work. Yeah, here you can see plugs where you could put in cell models, for example. So it has a nasal module, small airways module. I will not go into the details, but I'm happy to talk with you about that. It has a respiratory airway and alveolar model. So a lot of these modules are wetted, that you have the same surface as in your tracheal tract. We work with sponges and foams in order to get the same kind of surface area that you would have in the alveolar, and work with different materials and test items to optimize it. But we also have, as I just mentioned, these functional plugs to put in cell models and sensor equipment. And we have this is a full modularity system, and controlled exposure conditions are possible within this. So the opportunities we see are great, actually. You can see a lot of aerosol sources that are there, sprays, environmental sources, fires, medical inhalers, inhaled drugs, but also smoke, for example. And these aerosol conditions, evolution, the portion of the inhalers can then be translated to the biology, and we can work with different aerosol dosimetry and evolution tools to expose the biology to different patterns of aerosols. And you can see where it can be applied. So now we come to the tissue system that I guess most of you already know. What we envision, or what we're working on together, is to implement this chip into the inhales in order to also add the systemic component to the system. So not only having the lung, but also what happens after deposition to the lung, for example, to the liver, or what happens if you have the uptake and then the metabolism. So we already did a proof of concept with the tissue chips a couple of years ago. Actually, it was published 2020 on combining the lung with the liver. And due to time, I will not go into much details now, but again, happy to talk about that. We showed that The organs, the lung and the liver, are stable over a 14-day period. And we did a proof of concept experiment with aflatoxin B1, where you can see that the liver can rescue the effects of the aflatoxin if they're cultured together, compared to if you only have the lung, then the lung significantly has effects or is dying over time, whereas with the liver it can be rescued. So essentially what we working on now is to do this, to enter these chips into the inhale system, namely in the trachea module. As you can see here, we built a holder where you can put the chip in, again here. So you can put the chip into this holder. You can see here the trachea module and a membrane that is the lung module. Then you have the reservoir, and here the second compartment, which is then the liver compartment, and the pump in the back. So you have the systemic circulation. We built a new chip for this. It is PDMS-free, and now with a specialized COC in the pump layer, where we have the different modules. So first you can see is the holder. Then there's the membrane, where the PET membrane, where the lung module sits on top. Then the union nut that screws the thing onto the chip, and then a breathable lid in order to operate the system. We built specific cell culture inserts to culture them in standard wall plates before we transfer them onto the Humimic inhales chip. And you can see here the membrane holder and the PET membrane on top. We also did some absorption measurements with different problematic compounds like testosterone, TNF-alpha, and nicotine, and you can clearly see that the chip, the COC chip, the new chip, is much better than the PDMS chip, because we don't have the problems of absorption of compounds within the system. You will be able to hear more about the chip at the tissues booth, or also at other talks of tissues. I think it's quite a good improvement of the chip. And here you can see our first cell cultures that are compared to transwell cultures. You see here on the first slide the assembled Humimic chip in the holder, and then transferred after extraction of this holder, and then it's assembled on the inhales chip. And the next step is then to have the breathable multi-organ chip in the breathable aerosol exposure chip system, and sort of have everything combined in one. The aerosol exposure, then the systemic effect of the aerosol to the liver. And there are many other multi-organ chip assays available that you can implement instead of the liver, obviously. The limits are, the sky are the limits, or whatever. But anyway, I think we have a lot of options to look at aerosol exposure combined with the systemic component. And with that, I would like to thank you for your attention, and thank this great group of people that work with me on this tissues, and also internally at PMI. Thank you very much.
Thank you for the wonderful talk. I think we are going to be holding off questions for this session. So please hold off your questions, and then at the end of the session, you can come and ask the speakers the questions.
So our next speaker is Mirjam Kina. Are you here? Hi. And Mirjam Kina is from Alveolix. She'll be speaking about an advanced drug screening platform for respiratory viruses.
Thank you for the invitation to present our project here in this setting at this nice conference. So what I am working on is basically trying to develop a new platform, which is an in vitro platform for drug screening for respiratory viruses. So why are we interested in this? Of course, you all know that basically the respiratory tract is exposed to inhaled particles, which also includes viruses. So there are several circulating viruses that are normally there, and still they can infect actually all anatomical site of the respiratory tract. However, the most severe disease or fatality rate is observed in lower respiratory infections, and it is still among the top 5 causes of death globally per year. So the last event actually that happened and also threatened the public health was actually the emergence of a SARS-CoV-2 virus, which I think by now you all know is a coronavirus, and it has these typical spike proteins on the surface, which bind to the host cell. The receptor for SARS-CoV-2 is AC2, and the processing by TMPRSS2 basically allows the virus to enter the cell and infect the cells. So also this virus, it can manifest in all sides of the respiratory tract. In the upper airways, it can infect mainly the ciliated cells or also secretory cells, but the symptoms are usually mild, despite high viral titers that are reached in this area of the lung, of the respiratory tract. However, severe manifestation is observed once the virus reaches the alveoli and the distal lung, where the primary target cell are the 82 cells, which then leads to massive inflammation and microvasculature activation, and finally also to the breakdown of the air-blood barrier. And diffuse alveolar damage is a common pathological manifestation that is observed in the lung of diseased COVID-19 patients. So there is a lot of progress that has been made since the emergence of SARS-CoV-2, but there is still limited knowledge about the mechanisms of COVID-19 and infection into the cells, and also we have limited possibilities to screen novel drugs. And therefore, this is mainly due to the limited or to the extreme difficulty to model the alveoli in vitro. So currently, a lot of studies are still working with animal models, and in the context of COVID-19, they are also suitable to some extent, so they can recapitulate the full complexity, and they also develop COVID-like symptoms, such as age correlation with disease severity. And of course, then we also have the systemic response that is best to be modeled in animal models. But the limitations, particularly with COVID-19, is the species tropism for SARS-CoV-2. So basically, for example, mice are not a natural host of this virus. And also the anatomical and immunological interspecies differences that we have between the animal model and the human patients. And also usually the animal models that we have for the infection develop less severe disease and usually spontaneously recover, which does not recapitulate the severe progression of severe COVID-19 that we see in some patients. So therefore, that's actually the aspects that could be better modeled in vitro models that are human-derived. And there are currently advanced in vitro models at our hand, which provide also 3D complexity, such as the organoids, the 3D in vitro tissues, and lung-on-chip models. And we actually plan to use these 3 in vitro models to develop Alvirix, which is this drug screening platform for respiratory viruses that I mentioned before. So basically what we would plan to do is to develop a medium throughput in vitro model, which mainly will be organoids or transwell inserts, to pre-test some compounds and then only validate the most promising candidate compounds on the most complex in vitro model that we have, which is the lung-on-chip model. So starting to derive the organoids, which was the first step of this project, because we have a source of a very high quality primary alveolar epithelial cells, and we started to optimize organoid culture conditions for this cell type. So we isolate them from the alveoli of donors, And as you can see, putting them into culture, basically they start to form organoid structures within few days. And we also saw that they express typical type 1 and type 2 alveolar epithelial cell markers. And also looking at some additional 81 and 82 cell markers and comparing them to the starting material, but also to primary alveolar epithelial cells that are cultured on conventional transwell inserts, we actually saw that the organoids were quite well in preserving the alveolar epithelial cell characteristic. So basically, it was notable that especially the 82 cell character was much better maintained in the organoids upon 2 weeks in culture compared to the transwell inserts. And also at the next step, we also had a look at the host factors that are required for virus infection of these cells. So we also found that ACE2 and TMPRSS2 is expressed in our organoids at a lower level than with the starting material, but still we could detect in all the donors these 2 host factors. So to summarize quickly, the advantages in our organoids so far is basically that we have alveolar subpopulations or subpopulations of alveolar cells that form heterogeneous organoids. And also we have an enhanced preservation of 82 cell character in our organoid cultures compared to transwell culture. And also they express relevant markers for disease models. So in my case, this would be for COVID-19 models. We did not yet progress to infection protocols since this is very precious material. But as a next step, of course, we would like to work on the upscaling of our culture method for the organoids, that we can use them for toxicity pre-screens. And also then afterwards, we would like to try to infect them, that we can also apply them for efficacy screens and actually treat them with drugs. And of course, they're also a very nice tool because they improve the handling and the storage and expansion of primary alveolar epithelial cells, which currently is still a limiting factor to use these cells in vitro. So then I would actually like to switch a bit to the monolayer models, which are the lung-on-chip and the transwell models. And here, as I mentioned before, we have not yet used the primary cells because it's very precious material. So we use as an alternative cell line models. And we would like to actually develop these models for the different anatomical sites of the respiratory tract. So for the upper airways, we use the BCI NS 1.1 cells, which have been originally derived from basal cells of bronchial brushes. Then we have a model for the small airways. We use the CALU3 cells, which are a very nice model or a very standard model also in SARS-CoV-2 research. And for the alveoli, we are using an in-house generated alveolar epithelial cell line. So for this talk, I would like to focus on the lower airway models, so basically on the CALU3 cells and on our alveolar epithelial cell model that we have here. So starting with the development of the medium throughput in vitro models, basically the transwell insert cultures, we had first a look at the CALU3 cells, and you can see as a first experiment, we actually checked if the submerged culture or the Ali culture is better, or if we should really go into Ali culture or not. And what we see actually here is that already the CALU3 cells express higher levels of the AC2 and TMPRSS2 in a liquid interface culture, also highlighting the importance of Ali culture for the lungs. However, when we infected these cells, we did not notice a difference in the infection rate between submerged culture and the liquid interface culture for the CALU3 cells. And also, when we looked for the virus production, basically these cells are very susceptible to SARS-CoV-2, and we got a very high virus titer already after 24 hours of infection in both conditions. And this coincided also with the breakdown of the barrier, so we really observed cell death, and this of course led to the breakdown of the barrier. Then next we asked ourselves if we can transfer or just translate this protocol that we have for the CALU3 cells to the alveolar model. in the transverse. So this is basically, as I mentioned, we are using these alveolar epithelial cells that are in-house generated. So they are derived from primary alveolar epithelial cells and have been immortalized. And they contain a mixture of 81 and 82 cells. And we have also optimized them for on-chip application. As you can see, they maintain a high barrier function over extended culture in submerged and in a liquid interface culture. So I will not go into more detail into characterization, but my colleague has a poster as well if you are interested to pass by. So we also checked for the expression of host factors of SARS-CoV-2, and we found actually that they express AC2 and TMPRSS2, and also similar as in the CALU3 cells, we see that these host factors are higher expressed in a liquid interface culture as compared to submerged culture. So then we also infected them in a similar setup as before, comparing submerged culture to a liquid interface culture. And what we see here really strikingly is that in submerged culture, we have only very few cells that are infected with the SARS-CoV-2 virus, whereas in a liquid interface culture, we have cells that are infected already at 24 hours, reaching a peak at 48 hours post-infection, where almost 40% of the culture are infected by the virus. And then somehow these cells get rid of the virus after an additional 24 hours. When we look at the virus production, then we really see that the liquid interface culture is the only method that was able to produce high viral titers. So at 48 hours, you can see in blue this peak of virus production in a liquid interface culture, whereas in submerged culture, we have a quite inefficient production of the virus. And also when we looked at barrier integrity, basically in submerged culture, the cells are still happy, they are still there, they do not care about the virus. And instead, in a liquid interface culture, we have this breakdown of the barrier at 48 hours that also coincides with the high production of the virus. So this highlights the importance also of a liquid interface culture. But now this was quite an easy system that I showed you, but there we miss also some components that are present in the alveoli in the human. So the alveoli are not only consisting of epithelial cells, the 81 and 82 cells, but also of endothelial cells that are making the air-blood barrier. And also we have immune cells in the alveoli to clear infection. So this can be better modeled in an organ-on-chip system. And I'm using the system from Alveolix, which is this is the AX12 plate that I'm working with. So you can see this is a 96 well plate-like format. And one unit consists basically of 3 wells, but the cells are only seated in the central well, where we have a very thin membrane, where you can grow epithelial or endothelial cells on both sides of the membrane. So you get a very thin co-culture model. In addition, they incorporated stretch into the system. So by adding a microdiaphragm into their plate system, they can actually move the membrane and mimic the in vivo-like breathing motion. To handle this chip or to access the chip for experiments or to create the breathing function, you need 3 modules. Basically, you see the AX12 chip again that is on the docking station, and the docking station then connects the chip either to the breather, which is required to make the cells breathe, or to the exchanger to manipulate the cells or to do medium exchanges. So if you are also interested in more into the possibilities that you have with the system, you can also attend the talks of my colleagues from University of Bern and from Alveolix to present you some additional models. But here I will just introduce the COVID-19 model. And again, starting with the small array model, so with the CALU3 cells, we have cultured them on the chip. And we also asked if the addition of the endothelial cells makes a difference for the infection of this cell type, of the CALU3 cells. First, what we noted is actually that once we introduced endothelial cells in the system, the CALU3 cells had enhanced barrier formation. So already after 3 days, we had really a tight barrier, which was significantly tighter than without the endothelial cells. However, upon addition of the SARS-CoV-2 virus, we saw that in both systems, we had efficient infection of the epithelial cells. So there were a lot of them positive for SARS-CoV-2 proteins. And we could also not detect infection in the endothelial cell layer, as you can see in the co-culture model. Also, we found efficient production of the virus in both systems, so in monoculture and in co-culture. And also we saw the breakdown of the barrier in both models. So we can see the infected cultures, the red bars have a breakdown of the TR, whereas it's still intact when the cells were not infected. So we also tried to translate this protocol to our alveolar model. As you can see here, first checking our immortalized alveolar epithelial cells in monoculture, we found that at low SARS-CoV-2 titers, we have a little reduction of the TR, but it is not very, very high. And also the virus production was very inefficient in this setting. Upon addition of the endothelial cells, instead we had a production of the virus that was increasing over time. Still, it is quite unproductive if you compare it to the inoculum, as you can see on the left bars, but we have an increase in the production of the virus over 48 hours. If we compare the efficiency of SARS-CoV-2 production between the monoculture and the co-culture, we actually see a significantly higher titer if the epithelial cells are in co-culture with endothelial cells. So this gives us at hand two models to model SARS-CoV-2, and also highlights the importance of choosing the correct setting and complexity of the model to achieve infection in these in vitro models that we have. To shortly summarize, so we have the model for the lower airways, the callus cells. They're very susceptible to the virus and produce high viral titers. And we also observed that the infection always results in breakdown of the barrier. But we saw that the increasing the complexity of the model did not have a big influence on the infection dynamics. So basically, still we saw that a liquid interface promoted the expression of host factors in callus cells, but had little impact on the infection efficiency, as well as endothelial cells did not further enhance the infection in these model systems. Instead, in the alveolar setting, we were working with the immortalized alveolar epithelial cells. They express SARS-CoV-2 host factors, but they are unproductively infected in the monolayer cultures or in our model systems. However, upon increasing the complexity of the model, we were able to achieve better infection rates. So namely, we saw that in a liquid interface, it was crucial to be in or to infect the cells and observe the barrier breakdown, as we can also see it in patients. And also we saw that endothelial cells further enhanced the release and production of SARS-CoV-2 virus in the infected cultures. For future perspective, actually we plan now to characterize these models that we have now a bit better regarding their antiviral response and immune response, to evaluate if this is actually biologically relevant to what we see in the COVID-19 patients. And as a next step, we would also like to start developing a protocol for drug screening that we can actually apply this platform to screen for therapeutics for COVID-19. And with this, I would like to thank my colleagues from University of Bern and for Alveolix that made this project possible. And thank you for your attention.
Thank you for the very interesting talk, Mirja. And as we said, we don't have time for Q&A at this point, but what we're going to suggest is we have a group lung Q&A session after. So if you'd like to have any questions, please come and meet the speakers after the session.
And our final speaker for the session is Mark Shearer from the NIH.
Thank you for the introduction and thank you for having me here. Today, I'll be giving you a little bit of an overview of the program that we have set up at NCATS using engineered 3D lung tissues as assay platforms for drug discovery. Just to give you context, so at NCATS, for about 4 years ago, we established the NCATS 3D Tissue Bioprinting Lab as a multidisciplinary group of bioengineers, assay development scientists, and drug discovery experts to biofabricate and validate microphysiological systems and implement their use in the drug discovery and development pipeline. It does take a village to take these tissues, to develop, validate them, and use them for drug discovery. As the name sort of indicates, we started historically using bioprinting methods, but in the last few years, we've been expanding to other MPS platforms, including spheroids and tissue chips. And today I'm going to give you some examples of the work that we're doing on this regard. Probably most of the work that we're doing is in collaboration with technology providers and disease experts, because one of the fundamental aspects when we start developing a tissue is what do we need to include in the tissue, and that it's really with the help of clinicians and researchers that tell us, well, I think you need to have these cells. And first we, like most of everyone is doing, first we develop the normal tissue, and then once we have a validated tissue to the normal, then we introduce some sort of perturbation to create a disease model. We have a lot of emphasis on high throughput screening in our work. So most of the tissues that we're developing are in multi-well plate platforms that are compatible with lab automation found in HTS labs. And at the end of the day, really, if we can, and we use clinical approved compounds to validate, pharmacologically validate the model. So during this process, we do spend a lot of time validating the tissues, and by that means assessing that we have the right cell markers and the right maturation. Do we capture the right clinical biomarkers that we want to see in these tissues? And there's been a lot of talk in by previous speakers and this morning and debate discussion about like how you assess that these tissues are physiologically relevant. And it really depends on the questions that you're trying to ask, what disease, and the previous speaker gave a perfect example of that. And because we are very much HTS focused and screening focused, we're very interested in developing quantitative functional endpoints that we can use for screening. And again, I mentioned the pharmacological validation, and also something that came up this morning in terms of operationalization for HTS, the scaling up process, the assessing the robustness, the variation from well to well, the variation from plate to plate. So we can really use these platforms as drug screening platforms. At the end of the day, our goal is to implement these platforms in the drug discovery and development pipeline. So trying to make or help make better go, no go decisions when compounds are selected during the process. So a lot of the work that we're doing, as you will see, is establish fit for purpose. We have a lot of different technologies. How do you choose the right technology for the questions that you're trying to answer? How do you establish the context of use of each one of these models? What physiological complexity do you need? And what is the clinical predictability? We say we want to make these models because we think they're more clinically predictive. And the first question that you get is like, well, show me that they are indeed more predictive. So how do you establish that, right? So today I'm going to be talking about a platform of engineered 3D tissue models that we've been putting together in the last few years. Two words that I like a lot are modular and versatile. It takes a lot of work to create one of these tissues. And once you have the normal, let's call it the normal tissue, you want to try to leverage it for as many diseases as you can, whether it's cancer, for lung, whether it's cancers, whether it's infectious diseases or toxicity applications. So having a process to build these models and then being able to, in a plug and play way, to use them for different diseases is really something that is very powerful, and we try to do. So for the lung, and this is a general approach that we use in the lab, modular in terms of diseases, but modular in terms of physiological complexity. And that helps us sort of answer some of the questions in terms of exploring the context to use and exploring the fit for purpose. And I'm not going to go through some of the basics of the lung, but for the lung, we've been leveraging sort of existing commercially available tissue models of the different areas of the lung, whether it's the tracheobronchial, small airway, or alveolar. And also we are developing our own platform. So we have used sort of microfluidic platforms that are available. And really, our choices were based on a lot of it on throughput. As you can see, there's a lot of different long MPS platforms. How do you choose? So in our case, we choose the Metas organograph plates because it gave us the throughput in terms of developing some of the models, and because we were able to leverage some of the protocols that we were developing for the more transwell models. So the models that I'm going to be talking today are sort of commercially available, sort of long epithelial transwell models. And then some of the vascularized lung tissue models that we're developing in-house, and some of the work that we've done with perfused vascularized lung epithelial organ-on-a-chip models. For each one of these models, essentially go through this sort of parallel physiological characterization, whether it's with histology, whether it's with microscopy. And lately, we've been using also single-cell sequencing in terms of assessing the different cell types that we have in the tissue. At the same time, these are very low throughput type of assays. So we use functional HCS measurements with the goal to really being able to implement screens, whether it's cell viability, whether it's measuring a tear, whether it's looking at cytokine secretion, and we leverage a lot high content imaging as well. So for the lung, we started using the commercially available. In our case, we use the MATEC transwell models that have been mentioned before. These are essentially epithelial models. So we use them because they were readily available. They were in a transwell, so they're sort of amenable to using instrumentation that we use for high throughput screening. So they come ready to use. You mature them in the ALI for 3 weeks. These are from primary epithelial cells, whether it's alveolar or from the tracheobronchial. And then we use these models particularly for as a sort of quick way to get into viral infection studies. So that was at the beginning of the COVID-19 epidemic. So we wanted to assess, can we use these models for studying viral infection or respiratory viral infections? So we use them, and then we apply different assays, whether it's direct measuring of viral infection using antigen staining and microscopy, looking at secreted virus. We also measuring multiplex cytokines. And then, as I mentioned before, we started doing validation using single-cell sequencing and as well as tier measurements. This has been out in a preprint and now has been accepted. So I'm just going to go quickly over the data, some of the basic data. So what we did was really systematically test the tracheobronchial tissues versus the alveolar ALI tissue models, in this case from MATEC again. And we assess infectivity. So in this particular slide, oops. I'm showing this is the marks, so the blue is DAPI and the green in the middle is for the virus antigen. So this is the uninfected. So the top one is the tracheobronchial, the bottom one is alveolar. So you can see that these two are two strains of influenza, and this is one of the original strains of SARS-2. So you can see the differences with the different viruses in terms of time. So this is time after infection. So some are early, some are later, which is what was seen for SARS-2. And again, there's the differences between the tracheobronchial and the alveolar tissues that have been mentioned before in the in the previous stroke. So these are, I would say, simple, differentiated lung epithelial models, but they seem to recapitulate quite well some of the clinical features of the SARS-2 virus and influenza. So for this application, it seemed to be a good model. We also measure cytokines, and I'm not going to show all the data, it's in the paper, but one of the cytokines that clinically appeared to be that relevant for COVID-19 was IP10. So here I'm showing the data with IP10, and again, what I'm showing is for the tracheobronchial tissue, and then this is the alveolar, and you can see the differences between the influenza infection in terms of secretion of the IP10 cytokine, very quick, versus the SARS-2, which is takes longer time. There's a lag. There's less of it, and then the differences between airway and alveolar much higher in the alveolar for SARS-2 and also for the influenza. So you can tell the differences, and you can explore the differences between viruses and between tissues, and that sort of in agreement with the data that was shown by the previous speaker. Again, we did single cell sequencing, and I must say we're still digesting the data. It's a lot of data, but essentially we did it for the tracheobronchial, like non-infected, infected with influenza, non-infected, infected with the SARS-2, and then with the alveolar tissue, again, mock, influenza, mock, and SARS. And you can start seeing the differences again within the virus. We were able to detect all the relevant cells, but then we were interested in seeing the changes with each virus and try to correlate that with clinical responses. Can we sort of try to see if we can, in a way, use these tissues to predict clinical responses from these transcriptomics or single cell transcriptomics signatures? And there's genes that are common and a lot of genes that are not common. So it's a lot of very interesting data that, again, we're still digesting, but sort of hoping that we can then go to the clinical data and do associations with these data in the tissues and see how close we are in terms of predictability. We work with Dr. Sarah Cherry at Japan, and she had a platform for screening compounds in 2D. That was a high content imaging using CALU3 cells that were infected. And then they were using high content to look at viral antigens. So they did a screen of several libraries. And the idea here was to, again, can we use these ALI models to help prioritize the compounds before going into animal models? The whole concept of can we use these tissues to help make better selections? So this was published, but it was actually very useful. And here's some of the data for the SARS-2. You can see the infection here. This is the mock infection. This is influenza. Mock infected, and then you can test compounds. And all these compounds actually were active in 2D, let's call it 2D. And you can see that some of them really confirm in the 3 models, but many of them didn't. So we think that these are good models if you want to prioritize compounds for putting into animal models. The limitation with these commercially available models is that what you get what you get, right? So if you want to start exploring additional physiological complexity, they're very limited. And in addition, they're like lower throughput. So we actually spend time trying to develop, we are developing 96 well plate trans-well models. And this is some of the data that we have generated with finding the right markers, the good tier values. And then the other thing that we're doing is we had developed protocols to include vascularized beds in the tissue. So we do have a transcriptomic data indicating that the transcriptomic signatures of endothelial cells in 2D or in monolayers are very different from what you see in sort of forming capillaries. So we developed protocols, publish this data. So I'm going to go quickly. So we mix endothelial cells, fibroblasts, parasites. We print them at the bottom of a transwell, and then after a week, we see nice angiogenesis and vascularigenesis. So we applied this protocol. We knew we could make the the long epithelial, could we make the vascularized bed under the long epithelial? So it's work in progress, but essentially we're taking a modular approach that was mentioned before. We take some protocols to make the vasculature and then protocols to make the epithelial. Can we put them together? And this is actually working quite well. So I'm gonna go on the donut structure in a second, but essentially this we're doing in a 96 strand well. This is the bioprinted vascularized bed. And again, this is to show you the reproducibility of the protocol. And you can see this is, this particular model, and I'll go into it in a second, is for cancer. But if you don't add the small airway, you get this donut where you can see the vasculature forming. You can then add the small airway epithelial cells, and you do have an effect on the vasculature, angiogenesis and vasculogenesis. And this particular model was for cancer. So we added the cancer cells, and you can see the formation of the small tumors in the model. So again, showing you that we can develop these vascularized beds in a transwell format. And the reason to do a donut is it helps us with the quantitation. So we can easily create or use high content imaging to create masks and look at angiogenesis. So it's very important for us to be able to quantitate phenotypes if we want to use these models for screening. And in this particular case, it was a cancer model. So again, we can also look at the cancers. We can see whether the tumors are within the vasculature or somewhere else in the model. And that way we can, again, have a disease model, a quantitative assay for screening for this particular disease model. This model didn't have a perfused vasculature. It had a vasculature, it was in a transwell, but it didn't have a perfused vasculature. So we recently published this model. This is the one where we use the organoplate graph-friendly method. Essentially, it's a 3-channel chip. The outside is liquid. That's where we load the endothelial cells. And in the middle, there's a hydrogel where we add the mixture of the endothelial cells, parasites, and fibroblasts to make a vasculature. The beauty of these plates that we like is not only that it has throughput, but it has an opening here where we can put the long epithelial cells and create the air-liquid interface on top of the vascularized bed. And the protocol is actually very similar. It took a lot of work to optimize, but the bottom line is at the end, you can have a nice epithelial layer with very decent tier values, and you can, with the sort of modular way, you can investigate what happens if I don't have the alveolar cells, and really, if I have alveolar but not vasculature, and really you can start investigating the crosstalk between the cells and how that affects the function of the tissue. So we were able to make the epithelial layer in the ALI, and then we were also able to demonstrate that we could make a vascularized bed, and here are the markers with the microvasculature and the network, that it was perfusible. And we used a 70 kD dextran to actually show that we could fuse from one channel into the other channel. And we're still working on it, but again, I mentioned that we care about throughput. So we don't want to show just the one well that works. Here is the data from some recent experiments on one of these 64 plates, and it's actually looking very good. We have some of the wells that we're still working on, but overall we can see, and this is the progression from the beginning until the ALI, I think it's day 14. And here is just a blow up of one of the wells. So you can see the endothelial cells in one channel, the other, and the vasculature. And this is the whole plate. With a couple of wells here that I think last week they said that they're coming up with protocols that look very good now. So scaling, reproducibility, it's important for us. And how do you know that you get the same in every well? So you have to quantitate. So that's what we're working on before I go into the quantitation, just to show you that it's not only that you can form the vasculature in every well, but there's perfusible. And here is I'm showing the data, and as you can see, it's not perfect yet, but we're working on it. And sort of playing with the conditions that you have the right sort of diameter of the capillaries. So we use these models, it's work in progress again, to see infection. So it's not only about seeing infection, it's also how does infection affect the vasculature? Is there any effect direct or indirect on the integrity of the vasculature. And that's what we are doing lately. So we are quantitating. We're working with Gilles Carpenter in France to develop algorithms that allow us to quantitate the integrity of the vasculature, whether it's a sprouting, whether it's mesh area, whether what percentage of the vasculature is really perfusing. And as you can see, when you infect, in this case is with influenza, some of the parameters don't change, some do change. And that's the beauty of these systems. You can look at different parameters and see which one changed depending on the disease model that you're creating. This is it for me. I think it's a quick overview, and just to remind everyone that it's not only just making the model, but finding the right cells. And really, you generate a lot of data, in this case, high content data, that requires a lot of support in terms of IT. So it's a whole infrastructure that you have to create to be able to use these models for high content screening and implement them as a pipeline. And this is the group, and thank you, everyone.
Thank you, Mark. Let's thank all the speakers one more time. And then if you have questions, please come up.
Okay, thank you, everyone. I think we're going to get started with the next session. This is the Barrier MPS, and our first speaker is Anna Herold from the Karolinska in Sweden. Yeah, Anna, the floor is all yours.
Thank you so much for the introduction, and thank you so much for giving me the opportunity to talk today. I think it's a wonderful meeting, and it's quite exciting to speak in the barrier session. It feels like going back to when I was at the VIS, and I was always kind of the barrier to all new experiments. But that's history now. Yes, for sake of time, I start with the acknowledgement slide, so we don't miss thanking anyone. Okay, now I see actually that the presentation was not really updated. No matter, there's a lot of people here today that I want to acknowledge for some of the work. So I want to start with Don, of course, but also mention Ben, Maus, and Tayun, that will luckily speak after me, so that's easily, and Andries van der Meer is also here. And then, of course, acknowledge my own group now and funding. So I focus a lot on engineering the neurovascular unit, and that's an excellent barrier to start with. And when you talk about the barrier, then often we talk about the blood-brain barrier. So what we are interested in there is, of course, understanding how the neurovascular unit controls drug penetrance. That's one reason, but also generally how it controls brain homeostasis and how it's affected in neuropathology. And I mean, a quick review what it looks like. We all know that, you know, you have a capillary with endothelial cells and circumvented by pericytes and astrocyte end feet, and also outreaching neuron processes. And we worked with a couple of variants of how we want to model the neurovascular unit. And I've helped classify them a bit in our 3D approaches. Oops, sorry. Let's see if we can get this pointer. 3D approaches, where we have a monolayer of endothelial cells lined with different perivascular cells. These are very interesting for studying cellular interactions, but they can be a little bit more difficult to get perfectly reproducibles. And me and Andries did a nice study there back in 2016. Today I will talk a little bit later about a study that we more recently did, where we have a more controlled way of designing where we create the 3D-dimensional model. For more quantitative approaches, we approached the way of doing more this 2D layered chips, often now recognized more like the VIS layout of chips. And I will describe today a little bit about how we did a new model of that with a material that easily integrates multi-materials in one fabrication steps. But if we go back how we started on this, and when we were at the VIS some years back, we had the task to model neurovascular units. And we actually at that time started thinking how we can get more data out of it, of each part of the unit. So we disintegrated it into different kinds of units that we could sample individually. So you assume it won't be so easy to see what you point, but what we did then was disintegrating neurovascular units into 3 chips. So one blood-brain barrier, one more brain chip, and another blood-brain barrier that were connected through their secretome. So you have the mimic of the bloodstream here, and then the connection through the secretome. And the chip layout, you know, you have the beautiful cartoons, but then you have the chip layout looking something like this. And what we did then was, of course, what we all have to do with a different kind of multiphysiological systems that we do. We had to validate it for human characteristics. We could see that the response to certain drugs, with alternating the barrier function, we can look at different penetrance of drugs and also cellular interactions. At that time, we worked all with primary human cells. And what we have been working much more in my lab after that is actually transition this to more IPS-derived systems. Simply because we want to be applicable also in industrial setting and to be scalable. So I don't have to introduce induced pluripotent stem cells to this audience, I think. So we kind of skip that slide, but just to have something that we can get an unlimited cell source that can be applicable in wide settings. So the different kind of cells that we work on mostly when it comes to neurovascular units is, of course, the endothelial cells and the glial cells and the pericyte cells. We also do work with neuronal cells, but I won't go into them today. So when we start thinking about iPS-derived cells for the endothelium, we back then in 2018, in a collaboration with AstraZeneca, did a comparison between two major protocols. One more, the Wisconsin protocol, as I call it, and the other one more, the protocol that was from Leiden University. And in our hands, when we worked with those two protocols, our ideal what we wanted a brain-like barrier function. And our two metrics that we compared was the transendothelial resistance, and if you take the Wisconsin protocol, that is the protocol 1 in the slide, and the other protocol is protocol 2. And we could see that the barrier function was much higher in the protocol than the Wisconsin protocol. And when we wanted to compare apparent drug permeability, this was the only protocol that actually could discriminate CNS penetrating drugs from non-CNS penetrating drugs. And that was our motivation to continue with that protocol. And what we did then was actually a study that was done mostly by my colleague here, Tayun. And I will just highlight one thing that I thought was very interesting with this study. It was that we took this Madison protocol, it was improved by hypoxia treatment. And we looked at if we can discriminate transcytosis rates with this protocol. And we used a well-known system, the transferrin receptor targeting antibodies, and compare the transcytosis rates between one antibody that is known to penetrate the blood-brain barrier and one that actually gets stuck into the cells. And thanks to this improved endothelial protocol with hypoxia, we could actually on chip, on a blood-brain barrier-like chip, we could discriminate these two antibodies with real-time analysis. And in this chip, what we had was then this improved endothelium, but the rest of the cells, the perivascular cells, were actually primary cells. What we have done now lately is actually continuing looking at different kind of transcytosis processes with a variant of this endothelial protocol, the accelerated protocol published by Neil 2019. And so we have done a number of different transcytosis studies, but this is one where we use lipid nanoparticles. They were coated with chondroitin sulfite, and also they had a blood-brain barrier penetrating peptide from the TAT sequence, which is HIV derived. And when we did that, those nanoparticles had previously been shown to be strongly anti-inflammatory in the brain. And when we conjugated them with this TAT derivative peptide, we could see that there is no impact on the barrier function. These nanoparticles keep the barrier intact, but we could strongly see that we have it conjugated with the peptide, we have a much higher penetrance than if we have non-conjugated nanoparticles. And you can also maybe see the red dots here in this endothelial slide, whereas we don't see it here. So also this endothelium can support this transcytosis mechanism, which is actually debated what kind of mechanism that's going on there. And we could also not single out one transcytosis mechanism that was dominant, but that's another problem. Still, this endothelium supports the transcytosis. So that was kind of our validation of this brain-like endothelium. But it comes to the other cells then, and I will describe our astrocytes. So we have been working since long back to get scalable and functional astrocytes from iPS cells. And our general route is that we take the iPS state, and then we derive a stable neural stem cell state that we can actually pass it for over 100 passages without the large drift of the neural stem cells. And then we have done a couple of different protocols where we derive astrocytes from them. And just very recently, in a month ago or so, we published this paper, which is a very simple protocol If you follow it, it's just an astrocyte growth supplement that you give to the astrocyte during 28 days. And then we get a strong upregulation of astrocyte markers. We don't get upregulation, of course, of neural markers. We got a strong expression here of 3 lines. We show astrocyte markers stably expressed. These astrocytes, as it's not only the markers that you need, they're also have a strong inflammatory capacity, and both with IL-6 and IL-8, and also ICAM expression of inflammatory stimuli. And interestingly, they also have a very nice glutathione synthesis and secretion, which is actually on par or even better than if we compare with fetal primary astrocytes. And now when we have all these components, we recently integrate them together in a new barrier chip, which was made in a one-step injection molding as a material called off-stoichiometry thiolene epoxy. The beauty with that material is that it has both epoxy bonds and thiolene bonds. So when you do the injection molding, what you actually can do is that you can integrate materials like a good bonding of metals or a tracheous membrane, in this case of polycarbonate, into the membrane directly. So to monitor the barrier here, we actually did impedance-based barrier sensing, as opposed to the conventional tier. The electrodes are just integrated on the membrane. You can select your own frequency that you're interested in, but we went somewhere in the middle there where we could monitor both tight junctions and a little bit of capacitive cell attachment. And we went about to study this model because we know one caveat with that endothelial protocol is that it doesn't actually support inflammatory processes very strongly. So we looked at how much it supported stress processes, and this is specifically neutrosative stress, where you kind of mimic microglia activation through a compound called SYN1 that degrades to peroxynitrite. And what you see there is kind of individual barriers that are formed. You see they're not all the same. You see there over those little spikes, that's when we do media changes. And when we then challenge it with this stressor, the SYN1, we can see that the barrier is responding to it. So this kind of stress, this endothelium is responding to. And we could also see that it was sensitive to a prophylactic treatment with a clinically relevant antioxidant called NACA, which actually then can save the barrier from this stress. And it was interesting, since we have the astrocytes there supporting glutathione pathways, we actually could see that actually there was a clear glutathione activation in this case. So I want to kind of quickly remind everyone about a small thing, was a publication we did last year. When we talk about barrier system, a lot of people tend to show these beautiful slides and just show the ship, and then you discuss about, okay, the ship must not be absorbing certain molecules either into the chip or on top of the material. We recently did a study to kind of validate how much does it matter. So we tried to do it in a totally realistic organ-on-chip prototype setting. And then many people just concentrate on the chip. And they don't really set up the whole model as it is. So, oh, sorry, this was where my slides weren't updated. So what many people do is, of course, they just look at the material and look how much sorption there is in it. But what we did was actually setting it up in a peristaltic pumping. And it was very clear, of course, it depends on what kind of surface area you have, your chip to whole system area. And I just want to raise the issue of the take-home message of our study is like, be really careful what tubing you use in your prototype systems if you want to do any drug studies into them, and avoid most kind of soft tubings. Then, of course, it's very interesting what material your little chip is also made of, but of course it relates to the surface area of the whole system. And I want to then quickly switch over to our last system, 1%, going back to the 3D models. Previously, where we worked with something called, where we did kind of a fingering process to generate lumens into gels. But we recently developed a method to do kind of direct laser writing, where we use a femtosecond laser that creates not actually ablation of material, but because of its intensity and very fast pulse repetition, we can get air bubbles or bubbles into the system that pushes the gel material that you have in your system to the sides. And that is actually an extremely gentle process. So what you get in that system is that what this slide shows here is that we actually printed our lumens just next to a glioma organoid, and it wasn't damaged at all. We also quantified actually damage to the side. And thanks to this really, really high intensity but fast process and just a kind of bubble formation, we can create this vascular where we want it around organoids, which we recently published. Okay, we skip that for sake of time, these full videos when we do this. So this was, anyway, I want to finish up the talk with just, again, emphasizing two things. One is we really need to improve our chips in many ways. One is integrated sensing that I think is one thing, and then the integrated systems that many of us are describing. And the other one is to continue improving the cell sources that are needed. And typically these must be iPS-derived cells. And with that, thanks for your attention. And again, thanks to all the people contributing to this work.
Thank you, Anna. I think we have time for one very quick question, if there is one.
Maybe they don't hear you soon, but I can repeat your question if it's short.
It works now. So just a quick question. Beautiful slide presentation. I already told you I'm a fan of your work, especially the last one. So I was wondering, are you also plan to work on the immune component of the neurovascular unit?
Yeah, we have a couple of microglia differentiations going on. As many people working in this. I mean, it's again, what function you're interested in, what studies. Sometimes, I mean, you want to have everything in the system, right? Every single component, you want all the cells there, and preferably, you know, really specific to what region of the brain you want to work with. But yes, we are trying that, but I'm not too happy with them yet, actually, with the whole context.
If that's all the questions, should we move on to the next speaker? Great. So next up, we have Tae-Yoon Park from Ulsan National Institute of Science and Technology in South Korea. Okay, over to you.
Okay, thank you for your introduction, and thank you for inviting me today. I'm really excited to introduce my ongoing project about the human blood-brain barrier model empowered by engineered basement membrane. So Anna already talked about the blood-brain barrier, so I'll make it really quick. So blood-brain barrier is composed of the lining of the brain endothelial cell, and then it's covered by basement membrane, and is also covered by pericyte and astrocyte. They are like supporting the function of the blood-brain barrier. And there are two important barrier functions. The first one is a physical barrier function over there. And then here, the level is around more than 2,000 square centimeter. And then it limits the transport of the molecule to the like a parasite transport. And then second function is metabolic barrier function. So there are a lot of efflux pump expressed on this, the brain endothelial cell. And then they actually like pump out the hydrophobic drugs. So those are the really complex and then unique physiological barrier. And then it's becoming the major obstacle to like a brain drug deliveries. And that is the reason why many brain drug fail in the clinical trials. So it's extremely important to understand about human blood-brain barrier and then find a solution to overcome the BBB. So I'll make it quick. And then if you're interested in blood-brain barrier and then brain drug delivery, you may have seen a lot of in vitro human BBB model. And then we started to think about the basement membrane because we often ignore the importance of the basement membrane when we generate the in vitro BBB model. And the endothelial cells are sitting on this, the basement membrane. Basement membrane is ultra thin, the ECM layer, which appeared as a lightly mattered, the feltwork of the fine fibril. It's really thin, and then it's making a kind of structure support to make a lumen. And then around there, there's a pericyte and astrocyte because brain endothelial cell directly sitting on this basement membrane. So physical property and the chemical property of this basement membrane is closely linked with the health of the BBB. So it's also linked with the pathophysiology in the brain diseases by regulating the cell property. And then this is the the BBB, human BBB model using the microfluidic device. And then I just brought this to show this typical strategy, having this two compartment. One side is brain side here, and the other side is microvessel side. And when we make the barrier MPS, we use this two compartment and separated by some porous membrane. It can be PDMS membrane, or it can be porous polyester or polycarbonate membrane. It's a very typical strategy. And then when you see the limitation of this synthetic membrane, you can see there are a lot of disadvantage. We have to choose between the small pore size membrane, the large pore size membrane. The small pore size membrane generally like 0.4 micron to 2 micron. And then larger pore size, it's more than 3, where it can be 10 micron. So if you choose the small pore size membrane, you can easily make a functional tissue barrier. And the problem is we cannot observe the cell migration or metastasis and so on. But if you're choosing the larger pore size, the functional barrier, making the functional barrier can be a bit difficult depending on the cell type, but we can easily see the metastasis and the cell migration between two compartments. So the first strategy we made was like using the larger pore size membrane, like a 10 micron the pore size, and then instead we built the ultra thin nanofilm on this porous membrane to make a beautiful the tissue barrier, while we can see the transport of the cells between two compartments. It's really easy the method actually. So in this microfluidic chip, we added 2 milligram per ml of a collagen gel, and after gelation and the dehydration, and then make it condensed on this PET membrane. And then we could see that after the 2 milligram per ml of a collagen 1 condensation on this PET membrane, you can see that this collagen is blocking the pore. So we can make a beautiful the monolayer of the endothelial cell or epithelial cell, whatever. But through this pore, we can make the cell penetrate. So you can see that because if it is like a larger pore, the cells can easily fall through to the other compartment. And then by blocking the pore using the ultra thin collagen, we can generate the beautiful monolayer of the brain endothelial cell. And then also we can see the interaction between two types of cell, like a brain endothelium and the astrocyte. And the astrocyte and the fit contact to the brain endo is really important for the function of the BBB. And we can see that like the red layer is brain endothelial cell, and the green one is astrocyte. And through this big pore, and the astrocyte can easily contact to the brain endothelial cell. So you can define that through this one. Astrocyte and the fit is touching the brain endothelial cell. So, okay, and then the second strategy, we start to think about the importance of the mechanical property of the basement membrane. When you see the basement membrane stiffness, it's only less than 500 kilopascal, and the thickness is really ultra thin, it's 200 nanometer. And then because endothelial cell or epithelial cells are directly interacted with this basement membrane, and then through this, the vinculin and then taline and so on, the mechanical property of a basement membrane can be transmitted to this property of the brain endothelial cell. And we're thinking if we mimic this BM stiffness and then chemical property, probably brain endo will be very happy. So this was the motivation of this study. And then for making like a soft, the basement membrane, like our in vivo basement membrane, we can easily generate a hydrogel like this. But if you're using the hydrogel, the problem is it's not easy to generate the hydrogel membrane in the freestanding configuration. So for integration into the like a microfluidic chip or the transfer system, it must be freestanding configuration. And then there are some studies showing that if you're culturing the brain endothelial cell in the physiologically relevant the basement membrane, the stiffness, the cells are happier. And the cells are showing like quite the different the property. And to overcome the problem of this hydrogel system, we were looking at the other strategy. That how to make a soft basement membrane, and then how to make this soft basement membrane in the freestanding configuration. And then we are thinking of nanofibers. So imagine this house, this is the frame, and then we can put the window and so on. So to make a freestanding basement membrane, we were thinking of a nanofiber as a skeleton, but it must be sparse nanofiber. And then we coated this nanofiber with the basement membrane ECM hydrogel. That was our strategy. Okay, actually, it's not the first study. That there was like a multiple study, 2 or 3 study, to showing the nanofiber scaffold supporting the brain endothelial cell. And then if you see the stiffness of this dense nanofiber scaffold, it's like more than thousands of kilopascal. It's more than the physiological, the BM stiffness. And then we could see that iPSC drive the brain endothelial cell didn't like show really strong barrier function. So it was kind of the failed model. And our strategy, as I mentioned, we can make a really sparse skeleton using the electrospinning method. And then we can incorporate this sparse, the nanofiber into this transwell insert. And then later on, by coating with the ECM hydrogel, we can make a soft basement membrane. And then it's ultra thin because it's a sparse nanofiber. And then to make a gelation, we used the collagen 1, and then it's a kind of a double coating. We added the collagen 4 and the fibronectin on this collagen 1 hydrogel. So we could achieve a freestanding the membrane by using the sparse skeleton like a nanofiber, like made of the polycaprolactone and the silk. And we could achieve mechanical and chemical property by using this ECM hydrogel. And if you compare with the PET membrane, polyester membrane, and then our new membrane, we'll call it the narrow BM. And you can see the clear advantage of using our the basement membrane. So if you see the Young's modulus to see the stiffness, let's say physiological, the BM stiffness around 500 kilopascal, it's quite close to our system, while the PET is showing like a 4-digit higher level of stiffness. And then also the membrane thickness was much smaller in our system. And then advantage of this membrane is it's much more, highly permeable. So compared to the PET membrane, when we're checking the 3 kilodalton and then 10 kilodalton dextran, you can see that in our membrane, it's much more permeable. So we could make a really thin and then having a stiffness which is in vivo level and which is highly permeable, the basement membrane. So we could see that because we could incorporate this membrane in this through the thermal bonding into this transwell insert, we could like make the crosstalk between the astrocyte and pericyte chemically. And then we have built up this BBB model using the PT membrane and our the BM membrane. And actually cell morphology were the same. And we are expecting that the cells, when they are cultured on the new basement membrane, they're going to show much higher expression of the BBB marker. But unexpectedly, it was not like that. It was all the same, like PT, Narabian, whatever, they were showing quite similar BBB marker expression. And then interestingly, we could see that the physical barrier function of a brain endothelial cells, when they are cultured on this Narabian, they showed much higher the enhanced physical barrier function. And then we started to chase the like a mechanism and the reason why the BM is providing this like a support, barrier support. And then we're looking at the stiffness. And then first, we wanted to see if our brain endothelial cell can feel this different stiffness between PT and our basement membrane. Then we look at the vinculin. The vinculin has two phases. The first one is it's a key mechanosensory protein in the adhesion complex, so it can make the interaction between the cell and the ECM. And also they can interconnect through these two like neighboring cells. So they are working on making this adherent junction between two cells and then cell and ECM. And there are many studies showing that depending on the stiffness of this like a matrix or the basement membrane, it is showing different accumulation of the vinculin, the location and the size and so on. So first, we want to see the interaction between brain endothelial cell and then ECM, the basement membrane. So we could see the different cell morphology when we're culturing the cells in the PT membrane and our membrane. And you can see that the vinculin is, the size of a vinculin is much higher when like a culture than the PT. And then it looks quite different from the cells in the Narabian. The vinculin area is increased in the PT. This is like a repetition of our study showing that when the stiffness of a basement membrane gets higher, the vinculin area gets larger. So that, which means that our brain endo clearly feel the softness of our Narabian. And then we started to see the cell-cell interaction and the cell-ECM interaction together. And then I'm going to show this first. And this is showing the dual role of a vinculin in the barrier disruptive and then barrier enhancing endothelial cell response. So this is another previous study showing that when we're adding the barrier enhancing drug, you can see the vinculin is around the cell-cell border. And we're adding the barrier disruptive drug. They are not in the cell border anymore. They're going into the cytoplasm. Okay, so going back, we found that when we are culturing the brain endothelial cell in the PT membrane, it's not showing the vinculin accumulated into the cell border. They're just scattered, but we are culturing the cell in the Narabian. They're more like recruited to the cell border. So which means that the vinculin has two functions, cell-ECM interaction, cell-cell interaction. And then, which means that when we're culturing the cell in the Narabian, they are more focusing on the cell-cell interaction. That's why barrier function gets higher. And then we have seen the reorganization of the affectin because the vinculin is interacting with the cytoskeleton, and we wanted to see if the vinculin accumulated on the cell border can make the affectin having this force and the cell-cell interaction. So when you see the cells in the PT membrane, when the brain endothelial cell were showing just a scattered the affecting structure, but when we are culturing the cell and the nano-BM, the affecting is very beautifully recruited to the cell border, which means that the vinculin is capturing the cytoskeleton and make a beautiful cortical ring. So it make the cell barrier much higher. So when we see the Z01 and the affecting colocalization, when you're culturing the narrow BM, it's much higher. And this is mystery, actually, where I talk about the physical barrier function and metabolic barrier function. And the metabolic barrier function, it's not easy to achieve. We can see that efflux pump function in the brain endothelial cell, but it's not easy to incorporate to see the transcytosis too. And we've like in our the last publication, we show that giving the shear stress was a kind of a key to activate the efflux pump in the microfluidic chip. And interestingly, when we're culturing the cell, the same cell and the narrow BM, we could see the enhanced function of this efflux pump, including the ABCG2 and ABCC4. We could explain why culturing and the narrow BM is enhancing the physical barrier function, but we don't know why cells have a higher activity of the efflux pump. So it's mystery. We are working on finding the reason why. And then the advantage of this model is because it's a very sparse skeleton and it has a super thin, the ECM hydrogel. We can easily see the cell interaction, cell-cell interaction, and also we can easily see the cell migration, the metastasis, and so on. So we build up the ischemic strong model to see the immune cell trafficking. And then we made an OGD model. OGD model is like oxygen and then glucose deprivation model. And then by making the ischemic stroke model, we could see the drop of the barrier function and the increase of the GLUT1, just like the ischemic stroke patient. And the important thing we could— 1 minute remaining. Sorry, thank you. The interesting thing is we could see the cell interaction between the two compartments. So in this stroke situation, we can see that the apical side immune cells are going into the basal side. So so this is a video, maybe it's not working. So we could see that the apically localized immune cell can easily penetrate the basal side. So this is really like a huge advantage when we try to see the immune cell trafficking, which could not be easily achieved by using the porous, the polymer membrane. So this is the conclusion. We could build up the in vivo the BM engineered basement membrane. And then this is the first study to reveal the engineered BM can facilitate the recreation of the barrier function of the BBB. And then using this model, we could build up this ischemic stroke. And then actually we are now utilizing this model to screen the BBB penetrating the immune cell to target the glioma cell, okay? Okay, this work was majorly conducted with Professor Kim Dong-sung at Postec and then Professor Kang Ju-hwan. Okay, thank you so much for listening.
Great, thank you. Have we got time for one question? One quick question, if anyone's got one. No? Okay, let's move on in the interest of time then. Thank you.
Okay, up next we have Georgios Pavlou from MIT. Yeah, take it away, Georgios.
So, how does it work? And go back. Okay. So hi everyone. So thank you for the introduction and for giving me the opportunity to present here today. So I'm going to present you my project, which is about the development of an in vitro 3D neurovascular model for Alzheimer's disease. And few words about the Alzheimer's disease. So it's one of the most common type of dementia in the elderly population. The main hallmark lesion is the amyloid beta plaques and the neurofibrillary tanks. You can see the image on the top left here, you can see this, the black arrow points out the amyloid beta that covers an arteriole. And then with the white arrows, you can see the amyloid beta plaques that they are formed inside the brain parenchyma. Also, the amyloid beta clearance is disrupted in the Alzheimer's disease, is known. And then one of the main mechanisms for cleaning the amyloid beta, the blood-brain barrier, is known that is disrupted. And you can see briefly on the bottom left image a schematic of the kind of clearing mechanism from the blood-brain barrier. So there are several mechanisms there that they are dysregulated. And talking about the blood-brain barrier, as you saw also before, so the blood-brain barrier is located in the brain. And more specifically, it's in the brain parenchyma. So you can see the arteriole invading the brain here. So here it veins the brain, and then we have the small capillaries where it's actually where the BBB is, and then it will exit as a vein. You can see also the schematic on the bottom right, where you can see the BBB that is composed from endothelial cells, pericytes, and astrocytes. And generally, a few information about the BBBs, that it's a highly selective barrier. It's known about its low permeability, and this is thanks to tight junctions that they are highly expressed. Some of them is like the Claudin-1, Claudin-5, the cadherin, ZO1, and some others. As I said before, it's important for amyloid beta clearance, and we know that is impaired in Alzheimer's disease, while it happens the same in a lot of neurodegenerative disease that we have always leaky BBB. So going now to our in vitro system, so you can see our microfluidic device on the left side. We have two media channels on the side, so here and here, and in the middle we have the region where we can culture the BBB and the neurons together, that I will explain you later. So in this device, we have a circular design in the middle here, and then we have a hole also where we will add the neurons, but I will go into details soon. So we inject first the tree culture of for the blood-brain barrier, so the endothelial cells, brain endothelial cells, brain astrocytes, pericytes, all of them primary, mixed in a fibrin gel. We will inject them from here. They will go all over the device. And then by a self-organization process, they will form the blood-brain barrier. And in the timeline of 7 days, they will start acquiring BBB properties. Then on the same day, the day that we add the BBB, we add also the neurons here in the center hole, either the wild type or the Alzheimer's disease ones. And they are embedded, they are suspended actually in a matrixel. And you can see now how it looks from the microscope. So we have the blood-brain barrier here. You can see here actually the endothelial cells are labeled, but the astrocytes, pericytes are there also. And then the neurons on top, so all together in one device. And then we perfuse after 7 days. We can perfuse actually fluorescent tracers like the dextran that you see on the bottom right. And you can see that we have 100% perfusable networks. And also something else that I forgot initially to say is that for the neurons, we need around 20 to 30 days to differentiate them and start producing amyloid beta in a way it is something like producing the Alzheimer's disease state. So these specific neurons, they will produce amyloid beta levels that they will reach kind of the late stage of the Alzheimer's disease. So for this, the differentiation process take 20 to 30 days. So for this, there is a need for culturing the devices, like keeping the BBB with the neurons for 30 days at least. And that's why we have developed a pumping system in the lab, so that you can see here at the bottom left. So we connect this to the device that I showed you before. And this pump that is PDMS based is connected to an electronic part here. So that is connected initially to the air supply from the lab. So this air supply goes to a pressure regulator that is connected to a solenoid. And then we have an electronic part that controls the frequency that we are opening and closing the solenoid here. So with this, we can pump pump, like air pump, this pump here, like the heart, then we can circulate the media all over the device here. So they will go like this. They will go through the networks here and the co-culture actually of the networks and the neurons, and then they will exit, and then we have the circulation over time. You can see also how it looks in reality. So the pump here is plugged in on the device, also the side view. Then this is the air supply here that I told you before. So after this, before this, there is the electronic part. And then you can see also how it looks when we perfuse beads using the pump. Will it play? Yeah. So you can see these white small things going through the networks. I don't know if it's visible there. Okay. And you can see also the neurons on the top. So we can keep this for a long time. And here you can see also how it looks in our incubator. So we have managed to parallel run 24 pumps and devices at the same time. We change media every 2 or 3 days, but this can go more. It depends also on the bubbles that are created inside the pumps, which are always not good to have bubbles. And then we have managed also to keep the devices in culture for 35 days. Here, having all this is my experimental timeline. So I start 8 days before by expanding the cells. So again, human primary astrocytes, pericytes, brain endothelial cells, and then the 2 cell lines for the Alzheimer's disease, the wild type 1 and the Alzheimer's ones. Again, is the late stage of Alzheimer's disease. So we have very high levels of amyloid beta. Then on day 0 is the seeding day, where we inject both the BBB and the neurons. Then after 7 days, we have perfusable networks, and then we connect the pump. And then we keep the devices for at least 30 days. In between, we can, of course, we change media, and we can do permeability assays, morphology assays. And then at the end, on day 30 plus, we can extract actually the cells from the devices and do PCR. We can do immunostaining for Alzheimer's disease, tight junctions, amyloid beta, and the blood-brain barrier. And here is how it looks. This is an image after 30 days. So we have the blood-brain barrier here and the neurons together next to each other. Then we try to perfuse them with several fluorescent tracers. So here you can see Dexter and 40 kilodalton. So the image on the left here is the control case, the wild type. The Alzheimer's disease here. So just a note here that you can see the neurons and the fluorescent tracer on the same color because it happens that the neurons have the same color here. So they're expressing also the blue, while in the control case, the wild type, they are only GFP. So not to confuse you. So this is not Dextran here. So we have managed then to have wild type and Alzheimer's disease devices that are perfusable. So you can see some other perfusability examples here with these nicely made networks, very thin, reaching BBB morphologies. And then having done perfusion, so we were able also to do permeability. And I'm gonna explain how we do it in the next slide. So here is just to show you this interesting result that first we confirmed that having the neurons in our system, this decreases the permeability, as you can see. So here in the first plot chart here, we have only BBB devices. And when we added the neurons, you can see that the permeability values, they went lower, which can be explained like what happens in vivo, that we know actually that in the presence of neurons, either in healthy state or disease states, they're affecting a lot of things on the blood-brain barrier, affecting for sure permeability. And we can see that we have a decreased one in this case, which fits well for our in vitro system in a way to recapitulate our the in vivo conditions. So then we did a permeability assays, like I told you, for now on day 10 and day 30 in the devices that having the wild type and the Alzheimer's disease ones. And few words about the permeability assays, how we do them. So we take an image on time 0, like you see here, where it's the time actually that we perfuse a fluorescent tracer, like here, Dextran 40 kilodalton. And then after 12 minutes, we're gonna take the same Z-stack, and then we're gonna fit the intensities that we get there in this plot here, and we get permeability values. So using these, You can see on the plot for day 10 that we don't see any difference in the permeability values for wild type and Alzheimer's disease, while after 30 days, and don't forget that is the differentiation process for the neurons, so the neurons start to express more the disease, more amyloid beta. Then after 30 days, we can see significant changes in the plot, and you can see here that the BBB becomes leakier and in comparison with the wild type. So having this, we wanted to test the amyloid beta levels in our devices. So thanks to the design that I showed you initially, here we have a hole in the top, in the middle, that we can also sample for the neurons. So we can take media from this hole here and then do ELISA, for example, and check the amyloid beta levels. And you can see here in this experiment, we collected like on day 7, 14, 21st, 24th, 33. And then you can see here in this plots always the control on the left, the Alzheimer ones on the right. So you can see on this timeline that we have the increase, like very highly expressed amyloid beta levels. Here we check for 3 isoforms. So we have amyloid beta for 38, 40, and 42. In all the cases, you can see the levels that are very high. Something interesting that came out from this is that if you notice for the amyloid beta 42, the levels are decreasing. So you can see it's going lower. And this made us think that knowing actually that the amyloid beta plaques are formed mostly from the amyloid beta 42 isoform, that this could be explained possibly that the amyloid beta 42 is not soluble anymore, but then it has become like it's a plaque, like you see in the image here that I showed you initially. So then that's why it could not be detected in the ELISA. So for this, we stand for amyloid beta plaques. And first, we confirm that we have amyloid beta 42 and plaques on the neuron side. So you can see with the magenta these spots here. And then, of course, we wonder what happens in our vascular networks. And you can see first in the wild type, there is no magenta color, while in the Alzheimer's disease ones, you can see also in the zoom version here. So we can see on the externally of the lumens, so that we have magenta color, which means amyloid beta 42, which means plaques. Also with the other arrows here, I'm pointing some plaques that are forming in the interstitium here. And one more example, again, the wild type, nothing, while the Alzheimer's ones, you see, I don't know if it's obvious on the screen, but you can see here some magenta covering the vascular networks, also here. And again, there are a few spots in the interstitium. So with this, I would like to summarize my talk. So we have developed an in vitro long-term culture 3D neurovascular model for Alzheimer's disease. And we have managed to recapitulate partially the in vivo disease state, and this by observing the high levels of soluble amyloid beta in our system, by also visualizing, to my knowledge, one of the first times the in vitro the amyloid beta plaques, and then to show that the BBB is leakier in our devices when we have the Alzheimer's disease state there. So on my last slide, I would like to thank my mentor at MIT, Professor Roger Kam, and the rest of the people involved in this work, Claire Combe, Sun Zhang, Marie Florian, and Savannah Ashley. And of course, the collaborators for the neurons at the Mass General Hospital and the Harvard Medical School, Sehun Soa, Mehdi Zorfi, and my fellowship, the Cure Alzheimer Fund, and the NIH for supporting my project. And thank you for your attention.
Thank you, Georges. We have some time for questions. So if you have questions, please walk up to one of the mics and yeah.
Thank you very much for your talk. I have two small questions. One, and maybe I missed it, I'm sorry if I did. What do you call Alzheimer neurons? I mean, what is the modification on these cells? And secondly, did you see the A beta plaques extracellularly? I was trying to see in the slides, but I wasn't quite sure. It looked to be intracellular partly as well.
Yeah, no, that's a good point. So we had exactly the same questions initially. I will start with the second question. So the amyloid beta plaques, they are externally, so we have checked it and we have more images that I didn't manage to put on this talk. But we confirm, initially we thought that maybe it can be inside the vascular networks, but then with a high resolution, we confirm that they are covering the BBB externally. So it's external. And then for your first question, we call it Alzheimer's disease. So they have modification in a way they're in the APP and the SEN1 that they can produce more amyloid beta than the wild type. So yeah, the mutations are there to produce more amyloid beta.
What about phospho-tau? What about tau?
What about?
Tau, phospho-tau. Because Alzheimer, you know, you have a beta.
Yeah, for these cells, we don't focus on the tau, but they are also, they have developed in this lab for the phosphorylated tau. Yeah. So, but I'm not focusing on this. That's why. Yeah.
Okay. So A beta cells then rather.
Sorry?
They are A beta cells rather than Alzheimer's, you know, but
Yeah.
it's really very nice data. Thank you.
Thank you.
Thank you. I have a couple of questions, small questions. The first one is, are you using a gengenic factor to generate the vascularization of your gels? In previous paper from your lab, you typically use lung fibroblast or other cells that release angiogenic factor to generate the microvascular. So I'm curious if you are still using this one in your chips or not.
To make them perfusable, you mean? Or to make the assembly of the BBB?
Yes.
So instead of the fibroblasts, we have the pericytes.
And they can lead it. Okay. And the second question is, so if I'm understanding correctly, you're using neurons, and you put neurons in madrigel, and then all surrounded you have a fibrin. And all the gels are stable for over a month. Are you using metalloproteases to keep that stable, or? No, so it depends. Also, it has to do with the design. We have noticed that sometimes if, for example, you have different designs, then you can have gel being detached. But this is more because we connect them also in the pump, so it's not from the cell. So in our cases, we don't have digestion from the gels, so we don't need to inhibit, let's say, some functions of the cell. So we have them like this, no addition, just media, and they don't digest the gel. So also the madrigel remain there?
Oh yeah, the madrigel remains the same. Yeah, even under the flow conditions is okay. Yeah, but it's also stiffer, much stiffer than the fibrin gel. So.
Yeah, use full madrigel 100%, right? You don't dilute
it's diluted half, but still from the fibrin is different. So the whole flow is going around the device, so it's not going through the neurons. So yeah, even with the flow, it's not affected.
Thanks.
Thank you.
Okay, thank you, Georgios, once again. I think we're going to move on to our next speaker. If you want to present them.
Yeah.
Sorry.
Okay, so move on to the last speaker. Is Benjamin Friedman from the University of Washington.
Hello, thank you for the introduction and for the invitation. Topic today is fluidic absorption drives cystogenesis in a human organoid on chip model of polycystic kidney disease. This is a new paper that will hopefully come out from my laboratory, and it's really the first time I'm presenting it, and I'm excited to show you guys how it works. These are my disclosures. I'll start with the big picture here, and this is a different type of barrier from what we've been discussing. This is the kidney, and the kidney is a highly fluidic organ. It maybe is the most advanced microfluidic device on the planet. And these structures, these nephrons that create this filtration and then huge reabsorption of 180 liters per day, these structures arise only during embryonic development. So if you want to study the nephron and study the filtration processes that are involved, you really have to go back to the start before any of the organs have formed. And this is what we've been doing in the laboratory and many laboratories around the world, using human pluripotent stem cells to derive organoids, and then using the kidney organoids to study different disease states in the laboratory, and ultimately potentially study regeneration of the kidneys. So this is the protocol that I devised when I was a postdoc. Yes, it's good to do postdocs. This is how innovation happens. So it took me many years, but eventually I stumbled across a mechanism to generate human kidney organoids for the first time from pluripotent stem cells. And these are the types of structures that we get out of these differentiation cultures. There are complex podocytes, which are the filtering cells of the kidneys, attached to proximal tubular cells and distal tubular cells beneath those in a beautifully patterned distal to proximal axis. And these types of cells, we haven't been able to grow before, we haven't been able to culture before in primary cultures, and certainly we've never achieved this type of organization before. So it raises a lot of possibilities. But for me, what I wanted to do here was really to study the potential of these cells to model disease. And not just any disease, but a genetic disease state called polycystic kidney disease, which has been a major challenge to model at the cellular level for decades. And the fundamental feature of this disease is that these tiny tubular structures within the kidneys expand, they balloon out, and they form these cyst structures that eventually crowd out the healthy tissue in the kidneys and cause kidney failure. Now, we've known the genes that cause this disorder for 25 years going on now, PKD1 and PKD2, but fundamentally, we don't know exactly what they do. They seem to encode a channel receptor complex that localizes to this antenna-like structure called the primary cilium at the apical surface of the cells. But why this results in cystogenesis has long been a mystery. And one of the great challenges, as I alluded to, is that cell lines from patients don't form cysts in a PKD-specific way. They may balloon out and form some spheroid structures, but those types of structures are also found in wild-type cells that don't have any disease state. So fundamentally, we don't have a reconstitution of this disease state in cell culture. Well, until recently, it turns out that our kidney organoids can beautifully reconstitute this fundamental genotype-phenotype correlation that's observed in polycystic kidney disease. And these are some of the images shown here. On the left, you can see the non-PKD organoids, and on the right, the isogenic gene-edited PKD1 or PKD2 knockout organoids. And these knockout organoids specifically form these large cyst structures that are absorbing fluid and expand over time in the dish. So this is really PKD in a dish. Now, most recently, we've studied the cilium, which is this antenna structure that I alluded to, where these PKD proteins are localized. And we've essentially knocked out this tiny organelle, the cilium, in human pluripotent stem cells. This is the type of experiment you really can't even do in a patient because it would kill the patient. But with human pluripotent stem cells, you can get away with a lot. And in this case, the cells are just fine without cilia. They can differentiate into organoids, albeit at a lower rate. But what's really remarkable is that once these structures form organoids, they go on to form cysts that are exactly like the cysts we see when we knock out the PKD disease genes. And you can see how large one of these organoids can grow in this movie here. This is a flask, and it's got an organoid that's gained so much fluid that is about the size of a ping pong ball. So we recently published that, and I won't go into it any further, just to say that we can see that there is a mechanism here specific to the disease state that's reproducible in different models. So the question that we wanted to get at next was, what was the effect of flow in this system? All these organoids, they've been generated under static conditions. We don't know how perfusion would affect cystogenesis. So to get at this, we actually arrived at a very simple system where we take our PKD organoids or our control organoids, and we place them in a very simple microfluidic device, and we flow media over the tops of these structures for a series of hours. And when we do this, what we observe is that the PKD organoids actually swell over time. You can see this swelling effect here. The cysts expand as they encounter the flow. And we don't see this type of swelling in the control organoid. So it seems to be something specific to the PKD. So we wanted to see whether this was really flow or some other component of the system. That's the issue here. To get at that, if we can go back a slide, to get at that, we had to generate a diffusive static condition. Initially, we just tried to plug in essentially a pipe, a syringe needle into our device and call that the static condition. Well, it turns out that that doesn't work because no diffusion is actually coming through the syringe needle into these channels. So that's not really a static condition, guys. But what we were able to engineer was a true diffusive static condition where we used a wide bore syringe and connected that to our device. And in this case, we're able to see the food dye diffuse into the channel. So now we know we're getting diffusion, and we can use a large reservoir, attach it to our channel, and do the same experiment with similar volumes, but without the flow. And when we do that, I keep pressing ahead. When we do that, what we observe is that volume alone partially substitutes for flow in the cyst expansion phenotype. And you can see these data quantified here, where at the left, the pure static condition where there's no diffusion, well, those cysts don't grow at all. They're not very happy. But if you use a diffusive static condition of 1 ml, it doesn't make a difference. But if you use 25 ml, you suddenly see an increase in the size of the cysts, which approaches the increase that's observed in the fluidic condition. It doesn't completely reach it, but it gets close. And moreover, you can eliminate the channel device entirely and simply place these organoids, as you see on the right, in dishes of different size and see the same effect, where over time, the organoids in the larger volume expand without any flow. So volume is important here. So what is it in the volume that is affecting the cysts? Glucose is an obvious candidate. We know that the kidneys, one of their primary functions is to absorb glucose. And we can beautifully see glucose absorption in these chips over time. This is one of the best things about these microfluidic devices. They're so thin that you can take live movies and watch the glucose enter the organoids and enter the cysts. And when we do this, we can see glucose being absorbed into the cyst lining epithelium and subsequently filling up the cyst. And this occurs in both the fluidic as well as the diffusive static condition. And there is a lag time in the diffusive static, but it reaches about the same levels of glucose absorption over time. So we know that the cysts are actively absorbing glucose, as is the rest of the organoid, the cells within the organoid. We also know that there are transporters of glucose that are specifically expressed at the apical surface of the tubular structures in the organoids as well as the cysts. So this then led us to the question of whether we could simply inhibit glucose transport and block cyst formation. And indeed, what we observed is that when we added in glucose to higher levels, we saw increased cyst growth. And when we removed or when we blocked glucose absorption using Floritin, which is essentially a broad spectrum inhibitor of glucose transport, we could dramatically reduce cyst growth. So this was about a 70% reduction in the expansion rate of these cysts. It actually went below the baseline. And we know that these levels of Floritin are not damaging the organoids because we perform live dead analysis as well as other types of metrics to look at the toxicity of these drugs before we do these experiments. And these are non-toxic doses. So simply inhibiting glucose transport blocks cyst formation. Well, this is all well and good, but it's a little bit confusing because, in the body, the absorptive part of the cyst is facing inwards, and glucose transport would be leaving the cyst rather than filling it up. So we asked the question, what is the orientation? What is the polarity of our organoids? Is it like the cysts in the body, or is it different? And when we looked at the organoids, looking at acetylated alpha tubulin as a marker of cilia, as well as ZO1 as markers of tight junctions, well, we observed that there are tubular structures in the organoids that are properly polarized, as we would expect, with the apical surface lining the inside of the tubule. But the cysts, without exception, all face the other way, where the apical surface of these cysts, as you see in the upper left here, is facing outwards towards the media. So this is an inverted cyst. It is the opposite of the orientation of a cyst in vivo. And yet, it's capable of reproducing the phenotype that you see in vivo down to a T. So how are these forming? Well, we did closer analyses of these cysts, and you can see in the upper right here, it's really, the cyst is forming from a peripheral epithelial layer that almost surrounds the organoid. And those tubules that we're used to seeing are actually involutions of the membrane, of this peripheral membrane in these cyst structures. So that explains to us somewhat how you can get these tubular structures, but also have these peripheral cysts. And we took a step further and we took movies of these organoids as they underwent cystogenesis. And you can see this, one of these in time-lapse images at the bottom here. And if you look at that little yellow arrowhead, that's pointing out one of these involutions of the peripheral membrane. And then what you'll observe is that the peripheral membrane expands, the part of the organoid epithelium that's facing outward. It expands out very rapidly and forms the cyst, while that internal piece, which is the part of the cyst, part of the organoid that's polarized inwards, does not expand and remains inside this expanding cyst. So it's really consistent with the idea of an absorptive process coming through the apical side of the organoid epithelium that is driving cystogenesis in this system. Okay, well, now we had to think about how does this dovetail with what's happening in vivo, where the cysts have the opposite polarity. So we went back to a mouse model of polycystic kidney disease and took a close look at how the cysts were forming. And what we observed is that there were little pockets within these early cystic kidneys that were not lined by epithelium at all. Instead, they were pockets of fluid accumulation in the interstitium. And we could see this also by immunofluorescence at later stages. When you look at the cysts, there were areas where the cystic epithelium, which is, normally the kidney epithelium is very tightly associated with the interstitium and the endothelium. In this case, it seemed to be peeling away as if it was absorbing fluid and that fluid was pooling in the interstitium. Moreover, when we injected glucose into animals with PKD, we could see that this glucose was rapidly absorbed through the cyst lining epithelium and into the interstitium of the organ. And this gives us the idea that really what might be happening is that the cysts are absorbing fluid, and the problem is that the fluid is accumulating within the kidney, and this creates space within the kidney for the tubules to deform and fill out the cyst. So my major conclusion here is that, as the old saying goes, all models are wrong, but some are useful. In our case, our model has an inverted epithelium, but that inverted epithelium, which normally isn't there in nature, is actually driving cystogenesis through a reabsorptive process. And this teaches us that absorption could be playing a critical role in PKD, and this has not been examined for many years. In fact, the focus has been on the opposite. The focus has been on secretion as a driver of PKD. But maybe we've got it wrong. Maybe the organoids are teaching us how the disease actually works, even though they're not perfect. So to sum up, organoids reconstitute PKD cyst formation in a disease-specific way. Organoids on chips really drove us to this revelation that cysts form via fluidic absorption. And inhibitors of glucose transport are novel targets for PKD therapy. So I'd like to thank the folks in the lab. This has really been a multi-year project, and particularly Sienna Lee and Nellie Cruz and Louisa Helms have been drivers of this work. I'd also like to thank NCATS particularly, as well as NIDDK and the Laura Nowak Macklin Fund for supporting this innovative area of research in our laboratory. And I'll thank you for your attention.
That was brilliant, thank you. It's good to see a real use of kidney organoids. Just a quick question. Did you try staining them for the podocyte markers? Do you know how they organize?
Oh yeah, I mean, the podocytes are beautiful in the organoids. And in the cyst structures, the podocytes actually hang out on the exterior. So they're connected to the cyst epithelium. And they also polarize outwards towards the media, just like the cystic epithelium. But they do not really contribute to the cyst. It's the tubular epithelial cells and the distal tubular epithelial cells and the parietal epithelial cells. Those are what's giving rise to the actual cyst, which makes sense because those are the more absorptive cell types.
Yeah, great, thank you. Have we got any more questions? No, one.
Hi, thanks so much for the talk. I was wondering, you've presented on glucose data. Have you looked at any other small molecule metabolites or things that could be in interstitial fluid like electrolytes? And kind of follow-up question to that, have you guys investigated doing any kind of computational transport models and applying that to the data you've collected?
Yeah, both great thoughts. We haven't yet looked at any other small molecules, but yeah, it could be that there's something else driving the process, right? I mean, ultimately, it's got to be water that's flowing into these things and filling them up. We know that the cysts can exclude certain things from the media as well. So they're pumping in the opposite direction, and that may be co-transporter activity of some kind, which is driving the process. So now that we believe it's really import that's driving the cyst, I think it raises a lot of possibilities for what we could look at. So we haven't looked at those yet, but I hope to, and I would enjoy discussing over a beer maybe how to do that. The second question in terms of computational modeling is also a very good one. We haven't gone down that road yet, but it would be interesting. I mean, one possibility is that the fundamental mechanism here is the pliability of the epithelium itself. In other words, the transport levels may be the same, but the membrane itself of the epithelium is weaker and allows for distension. So that would be a cool thing to try to model computationally.
Thank you.
Thank you.
Nice talk. Thank you very much. When we grow organoid, we grow in matrigel. So in this kidney organoids, do you grow them in the matrigel? And also, I'm wondering about the effect of glucose, whether it increases osmolality and therefore it draw the fluid into the cyst. And I'm growing organoid, typically tumor organoid, and sometimes we see this cyst formation. And I'm wondering, is it kind of organoid phenomenon, or is it because of the PKD mutations?
All right, so first question, in terms of how we culture the organoids, they're actually cultured on a really thin layer, a thin matrigel sandwich. So it's just about 1% matrigel on either side initially. And that's in an adherent culture. And then we pop them off those plates into suspension cultures before we put them back inside the channels. And the channels are also coated with a thin layer of matrigel. So we use a thin layer of matrigel, but not a lot of matrigel. They're not baked in it. The second question, in terms of whether glucose, the osmolarity is actually driving the fluid uptake, yes, I think that's how it seems to be working. At least that must be the effect of these glucose inhibitors, is that they're blocking glucose uptake. And by virtue of that, they're blocking the fluid flow, which would be flowing in to balance the glucose on the other side of the epithelium. And the third question, you see some cysts in other organoids. I think cysts are a bit of a default configuration for a lot of epithelial cells. So, the fact that a cyst is there doesn't mean it's PKD. But if you wanted to test and see whether it's PKD, then the right experiment is to take isogenic lines, control versus PKD knockout, and see whether you see an enhanced cyst formation under those circumstances. I'll say that human pluripotent stem cells also express the same polycystin genes. They also have cilia. They also form cysts. But there's absolutely no difference in cystogenesis in the undifferentiated human pluripotent stem cell, whether or not you have the PKD gene there or not. So just because you have the mutations doesn't mean you're going to get a PKD cyst. And just because you have a cyst doesn't mean it's PKD. It really depends on being able to see the cysts specifically in the PKD genotype in the tissue of interest. But it does affect a lot of other tissues besides just kidneys.
Okay, great. Thank you. I think we'll leave it there. So I'd just like to thank all our speakers for this session, and thank you for listening.
So I know it is wonderful to chat with people, finally in person, not via Zoom, but I would hope that you're coming to celebrate with us the official beginning of a show which started this morning. So please take a seat. We try to do it nice and sweet, and I promise you some highlights for today. I'm Thomas Hartung. I'm the director of CAT, the Center for Alternatives to Animal Testing, which took a lead role in organizing this, but I'm standing here for all of the organizations and the many individuals which have been contributing. I will come to this in a second. So everybody found a place. It's something big which is happening here from my point of view, and perhaps we can start my presentation. It is something big because I think it is the first time. Please get it started. Can you start it again from the beginning? Okay. So I think we are able to actually, we are able for the first time to bring this community together, and we will rock you. This is my strong belief. And we will not only rock you, I think we will rock the biomedical field, because we are going to actually further a revolution which is taking place in finally bringing relevant human in vitro models to biomedical research. We have been meeting very often over the last 10, 15 years in various symposia, but there was never really a worldwide conference. And this shall be the series which is starting it all. With this World Summit, you saw our symbol, we are actually going to move forward. And when I say we will rock you, this is obviously borrowed from Queen, and I will use them a little bit for my not so serious introduction of what you're going to expect here. Queen, it's a kind of magic, one of their very famous songs, and I think our technologies, they are a kind of magic. They're enabling things which were not possible before, and as you know, any technology, advanced technology is in the end indistinguishably of magic. We are the champions. 55 organizations which are all vested into the field of microphysiological systems have come together. 52 organizations which are backing this event and have been helping us to make this, I think, incredible program happening at the end of a pandemic which was impairing our collaborations, our work together, but also impaired, obviously, on the number of people we could welcome here, with still some travel restrictions all over. And this is, I think, quite remarkable that we have around 500 people here in person, and by our increasing numbers of people watching us through the net, including, for example, 65 FDA representatives which have block booked our conference. So you are heard. You are heard by even more people, and we will make our recordings available. 52 organizations of champions. Fun fact, 52 years ago, it was that Queen was created. And this is Brian May, who proudly announces this t-shirt through this t-shirt. So 52 seems to be a magic number here. Queen, One Vision, yeah, that's one of their songs. And this is this one vision which is uniting us. The three hosts, I was thrilled that very quickly Susie Fitzpatrick and Don Ingber were joining me, two of the early and most important opinion leaders in the field of microphysiological systems, and you will have the pleasure of hearing Don give the opening keynote lecture in a second. Both of them gave keynote lectures in our virtual pre-meetings, and Susie has been instrumental to give MPS the role it currently has in her work in the FDA, which is so much impacting on the acceptance and uptake of these methodologies. But we also have a scientific advisory committee of individuals who were supporting us in helping this and who are sharing this vision of one technological approach making a difference. And then there is the sponsors. We want it all, and we were under the pressure of making it happen. and our platinum, gold, and silver sponsors and all the supporters we obtained were helping us by their financial support to make this all happen. Because friends will be friends, and that's what we relied on. We used our networks, we mobilized. I think for a first time, this is a really remarkable number of supporters which we found here. Yeah, this is the queen of MPS World Summit, Lena Smirnova. She's the killer queen, but she's also the love of my life, as many of you will know, as my wife. She really did stem a lot of the work. She was the chair of the program committee, the co-chair of the scientific advisory board, the chair of the International MPS Society Committee. So tons of roles. And I think without her direction, I could not have slept as well over the last 2 years. And you might wonder why I took this picture, which is pretty old, not her current haircut. The reason is simply it shows her with Brian May of Queen. So this is something I could not leave out in this presentation. So the Lush prizes were actually the occasion on which she encountered him. But no queen without a band. There's a band, and I'm showing here some of the people who were critical. Shepan Baran as the co-chair of the Scientific Advisory Board, and then our own Camilla Januario and Tracy James and Anvin Stratnik, who helped us to make this all happen. So this is the band behind it. Our World Summit, these are the numbers from last Friday, 137 speakers, 56 abstracts, 81 invited, plus the roundtables and debates. I think we are approaching something like 178 from what I understood. We have an enormous number of posters, around 700 registrations, increasingly online. So this is, I think, a quite remarkable thing. In the actual statistics you will hear at the very end of this meeting. And we have the social events, Party, one of the lesser known songs from Queen from their album Miracle. We started yesterday with a small intimate one for those who did not know what to do, arriving sometimes from other continents. Today we will celebrate at the welcome reception outside. We have a city tour, we have the macro party. Please secure your tickets. We will still make some available. At some point we have to close because we have to plan how many people are actually coming. So please do so fast. And we're also thrilled that we have the Jones Walker Sunset reception, which is free of charge, which was sponsored by the company as the finish of our conference essentially on Thursday evening. Yeah, I want to break free. This is what our technologies are doing at the moment. It is something which is really developing a life on its own. It is something which is leading to recognition. Our workshop on microphysiological systems from 2019, for example, led to this science paper just in September, which shows you the interest in these technologies which we all share. There's Breakthroughs, another song of Queen, and I put Jay Hickman, who was the chair of our funding committee, up here because they just gave our community a present by enabling the first IND, so the first permit to move into clinical trials just based on microphysiological systems, as their press release from April showed. I think this is an important step showing that these are enabling technologies. These are technologies which will move drug development and others. And this is one of the milestones which was achieved. But the most important milestone for me was that he and his fundraising committee really made this all possible with really strong work, sleepless work, I would almost say. In the end, another one bites the dust, and I hope these will be the animal models representing a Center for Alternatives to Animal Testing. I think we are complementing and hopefully increasingly substituting for animal testing, because if one wins, another one has to lose. And I think in this case, everybody wins by moving to new technologies. So let's jointly create a milestone in the revolution of the life sciences, a miracle which we want to achieve. The show will go on, and don't stop me now. Don't stop us now. I think it is a tremendous opportunity in time to see a scientific revolution taking place. And with this, enjoy being part of the rise of microphysiological systems. I hope you have terrific time with us and we see a lot of fun, a lot of collaboration, and a lot of insight and inspiring presentations, as you will hear later in this presentation. Thank you very much. And with this, it is my pleasure to ask a brother in crime in this case. Danilo Dan Tagle was one of the architects of the microphysiological systems revolution. It was over a beer that the two of us discussed at our Berlin workshop in 2019, the need for an international society, the need for conferences, and Dan did not stop there. He made it happen. The call for proposals he initiated from the National Center for Advancing Translational Sciences of the NIH was, I think, really the starting point for this event series. And for this reason, I think he should give his version of the whole thing. Dan, thanks for making this all happen, and thanks for telling us a little bit more about how this all came about.
Yeah, so good afternoon, and certainly a pleasure to be here. And I'm not going to be as creative as Thomas in linking rock and MPS or Queen and tissue chips. So I was tasked by the organizers to essentially give you some history in terms of how our journey began. So with that, can we go into the next slide, please? Oh, you have the control. All right.
It's not easy for me to give up control.
Well, we're finding that out. Okay, so I come from NIH, specifically from the National Center for Advancing Translational Sciences. For those of you who are not familiar with NCATS, it was established in 2012. So we just celebrated our 10-year anniversary back in December. It was established by the United States Congress primarily to address the longstanding issues in drug development, especially what's so-called the Valley of Death. So our mission is very different from the rest of the NIH in the sense that we are focused primarily on developing innovative technologies that will enhance the development and testing and implementation of diagnostics and therapeutics across many diseases and human conditions. So we're in some ways disease agnostic. So most NIH institutes are going to be focused on cancer, neurological diseases, kidney, but we cut across all those types of conditions. And really what brings us together are the issues that are pervasive in terms of drug development. What are some of those roadblocks? So what I'll be talking more is how we apply our 3D principles. So 3D, not three-dimensional, but it essentially stands for develop the technology. So any technology that we work on, and there are many, not just tissue chips or organs on chips, and then we demonstrate the utility of that technology. That means building confidence, making sure we work with appropriate stakeholders so that it takes hold into the field and really cause a paradigm shift. And then ultimately, we want to disseminate that technology, make sure that it gets into widespread use and thus really have an impact in public health. So just an overview of what's been going on over the past decade with tissue chips, or we have what's called the NIH Tissue Chips Consortium. So this is essentially a history of the funding that has gone on from the NIH over the course of the decade or so that NCATS has been in existence. Actually, the program itself predated NCATS. Had the pleasure working with Susie Fitzpatrick and colleagues at the FDA as part of the NIH FDA Joint Leadership Council for Advancing Regulatory Science. So that started in 2010. And so back then it was Margaret Hamburg and Francis Collins had the idea that they should bring together the head of NIH as well as FDA to start thinking about how we could advance regulatory science. And so one of the projects that we funded was actually Don Ingber, who happens to be giving the keynote. So Don, along with Kit Parker, proposed a heart-lung micro machine, and that was, in some ways, the early vestiges of what the promise of this technology would look like. And then NCATS got formed, worked with DARPA, with the FDA, as well as with pharmaceutical companies like AstraZeneca, GSK, and Pfizer, who provided us with reference set of compounds, essentially compounds that have gone through safety assessment using preclinical models, but have subsequently failed in human studies. And so the question is, how can we develop new technologies, in vitro tools that would have a better predictive rate than the current tools using preclinical models. And so that's where we focus primarily during the first 5 years of the program was in 2012 to 2017 was focused on safety and pharmacology and essentially work with a number of different partners within the NIH, other institutes and centers, and shown here. So like the dollar figures that we have been providing. So in some ways, DARPA provided $75 million to jumpstart this program on their side. And likewise, the NIH also provided an additional $75 million to help start the process. Over the course of the next 5 years, then we switch over to the biggest cause of attrition in drug development. So safety and toxicity accounts for like 25% of the attrition in drug development, and about 60% is due to failure of efficacy. And so we then ask, can we use tissue chips and pivot that technology to address this big issue of failure of efficacy? And so we have had the opportunity to fund a number of different projects, including some creative collaborations with NASA and the Center for Advancement of Science and Space. One of the first things we ask is, can we model aging on these tissue chips? Aging, as you know, takes years, perhaps decades to model using the current systems that we have. But in working with NASA, we learned that astronauts actually go through some physiological changes where they exhibit accelerated aging within months or weeks of being exposed to microgravity. And then that also brought about a new opportunity to work with payload developers as well as SpaceX in hearing what the requirements are for space flight. And they said, you got to miniaturize everything. You got to, flying payload into space costs millions of dollars. And so everything will have to be compacted. So we essentially shrunk down everything from the size of the incubators, about the size of a refrigerator, kitchen refrigerator, to something about the size of a shoebox. So those are technological innovations that we hope to translate and make common use for tissue chips. So those will be hopefully forthcoming within the next couple of years, where you have instrumentations that are portable and smaller footprint, and certainly turnkey. Turnkey, because when we talk to the astronauts, what would make for a successful biomedical experiment in space? They said, well, astronauts are about, we're 90%, we're good at flying fighter jets. We're not scientists. And so make it astronaut proof or fighter pilot proof. And so one of the things that we work with the space engineers and payload developers is to make everything automated and turnkey technology. And if you've seen some of the talks from Devin Meyer and a few others that have worked and been funded with this, we have launched so far about 6 or 8 flights at SpaceX to space, showing that there has been miniaturization and automation of the technology. We also wanted to capitalize on what Congress has provided us, especially with the opioid crisis. So these are public health emergencies, as so shown here. Tissue chips, who would have thought that tissues can be used to model nociception, addiction, and overdose? I think James Sickman talked about his project, Hesperos, in terms of looking at overdose with the BOTC neurons. We've worked with, other longstanding diseases, such as Alzheimer's and related dementia. We have a big project looking at certainly Mendelian and complex disease models in terms of being able to show that we can effectively capture the pathophysiology of this either rare Mendelian disorders with this or complex diseases with this model systems. And then the last area that we wanted to make an impact on is in the emerging area of precision medicine. So certainly the approach of one size fits all in some ways doesn't hold in this era of precision medicine or personalized individualized care. So this gives you really an opportunity because with patient-derived materials, either primaries or stem cell derived materials to populate and seed these platforms so that individuals can have their own, in some ways, as Francis Collins, our NIH director, said, your own you on a chip system. I think Chris Hughes gave an excellent example of that in his talk earlier this afternoon, where primary tumors can be put in these chips, be screened for potential chemotherapeutics or cancer therapeutics, and then used to inform the physician in terms of what would be the best care to give or intervention for that patient. And then, of course, we wanted to build confidence into this technology. And so we and NCAT stood up testing centers, independent testing centers. So these are sites that have not worked with tissue chips before. And so it affords us the ability to assess independently how robust the systems are, reproducible, how portable the systems could be. And then at the same time, we also are wanting to invest more in terms of making the data available. So we established the microphysiological systems database housed at University of Pittsburgh with Lance Taylor as one of the PIs there. And then make them into a self-sustaining model. This is obviously enabled by partners that we've had from the very beginning. So as I've mentioned, the FDA was right at the beginning of this program. And then sometime in 2013, 2014, we started working with the IQ Consortium as a group to help inform how the potential end users, the regulatory agencies and industry can adopt this technology. As I mentioned, partnerships are vital. As I talked about the FDA and IQ Consortium, but this is also a snapshot of what's been going on within the NIH. As you know, there are 27 institutes and centers at the NIH, and certainly a good majority of them have actually started looking and investing their own dollars, not just NCAT's money, but their own dollars into advancing tissue chips from, if we can go back. Can you go back a little bit? Yeah. Oh, red button. Okay. And so a number of from cancer biomimetics to modeling the nervous system in MPS and other things by other institutes and centers at the NIH. I mentioned the partnership that we had with NASA with the tissue chips in space, and of course, the most recent one with the growing interest in long-term space flight with mission to Mars. They wanted to push the technology. So NASA actually funded, along with BARDA, the ability to extend the longevity, the culture life of tissue chips up to 6 months. So not just talking about 28 days. We have partnerships with BARDA on the Immune Plus Chips program, where Don is also one of the awardees for that contract. Working with the EPA, which has a strategic vision of animal-free testing by 2035, working with the National Academies and the Institute for Laboratory Animal Research on a workshop on microphysiological systems for One Health. And certainly interacting and networking with other countries such as Europe, Asia, and Australia in terms of what's going on and making sure that we inform each other of activities and in some ways cross-fertilized. And so that leads us then into the MPS World Summit. As Thomas said, it started with, I think it was just more than a glass of beer. But essentially, how can we bring the community, the international community together? And so we're talking about the tissue chip developers, the suppliers, certainly pharmaceutical industry, the regulatory agencies, and looking at some of the common issues that pervade the field. There's, platform fabrication, integration, cell sourcing, cell scaffolds, qualification, a specific context of use, standards development, and really having the goal in mind of providing a forum for exchange of the latest research developments, technology developments, and also data sharing to be able to sustain and increase the use and adoption of microphysiological systems, and also work towards global harmonization, stakeholder use, and standardization of the platforms. And of course, we need to train the next generation of scientists and practitioners in tissue chip technology or MPS technology. And so with that, we actually issued a solicitation, as shown in the bottom here, to request for applications. And many applications came in. And certainly happy that Thomas and his group with Lena and Camila and others from his team really emerged as the most competitive and very top tier application, clearly above all the other applications that came in. And so here we are, at the inaugural MPS World Summit in New Orleans. As Thomas mentioned, the next one next year will be in Berlin. With Uwe Marx, who will be talking a little bit more, providing more details on that towards the end of this meeting on Friday. And then a planned meeting in Seattle, Washington in 2024. So there's more to come. Encourage the community to really get involved, who the organizers are. And then Lena and I will talk a little bit more on Friday about the establishment of an international MPS society. So thanks again to Lena for all her efforts in getting this and making it possible. And so with that, I'll end. Thank you very much.
So then thank you so much for being the midwife of the birth of MPS, which is already enduring us for 10 years or something. It's a pretty big baby, I would say. Thank you for also giving us a bit of the context, how all of this came about. The next speaker will be introduced by Norman. I hope, yes. Please take the stage. We are privileged to have a very special speaker joining us here.
Thank you for having me. I feel like in the spirit of Queen, a little Bohemian Rhapsody for the moment. Good evening. Welcome to New Orleans. I hope to see you back in 2025 as I looked at the schedule. I want to thank you all for joining us today for the MPS World Summit. My name is Norman Edward Barnum IV. I'm the interim president and CEO of the New Orleans Business Alliance. I want to introduce to you Dr. Gilda Barabino. She received her Bachelor of Science from Xavier University of Louisiana and a PhD from Rice University. Dr. Barabino is the second president of Olin College of Engineering, which is dedicated to educating the next generation of engineering innovators who recognize the needs, design solutions, and engage in creative enterprises for the good of the world. A chemical engineer by training, with broad interests in global health and interdisciplinary research and education, Dr. Barabino has pioneered new engineering approaches to problems in medicine. Her seminal research in sickle cell disease has informed current technologies and formed the basis for novel therapies. She is a passionate advocate for health equity and leads national efforts to engage engineers in the development of solutions to health disparities. Prior to becoming president of Olin College, Dr. Barabino served as dean of the Grove School of Engineering at the City College of New York and held appointments in the Department of Biomedical Engineering and Chemical Engineering and the City Universities of New York School of Medicine. There she established the Master's in Translational Medicine, a program that addresses unmet clinical needs through the integration of engineering, medical innovation, and entrepreneurship. Dr. Barabino has also held academic and administrative appointments at Georgia Tech Institute of Technology, Emory University, Northeastern University. At Georgia Tech, she served as the inaugural Vice Provost for Academic Diversity, and at Northeastern, she served as Vice Provost for Undergraduate Education. She is internationally recognized as a thought leader and highly sought after speaker, a consultant on race, ethnicity, and gender in sciences and engineering, with a particular focus on creating cultures and climates that support the sense of belonging. She has led a number of initiatives in these areas, including serving as the founder of the executive director of the National Institute of Faculty Equity. In recognition of her visionary leadership and outstanding professional achievements, Dr. Barabino has received many of the highest honors in academia. In 2021, she was elected to the American Academy of Arts and Sciences. She is also elected member of the National Academy of Engineering and the National Academy of Medicine. She's a fellow of the American Association for the Advancement of Sciences, the American Institute of Chemical Engineers, the American Institute for Medical and Biological Engineering, and Biological Engineering Society. She is president of the American Association for the Advancement of Science, the world's largest interdisciplinary scientific society. Gilda is a leader who changes the world. She is an innovator and an educator who leads by voicing what needs to be voiced, doing what needs to be done, and elevating others, individuals, organizations, and institutions. Please share with me now, welcoming my sister, my mentor, and my lifelong role model, Dr. Gilda Barabino.
Well, I must say, thank you, Norman, for that very kind and generous introduction. So good evening and welcome. I am so pleased to welcome you all to the 2022 Microphysiological Systems World Summit and to the great city of New Orleans, my hometown. The theme of this year's summit, Connect, Exchange, and Educate, are drivers that undergird creativity and innovation needed to further develop and propel the field. How we connect, exchange, and educate is important because it has an impact on who gets to innovate and invent. And that matters because who invents influences what is invented and how we best tackle problems as a global community. In that vein, I'll start with a lesson from history from the period that arguably has been considered the most innovative period in the United States and the world, the golden age of innovation between 1870 and 1940. Using patents as a measure of invention, seminal work by Lisa Cook revealed that Northern Blacks invented at the same rates as whites, equaling their share of the population, despite previously having been enslaved. Quite a feat, considering that prior to 1865 and emancipation, it was illegal for enslaved Blacks to be educated, and Blacks could not apply for or hold patents. Sadly, following the golden age of innovation, opportunities for Black inventions stalled and even reversed due to legalized discrimination impacting education, housing, economics, and advancement. And these lost opportunities continue to this day. According to a landmark study by Raj Chetty and his colleagues in 2017, often referred to as the Lost Einstein's paper, a predictor of someone getting a patent in America is less about aptitude and interests and has more to do with a person's socioeconomic class, race, and gender. Researchers linked patent records with income tax and school district records to test the theory. They found that both having high math and science scores and coming from a rich family were predictors for becoming an inventor. They also found that growing up in an area with more inventors and being exposed to innovation increases chances for becoming an inventor. They concluded that innovative output would increase if we expose more young children to inventing, particularly girls and children from historically excluded and low-income groups. At a systemic level, differential access to educational and other opportunities effectively limits innovation for the nation as a whole. The number of inventors in the United States could be 4 times higher if women, underrepresented minorities, and individuals from low-income families became inventors at the same rate as white men from high-income families. Those who do get through the gates often find their innovations discounted and have less successful careers, a challenge described as the diversity innovation paradox by the computational sociologist Bas Hofstra and his colleagues. And what's more, this system inevitably restricts the type of invention and innovation that is produced. In a recent report in Science, the researcher Rembrandt Koenig and his co-authors confirmed what's called the inventor-gender innovator gap. And they demonstrated that for US biomedical patents from 1976 through 2010, patents, this is a quote from their paper, patents with all female inventor teams are 35% more likely than all male teams to focus on women's health. Their study estimates that there would have been roughly 6,500 more women-focused inventions to help women navigate all manner of health issues and beyond if patents were invented between men and women equally from about 1975 onward. Women were represented in only 25% of patents during that time. The researchers convincingly show that women inventors are more likely to produce patents to solve problems that specifically or disproportionately affect women. So beyond recognizing the loss of human talent that arises when women are underrepresented in innovation, this finding highlights the types of problems and solutions that are overlooked in the current system within its structural imbalances. So, why doesn't the innovation system work to keep people like me inside? The reasons are complex and interrelated, from who has access to opportunities, To what conditions are created to foster innovation? To who is seen as an innovator? We have a historical legacy of not recognizing the scientific contributions of enslaved and descendants of enslaved Black Americans. Yet, Black inventors are responsible for enabling technologies that have enhanced our quality of life. Structural and social barriers, such as hostile climates, bias, and tokenism, make it difficult to achieve a sense of belonging and limit career choices and opportunities for Black students and faculty, further perpetuating the persistent underrepresentation. Scientists and engineers themselves often serve as gatekeepers in controlling who's recruited, accepted, and socialized into the profession. As Cheryl Lagon, a sociologist, says in her 1995 article on the impact of science and technology on African Americans, who does science is important for who will do science. Today, 3.9% of students in the United States who graduate with a bachelor's degree in engineering are Black, and only 4.1% of students who graduate with a PhD in engineering in the nation are Black. And I like to contextualize these statistics a bit by sharing a part of my own story. When I reflect on my own experience, I realize that I could have easily been among the missing Einsteins. I lacked Black role models and mentors in engineering, but I did have a deeply ingrained belief, instilled by my parents, that education was foundational and held the key to opening doors to my future, whatever path I chose to take. I grew up in a military family, and as we moved around, my early educational experiences were uneven, from integrated schooling in military-based schools to largely segregated schooling influenced by location. By the time I reached 8th grade, my family had settled in New Orleans, and I attended Sophie Wright Middle School and Benjamin Franklin High School. While I was at Franklin, I began taking classes at Xavier University of Louisiana, and I remained there to complete my undergraduate studies. And though I began as a pre-med chemistry major at Xavier, I later changed my major from pre-med chemistry to what was called straight chemistry, recognizing that I wasn't really inclined to become a clinician. I'll share with you, I'm pretty squeamish, and the sight of blood does not do well for me. So I ended up pursuing chemical engineering at the graduate level at Rice University. I was intent on applying engineering principles to solve problems in medicine. I chose sickle cell disease, a complex genetic blood disorder that predominantly affects Blacks, in order to give back to my community. And that's a motivation that is quite common for members of historically excluded groups. I also viewed sickle cell disease as one that sat at the intersection of race, health, and politics, and thus an opportunity for me to follow my interests in impacting health disparities and inequities. My thesis, Rheological Studies in Sickle Cell Disease, involved an investigation of abnormal adhesive interactions between sickle red blood cells and endothelial cells under physiological flow conditions, mimicking in vivo conditions. I used an endothelial line parallel plate flow chamber for my dynamic adhesion assay and produced seminal findings regarding conditions and mechanisms for adhesion that could form the basis for novel therapeutic approaches. We complemented our human-based in vitro studies with animal studies using sickle transgenic mice. And over time, we miniaturized our flow chambers and used microfluidics to further mimic physiologic conditions. And while conducting research on sickle cell disease, I didn't just consider the burden of the disease. I also considered the burden of racism and other social dynamics at play. I engaged with communities and advocacy groups understanding sickle cell disease. I wanted to understand the inequities and the health disparities impacting our ability to address the disease And I knew that to employ good science, I needed to understand all facets of the challenges I was attempting to solve. That experience, along with my own lived experience, continued to inform my perspective on the need to transform and redesign education, research, and innovation systems to include those who are traditionally excluded, and also to design responsibly with the end user in mind. We all have a shared goal to connect, exchange, and educate. As we strive to reach that goal, consider that innovation is conceived, perceived, and driven in various ways at both individual and organizational levels. How we frame innovation impacts how innovation is carried out, who becomes innovators, and what the ultimate outcomes are. In a very broad sense, innovation can be defined as a process that involves implementing new ideas to create value. Innovation thrives on access to technology, collaboration, and networks, identification and understanding of unmet needs, and market forces. However, what needs to be taken to heart and put into practice in our education systems and institutions and in the private sector and in our agencies is that innovation is also vastly improved by diversity in thought, lived experience, and team composition. We will serve our current needs best if we reframe innovation by broadening our idea of it to include diversity, inclusion, belongingness, collaboration, interdisciplinarity, intersectionality, reframing problems, and using a convergence of technology and concepts. Added to this, we must also make sure that we are doing everything we can to diversify who has access to resources that make people innovators, who is seen as a potential innovator, and who becomes an innovator. So simply put, we must change the innovation and inventor landscape, broadening the scope of those who see themselves as scientists, and ensuring young researchers in particular feel supported. So I urge you to look for ways to do so while you're here participating in this World Summit. I hope you also take away some lessons from your host city, New Orleans. Though I may be a bit biased, in my view, New Orleans is a great city. It's a city of creativity, innovation, and inclusivity. It's the birthplace of jazz. It is replete with examples of innovation and invention, including that required to mitigate being below sea level and keeping the city drained and dry, particularly during and in the aftermath of a hurricane. It's a blending pot, melding cultures and influences from around the world. It is a welcoming city. So please, I urge you, make the most and learn from this summit and from this city. Again, welcome and thank you so very much.
Thank you so much. Gilda Barberino has shown you that science and social aspects have to be seen together, that the spirit of a world conference is about inclusion, about coming together, bringing together and enabling very important concepts for our series of conferences we are starting here. And thank you very much for her own brother to introduce her. I think this was a special treat. And I also thank you for enabling the support of the city of New Orleans for this conference series. I'm coming to another brother in crime. Don Ingber actually needs no introduction. And if you need one, we have put one into our program here. But if there's anybody who has shaped the field of microphysiological systems, it is him. He and the WIS Institute in Harvard, which he directs from its inception, is a driving force in the field. You heard he was the one who hooked the FDA to invest into this on a broader scale above this initial program. He and his group have at sometimes on a monthly basis produced papers in the Science and Nature group of journals. It is most impressive. And if there's ever a Nobel Prize for microphysiological systems, I know one name among those who will get it. But at the same time, he has always been approachable, supportive, joining us whenever we had events. And I found this most remarkable in all of these responsibilities he is sharing for moving the field forward. Don, I don't want to take any more of your time. It is the right moment to open our conference with the first keynote lecture by this iconic protagonist of the field. Thanks a lot.
Well, thank you. It's a kind introduction and gratifying to see how much this field has grown since we all got started working in this area, and to see so many people who I've worked with me distributed around the world, seeing their faces again here today in person. This is my first in-person meeting in 2.5 years, so it's really quite wonderful. What I'd like to do is describe some of the work that we've done over the past 15 years or so in the organ chip area, where we have been able to recapitulate human biology, disease states, and therapeutic responses in vitro, and give you a feel. There's so much going on now, and you'll get this from this meeting, but give you a feel of our approach and why we're so excited about it. Now, I've been at Harvard for, 38 years. But for the last 13, I've been the founding director of the WIS Institute for Biologically Inspired Engineering. And the basic concept behind the Institute is that engineering has transformed medicine over the past 50 years by taking engineering principles, trying to solve problems. When we started this envisioned it about 15 years ago, we wanted to flip that on its head. We really felt that we were at a point where we now have uncovered enough about how nature builds, controls, and manufactures, that we could actually leverage biological principles to develop new engineering innovations. And this is what we call biologically inspired engineering. And we were lucky to start with a very large philanthropic gift, but we were challenged with taking on really tough problems. But if we could solve them, it would have transformative impact. And one of the biggest problems that I could see personally at the time was that the drug development model is broken. I think all of you are here because you know it. It costs billions to take a drug from the bench to FDA approval. There are many reasons, but one of them is that animal studies are required. They take years to complete. They're expensive. The reproducibility is not always so great. But the real problem is that, and there are ethical issues, but the real problem is that more often than not, they don't predict what happens when they go to clinical trials in humans. And in some areas, this is like over 90% failure, but on average, it's about 70%. So there's been searches for ways to come up with developing drugs faster, cheaper, and that are more effective and safer. And so when we started the Institute, and that's where we started. But as we got involved in this area, we've learned that it's much broader challenge than that. For example, now many drugs are biologics. Something like 40% of drugs in the development pipeline are biologic, therapeutic monoclonal antibodies, RNA therapies, CRISPR RNA is coming into mode. And some of them are so specific for human, they don't even cross-react with other species. And so right now we rely on non-human primate models, which have enormous ethical concerns. We learned during COVID that they're actually in short supply as well. But there's really a desperate need for human models, even in this case. And then one thing that we haven't talked about beyond drug development is this One Health challenge. I think Dan mentioned this briefly, which is that we now very clear that animals are often the sources of disease vectors, and COVID with bats was a prime example of that. And the way it's handled now is, they cull livestock, which is a huge economic problem, ethical problem once again, and it's not even clear it's effective. And there's no way you're going to have, labs developing animal models for every species, but preclinical models, MPS, organs on chips, that's one way you could actually confront this challenge. So when we started the Institute, we had about 5 platform areas, and I led one myself that we called biomimetic microsystems, but really it was developing what we eventually called human organs on chips. And the idea was to use computer manufacturing methods, computer microchip manufacturing methods that in 1989, 90, I started to apply to cell biological challenges working with George Whitesides to leverage these techniques in computer microchip manufacturing that had control over features at the same nanometer to micrometer scale that cells and tissues live at, to use them to create microchip-like devices containing living human cells that reconstitute organ level functions, not cellular tissue, but organ level functions. And obviously, this was to accelerate drug development and replace animal testing, but it would also be useful, it was very clear, to advance personalized medicine. And our first success, which I do think helped launch the field, or at least get recognition for the field, was work that Dan Hudd did when he was a postdoc with me, that we called a human breathing lung on a chip. Now, the idea was never to engineer a whole organ. I actually worked on tissue engineering in the 80s, had a company that did that. This was a total different mind frame. This was, I want to build a living 3-dimensional cross-section through a major functional unit of the organ. And in this case, we focused on the alveolus or air sac. And I think you all know the lung, has the alveolus is a pretty simple structure. If you go look at an electron microscopic level, there's air, there's a single layer of alveolar epithelium, a flexible porous basement membrane, and then a single layer of capillary endothelial cells, and then blood. But what this doesn't show is that it's an incredibly dynamic structure in that every time we breathe in and out, the alveolus stretches and then it relaxes, blood is flowing, air is flowing. And so if you were trying to distill down, like, what are the simplest design principles to build an organ? The first thing is that an organ are 2 or more different tissue types that come together and new functions emerge. And usually one of them is a vascular tissue. The other is that in our case, we felt that the mechanical environment was absolutely critical. So we wanted to have this dynamic flow of perfusion through the capillary networks, air, and also cyclic breathing motions. So this next video shows you how this works. At the top right is the chip. It's the size of a computer memory stick, optically clear, made out of flexible rubber. If you cut it in cross section, there are 3 hollow channels, each less than 1 millimeter wide. The middle channel is cut into top and bottom by another thin, this is PDMS membrane with engineered pores. We coat that with matrix. Then we put human lung alveolar cells on the top, human lung capillary cells on the bottom. We just recreated the alveolar capillary interface. The trick is that we have, oops, I'm sorry. I lost that one. Let's see if that works again. I'll let it go through here. I went through quickly, obviously. The middle channel has a thin membrane that has pores that actually can allow cell movement. This is coated with extracellular matrix. We have human lung alveolar cells up top, human lung capillary cells at the bottom, create the interface. And then the trick is that we have cyclic suction applied to the side chambers. Everything's flexible. This is polydimethyl siloxane, silicon rubber, and it stretches and relaxes at the same rate and degree as when we breathe. We then put air over the top to create an air-liquid interface, and then we could flow medium with or without immune cells through the bottom. And if we have endothelial cells on all 4 sides, as we tend to do now, we could even flow whole human blood without anticoagulants for a period of hours. So if this were to work, it should mimic an organ-level response. So imagine you have a bacterial infection up top. What happens is that there's a tissue-tissue signaling response. Cytokines are secreted by the epithelium. They activate the endothelium. White blood cells that were just flowing by now stick, roll, diapedese, or migrate across and then engulf because the endothelium express adhesion receptors. And now I'll show you imaging in the device. What you see here are fresh white blood cells. I always like to say we know they're fresh. They were from Dan, huh? They're labeled fluorescently. You don't see the endothelium and you don't see the epithelium because it's on the other side of the screen. To begin with, it's quiescent. They just flow by. Now, if we put bacteria on the other side, you get that signaling response. And these are pulled out under flow, which is important because the initial adhesion step for an immune cell to endothelium is shear stress dependent. And now you can do any imaging you want in the device. So higher magnification, one white blood cell right there, it finds a space between two endothelial cells that are unlabeled, goes through the matrix-filled pore, goes out of focus to the other side. Now you're going to see it by phase contrast coming up through the epithelium. And now you're going to see the white blood cells in red because the bacteria are labeled with GFP in green, and you see them being engulfed. So you just watch the entire human inflammatory response at high resolution in this little rubber chip. And I think this really got people's interest. Now, for those of you who are new to the field, I show some results from that first paper. We were actually not funded to build organ chips. We were funded on nanotoxicology. And people in the field in those days were realizing that pretty large nanoparticles were getting across tissue interfaces. It was not clear how this could work. And so the lung is a major site for this. So we developed the lung chip actually initially to look at that. And we used simulants of smog nanoparticulates that are in air. We had a fluorescent reporter for injury in terms of reactive oxygen species, oxygen-free radicals. And it was interesting that if we exposed the particles to the chip, if we had it with physiological breathing motions, we could see injury with ROS, and that actually recruited immune cells. But if we did the exact same experiment without breathing motions, just statically, we did not see this response. So if you did this in a trans well, you would not see this response. We also could quantify absorption. So you're seeing a graph at the left, but at the right are images of what's in the effluent of the vascular channel when we put the nanoparticles in the airspace. And we saw an 8 to 10-fold increase in absorption based on physiological breathing motions. Now, this wasn't mimicking physiology. This was never heard of before. This was a prediction. So we actually developed an ex vivo ventilation perfusion model in the mouse where we could separate breathing from blood flow. And we totally recapitulated that response. And this was a full article in Science. And this is really, I think, one of the first times that you could see that you could mimic organ level physiology and pathophysiology in a little engineered device. Now, we started to talk to pharmaceutical companies and they thought this was great, but they were more interested in toxicity models and also particularly disease models. That's really where the animal models were limited for them at the most. And so we hit two birds with one stone by developing a model of pulmonary edema, fluid on the lungs, by perfusing the FDA-approved cancer drug interleukin-2 through the vascular channel, and it's normally infused in a patient, IV. And it's known in patients that the dose-limiting toxicity is pulmonary vascular leakage and pulmonary edema. At time zero, with air above the epithelium, this is what it looks like. When you perfuse the interleukin-2 over 2 to 4 days, you see a meniscus of fluid filling the upper space, and it completely fills it by day 4. This is the same time course and the same dose that is seen in patients. Once again, when we quantify this, we only see this with breathing motions. We do not see it under static conditions. And again, we confirmed that this is true in vivo as well. And this is important clinically because most patients with pulmonary edema are on a ventilator where you can actually tune the physiological breathing motions and ventilation pressures. Now, this finding that mechanical deformation was important for this toxicity got my interest because my entire career, and we're talking now starting like my first paper was like 78, has been that mechanical forces are as important as chemicals, genes, which is now known as mechanobiology. And we'd worked on mechanotransduction for years, and I had identified one of the earliest signaling response that we could see in endothelium when you stretched the extracellular matrix was forces going through integrins to an ion channel called TRPV4 in the focal adhesion in 5 milliseconds. And that induced calcium influx, and it was upstream from all the other integrin signaling responses. And I knew that GlaxoSmithKline was working on an inhibitor of this. And they did not fund our work, but they gave us the drug, and we completely inhibited pulmonary edema in this chip with that drug. They, in parallel, did studies on, I think, dogs and rabbits in a cardiogenic pulmonary edema model. This is failure due to back pressure when the heart fails. And they got the same inhibition response. And we had back-to-back papers in Science Translation Medicine. Now, I'll never forget, we had one reviewer who said, this paper should not be accepted because it's too simple. There are no immune cells. And the other reviewer said, this is amazing because this paper is like synthetic biology at the cell, tissue, and organ level. They just showed you don't need immune cells for pulmonary edema induction by the drug, and it got in. And we have seen this again and again and again. And this is something we don't talk about enough about in MPS and organs on chips, is the ability to control individual control parameters, one at a time, 2, 3, 4 cells, forces, factors, gradients, et cetera. And when you hear about the interest in mechanism of action of both drug efficacy and toxicity, this is when these systems provide enormous value. So this one model, this long alveolus chip, has provided proof of principle for a disease model, a drug toxicity model. We've also published papers where we've shown toxicities with microfluidic organ chips that were never seen in animals, but for example, a lupus drug, a therapeutic monoclonal that no problem in animals, but there was pulmonary embolism in patients. We could replicate that on chips. Drug efficacy, therapeutic target discovery, new drug discovery, that drug, the TRPV4 inhibitors in phase 2 clinical trials, if not further. We've also done gene therapy delivery, adenoviral vectors on these chips. And it's won numerous awards. I am most proud that it won the International Design Award and beat out the Google car and a Frank Gehry building that year. But my family is most excited that it's now in the Museum of Modern Art's permanent design collection, which means I'm a permanent member of MoMA in New York City, and I get permanent discounts in the cafeteria and the gift shop. Now we've integrated immune cells. I showed you, we've also integrated stromal cells, like when we do pulmonary fibrosis models. And then we started to build other chips. So different pharma companies said, we're interested in asthma and COPD. Those are bigger challenges for us. And those are diseases of the airways. So we then made chips, this is Cambez Benham's work, where we took primary human bronchial epithelium on the chip, and we differentiated for 3 weeks under an air-liquid interface. And we've later put endothelium on it as well. You get beautiful differentiation. Cilia move in a directional manner. If you put fluorescent microparticles to quantify mucociliary clearance, the bottom right, those particles are flowing in the mucus on chip at the same exact rate as in all of our lungs at this very moment. We then made chips with cells from patients with chronic obstructive pulmonary disease, COPD. And even after differentiating for 3 weeks, they retained the COPD phenotype, which is measured here by decreased toll R3 and 4 receptors. Now, what brings COPD patients to the emergency room are usually exacerbations, either by viral infection, bacterial infection, or cigarette smoke. And here we mimicked viral infection with poly-IC and bacterial infection with LPS endotoxin. And we could see that no response in healthy chips, but the COPD chips responded either to the LPS or to the poly-IC. And then Cambez, working with our engineers at the Wyss Institute, built a cigarette smoking robot. This is a Gatling gun of real cigarettes, a cigarette lighter from a car. It actually mimics true smoking parameters, meaning pauses, breaths, intensity, and timing. And now it delivers real smoke, not extract, right to the airspace. Now, this is just 2 simple examples, but on the left, you see IL-8, a major inflammatory cytokine. And I'll go over here because I can't see that, but on the left is normal. There's no change in response to cigarette smoke. On the right, you have a doubling of IL-8 in these COPD chips, which is very much like in patients. At the right, you see a transcriptomic analysis, a gene microarray. The 3 lanes at the far right are from 3 different healthy donor chips after exposure to cigarette smoke, the changes in gene expression versus before. The 9 at the left are from a past clinical study of healthy people who also smoke cigarettes. We were really excited because we got pretty similar pattern in the top quarter versus bottom three-quarter of up and down expression. The reviewers were not impressed. They were very upset because ours were too similar to each other. We weren't mimicking the variation in patients and the intensities and so forth. And then we realized that we actually can do this type of study better than a clinical study because we were doing matched comparative modeling of the same patient before and after cigarette exposure, smoke exposure. So those changes are due to cigarette smoke, whereas the changes in these patients are due to cigarette smoke, but also work history, home environments, other things that they've done in their lives, and that eventually was accepted. And then we were actually asked to write a little commentary on how to do matched comparative modeling in vitro. Recently, Roberto Plebani was on sabbatical, and we got, with funding from Cystic Fibrosis Foundation, we were able to get cells from patients with cystic fibrosis. We put them on the chips, and at low mag, you could see there are more cilia here across the entire length of the channel. We also found that the beating frequency was increased versus normal, which is what's seen in patients, but you do not see this in a static transwell. You have to have flow. We saw increased mucus production as in patients, increased baseline inflammation in terms of immune cell recruitment, increased cytokine release. And then we looked at microbiome. We looked at Pseudomonas aeruginosa, which is in everyone's lungs, but it overgrows often in patients with cystic fibrosis, major cause of morbidity and mortality. And again, on the chip, we see much higher growth in these chips than in vivo, than in conventional systems. So one thing I want to emphasize, and I think so far today in the meetings I've been at, we're all focused on drug development, and that is basically how we all started in this field. I'm presenting our two-channel microfluidic model of MPS, but you're going to hear in this meeting there are so many different versions of this now. Each has its own strengths and weaknesses. But what we have found is that the real strength of organ chips and MPS is that they provide a window on molecular scale activities inside living human cells within a relevant tissue and organ context. And so this means that it actually can enable new mechanistic insights as well as drug discovery. And at the same time, you have an experimental system to test the predictions that you come up with that's human relevant. I think this is really the Intel inside is like the power of the whole of this whole technology. Now, a few examples. Brian Hassel was a grad student. I've worked on cancers since 1976. And big advance in that field was going from subcutaneously implanted tumors in mice to what it was called orthotopic cancer models, where you now take a mammary tumor and you inject it in the mammary fat pad, or a prostate tumor and you put it in the prostate gland, and it grows a little bit more like in human. So we had these human chips. So we said, let's make human orthotopic cancer models. So we took a human non-small cell lung cancer, cell line H1975 that a lot of pharmaceutical companies use. It's an adenocarcinoma form of this cancer that in patients emerges in the small airway, but it grows preferentially in the alveoli. And so we cultured it in the airway chips and the alveolus chips. We plated the cells at a low starting ratio to normal from the beginning. And what we found is quite interesting. On the right, the top is airway, the bottom is the alveolus. The gray dots show you the growth of these cells the way they've always been grown for 50 years in plastic dish, serum-containing medium. They grow rapidly. The white circles is a plastic dish with the medium we use on our chips, which we only feed through the vascular channel because there's air above. There's no growth of these cells under those conditions. So all the growth I'm going to talk about is due to tissue-derived microenvironmental factors. Okay, so what we find on the airway chip is they don't grow at all for about 10 days, and then they start growing. And I will add that the airway chip is thick enough that we could go in with a needle and inject the tumor cells after the chip is formed, and there we get dormant tumors. But when we culture them from the beginning on the alveolus chip, they grow rampantly from the start. Now, what was really amazing is we did it with or without breathing motions, and breathing inhibited cancer growth on this chip by 50%. We could also measure invasion very nicely because you could just have GFP labeled tumor cells and watch them go through the pores, as you can see at the bottom. And we inhibited invasion by 50%. And using this system, we could show that the breathing motions regulate the therapeutic response of a first-line drug called ruxolitinib, a kinase inhibitor, such that when you treat the cancer in the alveolus chip without breathing, you have potent inhibition of growth. With breathing, it grows much more slowly, but it just keeps growing right at the same rate through the drug treatment. And we could show that this actually is regulated at the level of EGFR receptor phosphorylation. Now, we then started working with real viruses, and we were funded by NIH and DARPA. And the reason we were funded is that this is about 5 years, in 2017, there was enormous interest and concern about potential future pandemic respiratory viruses. But the focus was on influenza. So we developed a model of influenza infection. We took our airway chip. This is GFP influenza H1N1 at the right. You could watch the infection in real time. You could show that you get disruption of the epithelial junctions, cilia. You even get endothelial damage, which hasn't been studied before. We could use different viral strains, H1N1, H3N2, H5N1, and we could show that we get similar virulence as seen in patients. I don't have it here, but we've done it in COPD chips, which are 10 times more sensitive than healthy chips, which is just as seen in patients. We could quantify barrier function and show it's compromised. But more importantly, we could measure the host response. And this is essentially a cytokine storm that we hear about in COVID all the time. This is 6 different cytokines. The different strains of influenza induce cytokines to the same sort of levels that they do in patients. We could watch immune cell recruitment, as I showed you in the movie, but now we could watch them actually clearing virus from the channel above. And we could also measure drug effects. And in this case, we use Tamiflu. It's also known as oseltamivir, the first-line antiviral. And you have potent inhibition at a clinically relevant dose because we can flow drugs through at the clinical Cmax. Not bathe the cells in it like in a static dish. We also did a really interesting experiment. Long C, who was a postdoc in my lab, basically mimicked human-to-human transmission of virus and showed that we could model spontaneous viral evolution of variants of concern, if you like. Basically infect the virus, in this case, influenza, and treat it with a drug at 90% efficacy. And here you see amantadine. And then after a couple of days, you take a droplet and wash it through the airspace and move it to another chip, just like you were coughing. And we do this again and again and again. And what we find with amantadine is that 8 passages, we get virus that's completely resistant to drug. When we do genome sequencing, we find all of the known mutations that have been published in patients who became resistant to amantadine clinically. And we identified about 3 mutations that no one's ever seen before. So that means you have the potential to see what's coming down the pike, which could have impact on vaccine development and therapeutic development. We did this with Tamiflu. It took 25 or 28 passages, but we got resistant virus, which is terrifying. We even got virus that was resistant to both, which is even more terrifying. Now, this is where we were. I'm sorry. This is where we were. We started for the first time saying, could we use these chips to repurpose existing approved drugs for new applications so that you could get past the drug development 15-year timeline and do things quickly. And we went to the data that was published from Kamba's work in the smoking chips. And we noticed that there was many serine proteases that were upregulated. There was literature to suggest that they may be involved in the infection process. We tested 2 existing drugs, nafamostat, which is a anticoagulant, and trasolol. Both of them are broad spectrum proteases. And we had potent inhibition of H1N1, H3N2 on the chips. And interestingly, we found that if we take one of these drugs, nafamostat, and combine it with oseltamivir, Tamiflu, we could double oseltamivir's therapeutic window from 2 days to 4 days. And this is important because FDA has only approved this drug to be administered in the first 2 days after you get influenza, which it usually takes more than that to diagnose that you have influenza. And so this actually could have clinical relevance. Now, this is where we were when COVID hit, January 2020. And Long Si and Haiqing Bei are two postdocs who were trained as virologists in China and worked in my lab. They were following this on social media. The day after they published the gene sequence in January for SARS-CoV-2, they synthesized and designed and had synthesized engineered pseudovirus that expressed the SARS-CoV-2 spike protein so that we could use it in our BSL-2 lab. We don't have a BSL-3. They also quickly, and if you remember, those were desperate times, they went to the literature and they found 30 drugs that had shown some activity against SARS-CoV-1 and related viruses and Ebola. And they tested them, but they quickly tested them in an HUH7 cell line, which they had used when they were graduate students in virology, which is like the Vero6 line, is used because viruses grow well in them, likely because they have abnormal protective interferon responses. And out of those 30 drugs, as shown here, there were 8 that give you dose-dependent inhibition of viral infection in the low micromolar range, shown in gray bars. But what you might not be able to read is that 2 of these are chloroquine and hydroxychloroquine. This was February of 2020. Then we decided to test this in the organ chips. The first thing we did is we wanted to confirm that they express the ACE2 receptor and TMPRSS2, which is a serine protease that we now know is very important for SARS-CoV-2 infection. We had high levels of expression. Now we tested those same 8 drugs. And what we found is that chloroquine and hydroxychloroquine had no effect, nor did a number of others that eventually went to clinical trials. We found 3 compounds that did have some activity. The most potent was amodiaquine, which is another anti-malarial used for 50 years in Africa for prophylaxis. And we had funding from DARPA to expand this and collaborate with Ben Tenover at Mount Sinai and Matt Freeman at the University of Maryland, who have BSL-3 labs. And with Ben, we used a hamster SARS-CoV-2 model, and we could show that we get potent inhibition whether we give the drug prophylactically or after infection. And this drug, in part because of these data, is now in clinical trials across 20 sites in Africa. Separately, Haiqing in the group was working on the alveolus chip, and he was working on H3N2, and he did our classic experiment of breathing, non-breathing, and surprisingly, he found that breathing suppressed viral infection significantly. It suppressed cytokine production significantly. He did RNA-seq and many other studies that were published recently in Nature Communications, and he found that the one gene that was most highly expressed looking at breathing, non-breathing was S100A7. And this was important because there are S100 proteins that are related to this that were published, that were shown to scale with morbidity and mortality in COVID-19 patients as well. And it binds to a receptor called RAGE, it's receptor for advanced glycation end products, which is known to play a key role in inflammation. And we found a drug called aziliragon that's a RAGE inhibitor, and it potently inhibited the cytokine storm, basically, completely in our models. And this drug went through phase 3 clinical trials for Alzheimer's, oral, highly bioavailable, totally safe. They were like no side effects, but it missed its mark. And we reached out to the companies that have control of it. And this drug, the data shown here were included in an IND application to the FDA by a company called Cantex Pharma, who has control of this to start COVID-19 trials. So we're really hopeful that this will move along as well. Now, we've made many different chips. We started doing a human intestine chips in 2012, where we used CACO2 endothelial cells, an intestinal cell line that pharma's used for years. But I'm sorry, let me go back here. Yeah, now CACO2 epithelial cells, pharma uses them on trans walls, takes 3 weeks to get a poor barrier. They grow like flattened cells. We use the same medium that's been used for 50 years to culture them in trans walls and on 2D dishes where they have poor differentiation. And with peristaltic-like motions and trickling flow, we get spontaneous formation of intestinal villi in 5 to 7 days. Now we use organoids from primary from patient-derived organoids. We expand them in 3D gels. We then remove them, fragment them, place them on the chips, and we get absolutely beautiful intestinal villi. But now we can measure absorption, transport, mucus production, microbiome, et cetera. And this work was done by Magda Cassandra, who is here, and Alexandra Sontheimer Phelps and Youngjung Kim. Now, if we go back to the original studies with the Keiko-2, again, this originally came from a tumor. They grow rampantly in a poorly differentiated state on a plastic dish. Just giving them peristaltic-like motions and flow, they form villi. The proliferative cells move to the crypts, the bottom with EDU labeling. You wash out EDU, they migrate up to the top. You get all 4 lineages of small intestine. For the first time, they produce mucus. They have P450 activity. The only change was the physical, microfluidic, dynamic environment and peristaltic-like motions. We also found that, that bacterial overgrowth was regulated by the peristaltic-like motions. When I went to medical school, you're taught the patient is anesthetized. When they come out, you want to get them drinking quickly and eating so you don't get ileus. Ileus is bacterial overgrowth that leads to sepsis and death. Medical textbooks say it's because you're stopping flow. Well, we keep flow going. It's the peristaltic motions that stop, which you cannot separate in vivo. We can separate them on the chips. When we did, Magda did the primary organoid-derived chips, we compared the transcriptomic analysis of the organoid from the duodenum, the chip, which is the middle lane, and the in vivo duodenum, and we could show that the chip is closer to in vivo than the organoid from which it was derived. Alexandra made chips with colonic organoids, and this is a cross-section through the chip that shows you the morphology is amazingly reminiscent of what you see in vivo in goblet cells with magenta and apical microvilli. But now we could study mucus production, which is very important for ulcerative colitis, cancer, et cetera. And the cool thing is you could turn the chip on the side, visualize with differential interference, and you could see this white fuzzy stuff is mucus. And you could do this in living cultures over time. You just take it out every day, put it on its side, image it, and put it back in. And for the first time ever, we recreated the bilayer structure of human mucus and the thickness that is seen in vivo with a penetrable and impenetrable layer. Now, I think one of the biggest advantages of microfluidic systems is that we can culture living microbiome in direct contact with living human cells for extended times. You can do them in organoids for about 24 hours. You inject them with a needle and the lumen and then they die, but we can do them for multiple days. And we published, Sasan Jalali Farizenhad was the first author on this work, where we created a hypoxia gradient by having oxygenated medium through the endothelial channel and nitrogen gas around it. And we can maintain oxygen levels below 0.5%, which means we can sustain obligate anaerobes in the lumen of the chip. We have oxygen sensors we integrated in the chip so we can measure this in real time. We've also integrated electrodes to measure tear in these chips. And so you have a gradient so that you could have obligate anaerobes in the middle, a mixture of obligate anaerobes and aerobes, and then you could keep the human cells alive as well. And with this, we published that we could take literally a stool specimen, collect a sample, dissolve the bugs out, put them on the chips, and we can maintain similar complexity as in human stool. This is over 200 different strains of bacteria from 11 different genera for 5 days on these chips. And the chips, if it's healthy from a healthy patient, the chips actually have a better barrier function. The right, it's hard for you to see, the little speckles are actually microbes. Now we have a paper that that's in press at Nature Biomedical Engineering, where we're modeling environmental enteric dysfunction. This is malnutrition in low resource nations. And this has many factors, including malnutrition, as well as microbiome, as well as pathogens. And again, we can control these individually. And our initial results, we were able, with Gates Foundation funding, to get organoids from children with EED in Pakistan. They sent us the organoids and we put them on the chips compared to healthy. And the first thing we could see is that If you have healthy versus EED chips, we see decreased absorption of fluorescent fatty acids and proteins. What we do is we can culture these chips in complete medium or nutrient deficient medium, niacin and tryptophan deficient medium. And what we found, interestingly, at the right, this is transcriptomic analysis, we were lucky to have transcriptomes of the patients where we got the organoids from, and this, high and low different genes, which you obviously can't read. But what we compared were healthy versus EED chips in complete medium or nutrient deficient medium. And the net sum, as shown at the right, is only when you have the EED chip with nutritional deficiency do they begin to get more red at the top and blue at the bottom. And now we're beginning to integrate microbiome in these as well. And we actually have been able to model microbiome dependent effects. I don't have a picture here, but you get villus blunting, you get barrier disruption, you get increased inflammation on these chips as well. Another example about discovery is Alessio Travaglieri is here at Roche now. When he was a grad student with me, we asked the question of why are humans more susceptible to enterohemorrhagic E. coli than mice? And you could have a small number of these bugs in a human and you get sick, and mouse can have orders of magnitude more and they don't notice it. And so we collaborated with Lacroix's lab in Switzerland, where they grew bioreactors with human versus mouse microbiome, complex microbiome, and they just gave us metabolites. And we tested them on our chips, and these are colon chips. The yellow are stained cells, and what you could see here is if you have the E. coli with human metabolites, you get desquamation of the epithelium, which is loss of yellow, whereas much less on the mouse chips. And we could then use this system with metabolomics to identify pretty quickly 4 specific metabolites that were responsible for this. Unfortunately, they were 4 in human microbiome that increased injury as opposed to 4 in mouse that we could add to human to protect. But it just shows you the discovery capabilities of these systems. So now there are other key goals that we've uncovered in the field, which I think remain for all of us. For example, we want to predict drug responses to drugs with clinically relevant doses. Right now, most publications on drugs in vitro bathe cells at high concentration for extended times, where in patients you have drugs go up and down, pharmacokinetic profiles, and there are clinical Cmax doses that our tissues see. That's not the way it's usually tested in vitro. We want to develop personalized disease models for individuals, create models that replicate complex immune responses, and also recapitulate species-specific effects. And so David Chu and my group developed a human bone marrow chip where we take CD34 cells from patients, bone marrow or blood, put them in a matrix gel in one channel with bone marrow-derived stromal cells, and then we put endothelium in the other and just perfuse through the vascular channel, and we get nearly normal neutrophil and erythroid differentiation over a month. This is a Guimes stain at the right where you see multiple different cell types. We maintain the progenitor cells at much higher level than conventional cultures. We were funded by the FDA actually to do this, to look at radiation countermeasures and radiation toxicity. And here what you see is that we could replicate radiation-induced injury, but we do it at a clinically relevant dose, which is not what you see in animal models. Here you're in the 1 to 2 gray, and each dot is a different color. You probably can't see it, but these are 6 individual patient donor chips, and we get excellent reproducibility. We then used 5-fluorouracil, which is a cancer drug that causes marrow toxicity. Again, publications, marrow cells in culture, you see toxicity, but high dose, 5 days. In patients, they see it they perfuse patients IV 4 micromolar for 2 days. And when we do that on chips, as shown by this highlighted color, we see toxicity on the chips at that dose, but not in suspension culture or static cultures. And at this point, AstraZeneca came to us because they had observed a really peculiar regimen-specific toxicity in clinical trials. Very unusual for a pharma company to come to an academic when they have something in clinical trials. They had a cancer drug that, if given over 2 hours, you induce neutropenia and anemia. Same dose over 2 days, you only get neutropenia. Couldn't model it in animal models easily. They had the pharmacokinetics shown in gray at the top left, meaning they did mass spec to measure blood levels of the drug in their patients. And because we have flow, we could precisely mimic the PK in our chip, and we precisely mimic the difference in regimen-specific toxicity. Now, we use these chips to develop a personalized model with children at Boston Children's Hospital who have a rare genetic disorder called Shwachman-Diamond syndrome, where you have abnormal blood cell formation. And what you could see at the low mag is there's less cellularity in the chips, in these SDS chips. You could see quantifying less neutrophils, less red blood cells, less CD34 cells, and we saw abnormalities in neutrophil and erythroid maturity. But what was interesting is that we identified a phenotype never seen before, which was abnormal neutrophil maturation, low CD13 to C16 ratio. And this was never seen before. And so we went back to the patients. We only tested 2 patients, went back to 8 patients, analyzed their expression, and found that there's 2 subpopulations in these 8. We happen to get 2 out of the low ratio, but there's high and low ratio. We're not clear what this means, but we get basically insight into etiology of disease using these chips. Now, in COVID, this was the cover of New York Times Sunday section about a year ago, and it said there must be a better way because there was a shortage of monkeys to do vaccine testing. And it's like, wouldn't it be nice if you could just do this on human cells in chips? And so Garija Guayala in my lab did this. This was published recently in Advanced Science. We just simply take peripheral blood B and T cells, concentrate them in high concentrations, equal ratio in a matrigel in one channel of our chips, just perfuse medium through the other channel, and we get spontaneous aggregation of cells into clusters. And if you give an antigen, you get germinal center-like formation that has all of the appropriate cells. We literally took flu zone influenza vaccine that you could buy in a pharmacy, that we vaccinate the chips, we have dendritic cells there, and you get germinal centers, you get plasma cell formation, you get antibody class switching, you get specific IgG against the hemagglutinin antigen in the vaccine, and we could collect effluent and measure cytokines, and we were lucky that David Walt at the Wyss Institute happened to have clinical samples, and we could show very similar cytokine profiles in our chips versus humans. And we are now using this with GSK and other Gates Foundation and other companies to look at adjuvant testing, which can be modeled very nicely on these chips, as well as new vaccine development. Now, there were questions earlier about animals, and early on, when we were still at the Wyss, before we spun this out, Janssen Pharmaceutical and then AstraZeneca came to us and said, will you make rat, dog, and human liver chips? And we were really focused on no animals, human, but they explained that FDA often requires rodent and dog preclinical studies for drug-induced liver injury, and they often get conflicting results. And so we did this. This eventually moved to Emulate, the company we spun out, where most of the work was done by Kyunjin Zhang. But this was published a couple of years ago. And what you see here is one drug, bosantin, human, dog, and rat. These pharmaceutical companies knew the results already in animals, different species, and humans. And we precisely mimicked the toxicity in these 3 species. We also did multiple other drugs, and we were able to model cellular injury, steatosis, cholestasis, and fibrosis. But what I think is even more exciting, and this is largely done at Emulate, Lorna Ewert, who's here, and her team, is that Emulate has done, and they're now over 800 human liver chips. Each one of them has 4 cell types: primary human hepatocytes, liver sinusoidal endothelium, stellate cells, and Kupffer cells. And they tested 27 hepatotoxic and non-toxic drugs that were identified by the Pharma IQ MPS consortium that you heard mentioned earlier, which are pharmaceutical companies that have come together, and they published a paper that identified these 27 drugs. We know what they did in animals. We know what they did in humans. I think 17 or 18 had no toxicity in animals, but did in human. And if you could predict these better than animals, Now we have something to talk about. So the results are pretty impressive. The chips predicted human drug-induced liver injury with a sensitivity of 87% and a specificity of 100%, which is about 7 to 8 times better than animal studies. And this has now been done in 3 different donors. I hope that this is the beginning of a change of how we do things and really reducing animal tests for a specific challenge. So as you heard at the Wyss Institute, we have, I think we're over 15 or 20 different organ chips between my group, Kit Parker, Chris Chen, Jennifer Lewis, and others. But many years ago, in 2012, I had a review that said, because we have endothelium line channels, we could collect them fluidically to create a human body on chips that would have a more physiological flow of compound across tissue-tissue interfaces in and out of tissues, which is really what PK is all about in the body. You can imagine an oral drug going to the lumen of the gut chip, watch it be absorbed, measure it be metabolized by the liver chip, peed out by the kidney. What does it do to the heart or bone, or an aerosol drug in the lung? And we eventually did this with funding from DARPA. Richard Novak led the engineering on this. We don't connect by tubing because there's just too much dead space and it's too rigid. We use a robotic sampler, liquid handler, and that could interact with the pharmaceutical industry as well. And you could go drop to drop from chip to chip. You could change the order. You could take 1 drop for mass spec to measure drug levels or cytokines, and then the next goes to the chip. And in this published study, we had, I think, 8 chips: gut, liver, kidney, heart, lung, bone marrow. But I can't even remember anymore what we had. We had brain neuronal networks from Kit Parker, and skin. And we kept them alive on this instrument for a month. But more importantly, the goal of the DARPA grant was to predict, based on in vitro data from chips in combination with computational modeling, human drug pharmacokinetic parameters, not correlate, quantitatively predict. That was the goal. It was a classic impossible DARPA goal. We did this by making a simplified human body on chips where we just made a first pass model of gut, liver, kidney, in this case, proximal tubule, and then we integrate an arteriovenous reservoir to mimic blood mixing when heart pumps. So we could take samples from the reservoir, and it would be like a peripheral blood sample rather than measuring a level after one particular chip or the other. And what you want to do here is to come up with parameters like the Cmax, the maximal peak drug concentration in blood or plasma, and the half-life. And we did this, and this was published. Anna Herland, who you heard speak earlier, led this work. We did 2 different drugs, oral nicotine and intravenous cisplatin. We had gut, liver, kidney, and this reservoir. Nicotine through the lumen of the gut chip, we measured with mass spec drug levels at all places. We collaborated with Andre Prequis at CFDRC, who's done physiological PK/PD modeling for pharma for years. So they had all of the blood flows and the masses of different organs. We just scale up from our little chips. And as on the right here, you can see that the dotted lines in color are predictions from the computational model. The dots with error bars are from a past study published in Sweden of nicotine gum versus 2 different forms of chewing tobacco called snus at 3 different doses. And we precisely predict the PK parameters there. And we did the same thing with IV with cisplatin with 2 different doses published in the past clinical study. So to summarize, we really believe that these chips that have 2 channels with an endothelium lining 1 faithfully recapitulate human pathophysiology, mimic human responses to drugs and radiation using clinically relevant exposures, quantitatively predict human drug PK parameters, and thus we think that they're really, we believe, ready to replace some animal models, both in the labs and in the drug development pipeline. And so, to end, I mean, these have many uses. They are this synthetic biology at the cell, tissue, and organ level. You could ask, is one cell type sufficient to mimic clinical response? If yes, you don't need all the rest. You want the simplest system that maintains all functionality that you're interested in. And this is what I call like one molecule of problem. Go further, and now you're down to atoms. But if you could do it with one cell type, God bless you. If not, something's missing. You add back endothelium, you add back immune cells, you add back microbiome, and you can control each parameter individually. I had a recent review of those interested in Nature Review Genetics. I think I have an 8-page table with 8 pages long summarizing, updating the field, and I know I missed a lot, I'm sure. But I ended with, the implications for personalized medicine. Right now, we have iPS cells and organoids that come from individual patients. We have primary cell lines. We can make single chips. We can make multi-organ human bodies on chips. We can model rare genetic disorders. But you can now imagine doing things like, toxicity testing, which would be, you can't do a clinical trial, for example, with lethal radiation, or it's very difficult to do trials with pregnant women. You could test male versus female. Clinical trials don't represent them equally. You could do different ethnic groups. You could do children versus old people. But you could also do what I think may be really the game changer. Right now, big pharma usually spends millions of dollars to do big clinical trials, and they almost always fail. And then they use statisticians to find out, is there like some subgroup, maybe a genetic subpopulation that responded better? And if so, they do a targeted study, and if they're lucky, they get approved for an hour application. With organ chips, iPS organoids, you can imagine flipping this on its head. Imagine you have a small group of, let's say, Hispanic women with COPD who are ultra sensitive to cigarette smoke. You get 50 of them, you make chips, you make their lung chips, their liver chips, their kidney chips. You find out which drugs work best for them in terms of efficacy, which are least toxic. And now you use those 50 patients for a targeted clinical trial. I think it would decrease the cost, shorten the time, increase the likelihood of success. And I think this is a different way of thinking about drug development. So that I must end, and I must disclose that as a Harvard faculty member, I have to disclose I'm a member of the board, chair of the SAB, and hold equity in Emulate, which started in 2010 with our science paper and now has automated systems so that hopefully people get the same results wherever they are around the world. And I welcome you to visit their booth here. And thank you so much.
I think I did not promise you too much. Incredible portfolio of any organ, any disease you can imagine, adding to it every year with a high-level publication or a bunch of high-level publications, actually. I think this was really incredible start for us. And now it's time for party. I think everybody is waiting for this. Now the only organ to be perfused is our liver, I assume. We will have a welcome reception in the Mark Twain courtyard. Please come over. I just did read a book of quotes from Mark Twain. So I thought I will read you 3 of them, which I think fit to our work. The first one is, continuous improvement is better than delayed perfection. Fits our business, yeah. The second one, the dictionary is the only place where success comes before work. And the last one, the secret of getting ahead is getting started. So get started for the party.